Hello and welcome. I'm joining you this morning from the land of the Ngunnawal people, the traditional owners of the Canberra region. The Academy acknowledges and pays respects to the traditional owners and their elders, past and present, of all the lands on which we operate, live and work. They hold the memories, traditions and cultures and hopes of Aboriginal and Torres Strait Islander peoples of Australia. Thank you for joining us. I'm Paul Richards, the Director of Communications and Outreach at the Australian Academy of Science. Today, as we did yesterday, we'll hear from fellows elected to the Australian Academy of Science in 2020 and 2021. Traditionally, we would all be in the Shine Dome together to hear these presentations. But today, I'm here in the Ian Walk Theatre with Academy staff spread across the Shine Dome to deliver this event in a COVID-safe manner. It's our hope that at some time in 2022, we can be here again together in the Shine Dome, the home of science, to celebrate scientific excellence. And so our new fellows can sign the historic charter book that's been signed by every fellow ever elected to the Australian Academy of Science. Even though things look a little different, we're so grateful to be able to still recognise Australia's scientific excellence in this virtual way, with the knowledge that we can share the experience with larger audiences than ever before. We thank you for joining us today. And we also thank our event partners who continue to support the Academy and have helped us deliver Science at the Shine Dome 2021 online this year. A big thank you to all our partners on the screen, but in particular CSIRO and DST, our platinum partners. Now, before we hear from our new fellows, please welcome Academy President, Professor John Shine.
I too am delighted to be here today for the presentations from fellows that were elected in 2020 and 2021. I'd like to add my thanks to the event partners whose generosity has made this online Science at the Shine Dome possible and allowed it to reach even more people across Australia and around the world. As mentioned, today's event would normally be held in person at the iconic Dome in Canberra. Though the location is different this year, what remains is the same, is that we are here to recognise and celebrate the extraordinary scientists elected to our fellowship in 2020 and 2021 and to celebrate scientific excellence in Australia. They were certainly humble beginnings when 23 founding fellows gathered in 1954. I doubt that they would have imagined a day when 873 scientists had been admitted to this institution. With 46 new fellows and 4 corresponding members elected in 2020 and 2021, today we boast 573 living fellows with a further 33 corresponding members. And of course, we pay tribute to the contributions made by those who are unfortunately no longer with us. While the founding fathers were all men of Anglo-Saxon descent, the modern Academy is a diverse and vibrant fellowship of women and men aged from 39 to 97, highly regarded scientists who have already left a lasting impression in their respective fields. The Academy is its fellows, living and deceased, who in so many ways have fundamentally shaped Australian science. They volunteer their knowledge and their expertise to bring science to the service of the nation. I would argue that few, if any, other organisation in this country brings together such a distinguished group of individuals who have contributed so much to society and who have been driven by public good. They truly are a unique national treasure. It is the fellows who enable the Academy to achieve its objectives of providing independent, authoritative and influential scientific advice, of promoting international scientific engagement, of building public awareness and the understanding of science, and of championing and celebrating excellence in Australian science. Without our fellows, none of this would be possible. In fact, of course, the Academy wouldn't even exist. Our fellows are involved in and contribute to the Academy in many different ways. By serving on the National Committees for Science, which are the national standard bearers for scientific disciplines. By providing expertise and gathering scientific evidence to influence decision makers, the national dialogue and academy activities. By contributing to the selection of new fellows and awardees. By representing Australia internationally. By ensuring the public can receive accurate and trustworthy scientific material online, in schools, or wherever they may be. This list is by no means comprehensive, as the range of activities the Academy leads and is involved in is very rich and diverse. Today, we're here to hear from 49 of these extraordinary people who continue to carry out, practice and advocate for scientific excellence in Australia. The annual New Fellows presentations are a unique opportunity to capture, in the course of one day, the depth and breadth of scientific excellence in Australia and to take a glimpse of the next frontiers of knowledge creation. I very much look forward to learning more about this work. So thank you all for joining us today to hear from our recently elected fellows, and we certainly hope you enjoy their amazing presentations.
Thank you, John. And we thank our partners who have helped make this event possible. We'll now hear from one of our platinum partners. Please welcome Chief Defence Scientist and Academy Fellow, Professor Tanya Munro.
Hello, my name's Tanya Munro and I'm Australia's Chief Defence Scientist. And it's my absolute pleasure to have a chance to speak with you today. Firstly, I'd like to offer my very warm congratulations to each and every one of the new fellows of the Australian Academy of Science. This is an outstanding achievement, recognition of excellence and contribution, and I hope that you are celebrating this wonderful milestone in your career. I still remember very clearly the day I got to sign the New Fellows book back in 2012. And I'm very proud to be a Fellow of the Australian Academy of Science. Today, I'm speaking to you about why Defence Science and Technology Group is partnered with the Academy, and to give you a sense of why excellence in science, such as you represent as new fellows, is so critical to the Defence mission. The strategic context in which we are living is changing rapidly. And one of the key driving factors in that contested environment is the rapid acceleration of science and disruptive technologies. In Australia, we have some quite extraordinary minds doing wonderful things. But we're at a point of time now where we need to get much more sharp and focused at how we can shape and harness the strengths we have in Australia's research and development in order to secure Australia's interests into the future. Within Defence Science and Technology Group, we have the lead for Defence in shaping up the research and development across our nation and with our international allies for the defence and national security of Australia. And last year we launched the strategy More Together, which, as the title describes, is about partnership, is about building scale around key areas of focus so that we can have impact. Those areas of focus are our star shots and our major defence acquisition programs. What the star shots do is articulate some big, hairy problems that we know we need to solve and which we can't solve with current technology. My mission is to try and work with colleagues across our nation to be able to make sure that Australia can harness our wonderful excellence in science to be able to deliver for Australia's defence. We do have just wonderful examples of excellence in our midst. And one that I'd love to draw out was the example that won the Eureka Prize this year for Safeguarding Australia, which was something called the Desktop Compositor, a partnership between our defence scientists and industry that allows decision makers to fuse and understand information from different classifications at the point they make decisions. Going into the future, I think it's going to be increasingly important that we can bridge some of the different cultures and silos across our system and have greater mobility between our universities, our government research labs, and our industry. To get after this, my group has launched a new program called Navigate, which is a mid-career entry program into defence science and technology. We have a strong graduate program, but we simply can't expect that people come in at a graduate level and are able to connect together the different elements of our system. So we're looking to build that deep connectivity at the mid-career level as well. And I think that's very exciting. I acknowledge that in many of the areas of science on which we're really dependent, some of the physical sciences, data sciences, engineering, are fairly male-dominated. And so we've set ourselves a hard target of a 40-40-20 profile of gender in this Navigate program to try and help leap us forward in bringing in a more diverse workforce. But again, thank you for listening. Thank you to the Academy for a powerful partnership that brings together purpose and some of the big defence challenges we face and the excellence that you represent as new fellows. We can do a lot more together, and I look forward to working with many of you in the years ahead. Thank you.
Thank you, Tanya, and we greatly appreciate your ongoing support. Today, each fellow will present for 10 minutes, followed by a 5-minute Q&A session. Throughout the event, we encourage you to ask questions. Please submit these using the Q&A panel on the right side of your screen. They can be submitted at any time, and we'll try to get to as many as possible. Now, to begin the presentations, please welcome Academy Vice President, Professor Helene Marsh, to introduce our first fellow.
Our next new fellow presentation is from Dr. Jennifer Stauber. Jenny is an international leader in ecotoxicology, particularly metal contaminants in aquatic environments. She's invented a range of new risk assessment protocols that are applied worldwide in research programs, industry and government, including the National Water and Sediment Quality Guidelines for Environmental Regulation in Australasia. Now to Jenny's presentation.
Good morning, I'm Jenny Stauber and I'm based at the CSIRO Land and Water in Sydney. And today I'd like to describe some of my research to assess the risk of contaminants in aquatic systems, particularly some new tools that we have developed over time. Contaminants are just any substances that are present in the environment at above background concentrations, and they only become pollutants when they actually cause an adverse effect. There are many different types of contaminants. We can have biological, physical, and chemical, and it's the metal contaminants, the chemical metal contaminants that I'm going to focus on today. We know that there are literally tens of thousands of chemicals marketed and in use around the globe, but we only have environmental protection guidelines for just a few of them. So what I'm going to talk about today is some methods we use to risk assess, and we have methods to assess exposure and methods to assess the effects of the toxicity of contaminants. So metals in particular have a number of different modes of toxicity. They can inhibit various processes like respiration and photosynthesis. They can also inhibit other ions getting into the cell through these ion channels. They can cause gill clogging and produce reactive oxygen species. All of these things can inhibit processes like growth and reproduction and survival. And I've just got one example here with some work we did on corals some time ago with the metal manganese. Now, manganese is normally thought to be not toxic at all, but adult corals are particularly sensitive to manganese, and rather than the zooxanthellae being expelled and the corals bleach, as we find with high temperatures, it actually causes the animal polyp to slough off the coral skeleton, and we're left with these bare coral skeletons. When we're assessing the exposure to metals, we have very challenging environments. We might have to go to the wilds of Papua New Guinea, where we've got seasonally very high turbid rivers and inaccessible locations. And we've also been to places like the driest desert in the world, the Atacama Desert in Chile, where there are mines high up in the Andes that still discharge their tailings liquor into the coastal environment. And more recently, we've been challenged by having to go and deep sea and look at impacts on deep sea biota many thousands of metres below the surface. And this is where perhaps mine tailings are being placed now and in future. But when we go to these locations, it's not just about measuring the total metal concentration, because that really doesn't give us much of an idea of the effects or the impact of that metal. And here I've just got an example where we've got a range of different sediments along the x-axis, and they all contain the same amount of metal, copper in this case. But you can see they have very different effects on this little amphipod species. When we looked at amphipod survival over 10 days, we can see that some amphipods survived completely and other sediments caused complete death of the amphipod, and yet they all contained the same amount of the contaminant copper. What this means is metal bioavailability really depends on a number of different things. It depends on the chemical form or speciation of the metal, and the metal might be present as a free ion, or it might be present complex with organic matter and humic and fulvic acids, which will reduce its uptake into the cell. And these speciations, or these species of metals, really depend on the water chemistry, the pH, the salinity, and the organic matter, as I mentioned. But there's another factor that also controls bioavailability, and that is competition at the cell surface, at the organism gill, for example. If it's a fish, we can have competition with other ions with the metals, which controls their uptake into an organism. And then, of course, we have detoxification processes and efflux of the metal out of the organism. But all these things are important in understanding the bioavailability of the metal and its toxicity and impact on organisms in aquatic environments. So to study bioavailability, we use a number of different lines of evidence in a multiple lines of evidence approach. So we use chemical techniques, ecotoxicology techniques, and ecology and biological field monitoring techniques. So these can be indirect, so we might use chemistry, different analytical methods to measure surrogates for bioavailability, for example, to measure labile metal. Or we can predict the species of metal that might be present in an environment using modelling, and then we can compare these predicted and observed labile concentrations with water and sediment quality guidelines. But a more direct way to measure metal bioavailability is to use sensitive bioassays that measure the response of the organism when they're exposed to the metal, and then we can combine these with other lines of evidence. So a lot of my research has been involved in developing new bioassays with different aquatic organisms, particularly microalgae, and to apply the technique of flow cytometry to environmental bioassay development. Flow cytometry many decades ago used to just be used in pathology labs to count blood cells, but we've applied it to counting, not just counting algal cells in bioassays so that we can measure growth and reproduction, but also so that we can look at the modes of actions of metals in cells and different organisms, such as inhibition of enzyme activity or production of reactive oxygen species or the cell division cycle. Now, most of the metals and the chemicals we assess with bioassays usually just use one organism and one chemical. But with flow cytometry, we've been able to put mixtures of species together in the one test. So we've developed these multi-species bioassays. In this case, it's with several different types of algae. And we can see not only the interactions of the different species with one another, both before and after we add the metal, and to see how that impacts their response, the bioavailability and the toxicity of the metals. Bioassays can also be used for other purposes, and they're particularly useful when we have complex mixtures of contaminants in an effluent and when we don't know the cause of the toxicity. So we can use chemical detective work combined with bioassays to identify the actual chemical in the effluent that's causing the effect or the impact. So for example, we might take a sample, we can chemically manipulate that sample to remove a particular class of chemicals, and then we can test that sample before and after we have removed the chemical, and then we can see whether the toxicity has been reduced. And if it's been reduced through that manipulation, we know that that group of chemicals is going to cause the toxicity, and then we can hone on down and identify the actual chemical of concern. The other thing that we use bioassays for is to derive water and sediment quality guidelines. Now, because different species all respond differently to chemicals and different chemical concentrations, we have to test chemicals, especially metals, with a whole range of species, algae, invertebrates and fish. And when we do these tests, we can put it all together in these cumulative probability distributions and then calculate what concentration of the chemical will give us 95% protection for the species. And this is how we derive all of our environmental guidelines for toxicants in Australia and New Zealand. We don't always want to have to measure, though. We can't measure every chemical and every organism's sensitivity to that chemical or metal. So we have also developed models to predict toxicity. So these are models, the biotic ligand model and multiple linear regressions, which just enable us to go to a specific site, and if we know the water chemistry, we can then predict the toxicity to that particular organism, so we don't always have to measure it. So over the last few years, I've spent some time also developing these multiple linear regressions so we can predict toxicity. The other new tool that we've been using is environmental DNA. So taking DNA from the environment and looking at how community biodiversity changes along a contaminant threshold. And this is the work of Megan Gilmore, one of our PhD students, and she has taken samples in New Caledonia along a nickel sediment contamination gradient from a nickel mine down across the bay into the ocean. And by extracting the DNA, she's been able to then identify what species appear and what species disappear in response to the nickel. And you can see here that we can plot the taxa. The black dots are the sensitive taxa that disappear as the nickel concentration increases. And then there's species that are more tolerant and they appear as the nickel concentration increases. And where these two cross over, this can give us an idea of the nickel concentration that we could use as a community-based guideline for where the whole community biodiversity may be different. So finally, over the last few years, I've been particularly interested in the interaction of multiple stresses. So not just contaminants and metals, but other physicochemical stresses, particularly from climate change, such as temperature and hypoxia. And so we're starting now to look at these interactive effects on toxicity and the risk of contaminants in different aquatic systems, marine, freshwater, and sediments. So I hope I've shown you today that we need to look at both the exposure and the effects of contaminants to really understand their risk in the environment. We use these multiple lines of evidence approaches from a wide range of disciplines to answer the questions. We have several new tools such as environmental DNA and these predictive models that allow us to predict toxicity and therefore manage the concentrations and the impact of those chemicals. And of course now we're also considering multiple stresses and their interactions as we move into climate change. I'd just like to thank the group at Lucas Heights, the CSIRO Contaminants Research Group, all the students and university collaborators over the many years at the Academy, and also our international funders, particularly the metals associations globally. Thank you very much.
And please now welcome live with us Dr. Jenny Stauber. Congratulations, Jenny, and thanks for your presentation.
Thank you very much. Glad to be here.
Now, we've got some questions for you from the audience. The first one I'll kick off with is that when bioassays are used to identify a source of contamination, is there a need to undertake further tests to cross-check the results from bioassays? And how long does it take to determine if there are any contaminants in, say, a river using bioassays?
It's relatively quick. We have some very quick bioassays that still tell us about chronic toxicity, but then we can actually double check our results. So when we're using that chemical detective work, once we've honed down on what we think of the class of compounds, we then have various validation techniques where we add that particular chemical to the effluent and see if the toxicity increases or decreases. And that way we can gradually hone down and then remove that contaminant. So for example, for Melbourne water some years ago, we identified that ammonia was one of the main issues in their effluent discharge going out into Bass Strait. So they were able to upgrade their treatment plant to reduce the concentrations of ammonia being discharged and therefore reduce the impacts on those organisms. And that was all through this, what we call toxicity identification evaluation procedure.
Yeah, it's such an important tool to be able to identify those things and know how to act. Has your research identified if certain chemical contaminants are more prevalent in developing countries compared to those in developed countries?
Yes, we have done work in India and China in particular. And there are some contaminants there, of course, that they have much greater contaminant issues in the Ganges River, for example. And we've spent some time helping them understand the effects and be able to assess the risks and improve the situation. The contaminants are, in developing countries, you know, they don't have the same regulatory environment that we do either. So they don't have the same strict license conditions and effluent treatment that we have in developed countries. So it is a little bit different, but it is gradually improving. We, of course, still have some contaminant issues here in Australia, particularly when there's sewage overflows or mining discharges or point source things. So it's certainly better than it was in developed countries, but we've still got a little way to go, particularly with those new emerging contaminants like PFOS and antibiotics and pharmaceuticals from personal care products.
That's terrific. Now, I've got a question that's just been sent in by Peter Currie, who's another of the fellows who will be speaking a bit later this morning, watching along. Thanks for your question, Peter. He asks, can you detect species-specific evolution of resistance, I should say, to heavy metals in your work?
We haven't really looked at genetic susceptibility. We have looked a little bit at tolerance. So we have done some work when we look at how pre-adaptation to a contaminant. So if you've got an organism that is used to being in an environment where there's higher concentrations of metals, for example, we can see that tolerance of that organism, but we've not looked at whether that's passed on to the next generation of that organism.
Now, we hear the term green chemistry being used today. Is the chemical industry globally working to minimise the adverse impacts of chemical contaminants to the environment and to human health?
Absolutely. So they're doing the usual, first of all, perhaps changing the product to reduce the toxicity, avoiding or reducing its use in the first place. So one example would be in Queensland with the pesticides and insecticides going into from our catchments into the GBR. So that's an area where the formulations are constantly being improved through green chemistry and understanding how we can substitute different compounds for ones that are perhaps legacy chemicals.
And perhaps could you describe, I guess, the impact of chemical contaminants on the overall impact on climate change and just where that fits in addressing climate change as an issue?
Yes, it's something that's been actually not looked at all. So all the IPCC reports do not mention contaminants, and they don't really talk about how contaminants can interact with those climate change stresses like temperature or hypoxia or acidification in the oceans. And so what we're doing with international colleagues is trying to understand how those climate stresses can change the behaviour of contaminants in the environment and therefore its effects, and how contaminants can change those climate change stresses in the environment. So it's a two-way thing, and we're hoping to get more recognition of those interactive multiple stressor effects from climate change and contaminants in future, because it's really been an area that's been completely ignored so far.
I found that a particularly fascinating connection in your presentation. And just finally, Jenny, how's the pandemic impacted your international research collaborations? I guess you're looking forward to being able to pick them up a bit more again.
I am indeed, and we're having a climate change workshop in Oslo next June, which I'm on the steering committee for, and I'm hoping that I and several other colleagues from Australia will be able to get to. So it has impacted us a lot. We've had to do all of our conferencing, by Zoom, as others have. We're sort of ready to actually get together with those networks again, definitely.
Wonderful. Well, look, thank you so much for your presentation and your time for the interview this morning. Congratulations again.
Thank you very much.
Now, to introduce our next fellow, please welcome back Professor Helene Marsh.
Our next presentation is from Professor Robin Gasser. Robin's fundamental research into the biology of parasites, their interactions with their hosts, and the diseases they cause has led to a range of advanced technologies to explore their genomes and functions. This research underpins the development of innovative methods for the diagnosis, treatment and control of socioeconomically important parasitic diseases of animals and humans for subsequent translation and commercialisation. Please welcome Robin.
Today I'd like to tell you a story about our wormy world. And before I do, I'd like to acknowledge the traditional owners of the land, past, present, and emerging. I'd also like to express my gratitude to the Academy of Science. I'm very humbled by being part of such a great institution. I'd like to acknowledge the support that I've received from my family, my research team, our collaborators internationally, and of course our many sponsors who have supported our research over the last 35 years. So someone asked me once, what field do you work in? I said, I work in parasitology. After a short pause, the person said, I love Paris. I think Paris is fantastic. And so I went on to explain that actually parasitology is the study of parasites of animals, humans, and plants. And there are a very broad spectrum of different parasites. Some are microscopic, such as the protozoa. Some are macroscopic and are usually worm-like things, wormies, nematodes, and flatworms. And then we have the group of the arthropods, including insects and ticks and mites. There are many others, but these are some of the main groups. And we particularly focus on worms, and I'm going to give you some examples of worms that have major impact globally in humans and also animals. And I'm focusing particularly on neglected tropical diseases caused by parasites. Here we have some examples of Leishmania, caused by a protozoan organism called Leishmania. It infects tissues under the skin and in the viscera, causing granulomatous change over time. Then we have these sorts of parasites called echinococcus that cause echinococcosis in people. This is actually a beast that has, is caused by an infection in dogs that transmits to humans. In humans, you see the growth of parasitic larval stages that cause disease. Then we have some roundworms like ascaris that cause ascariasis. For example, filarial nematodes that cause elephantiasis, and then this example here, which is schistosomiasis in people caused by a parasitic flatworm called schistosoma. And what I wanted to do is just illustrate on hand of these images the impact that these parasites have globally. They affect about 1 billion people globally, cause massive disease, particularly chronic disease over time, and they're very hard to control. The diseases are complex. Transmission of parasites is complex. They're quite hard to treat. There aren't many drugs for some of them. There are no vaccines. They're difficult to diagnose and control. And in fact, there's quite a lot of instances where control and treatment fails, and sometimes even due to drug resistance. I'm going to just explain this example here with schistosomiasis. Schistosomiasis is caused by the presence of schistosomes in the vessels inside the portal system near the liver and near the intestines. The adult worms live there and in the bladder in particular. And it really depends on which species you're talking about as to what sort of disease it causes. But in general, the parasites release eggs that then get entrapped in tissues, and those eggs induce granulomatous change and disease that gets progressively worse, leading to all sorts of changes in tissues as well as ascites, the release of fluids into the abdominal cavity, and that can be really, really chronic. It depends on the species that you're dealing with as to the type of disease. But some species, including Schistosoma haematobium, induces cancer in the bladder of people and can also induce and make people susceptible to viral infections such as HIV. So the parasite itself is transmitted via eggs that are released in urine or feces that get into an aquatic environment, that get into snails and then infect snails and asexually replicate. And eventually the larval stages are released, they swim off and they infect the host, the human host, by penetrating skin. Now for some species, animals represent reservoirs for infection. So there's also complexity in around other animals being able to contribute to human infections. So this is really quite hard to control. In a sense, there is a drug available, praziquantel. There's only one drug, so it's not highly effective in all instances, and there's no vaccine against this disease complex. And so it's just one example of the impact of schistosomes. So really our mission in parasitology is to try and gain deep insights into parasites, into how parasites interact with their hosts, and into the diseases that they cause, using cutting-edge technologies such as genomics, informatics, and a range of other laboratory methods as well. And eventually, once we have a good understanding of these diseases and parasites, we can try and translate towards developing better treatments, vaccines, and better diagnostics. Our focus has been particularly on identifying and classifying parasites. And generally, a lot of these beasts are difficult to identify because they don't have any really characteristic morphological features. And that's why we've been focusing on the use of molecular tools to classify parasites and identify them and diagnose infection. And those tools, analytical and diagnostic, have been useful to study the biology, epidemiology of the parasites and look at host affiliations and disease. And we've even done quite a bit of work on looking at how much variation occurs in particular species of parasites in populations. And quite frequently, you can identify that some parasites, although they look the same, are actually different, and you have different genetic variants of the parasites behaving in different ways and transmitting to different hosts. So this is quite an important area, and without fundamental understanding of these parasites using molecular tools, often you cannot control them properly. A lot of our recent work is focused on genomes and associated areas. So we've made a lot of progress, and that progress has really been dependent on discoveries that have been made in other areas, particularly in model organisms. So there's been a lot of advances over the years in genome sequencing technologies and in the use of digital technologies, so informatic technologies, computer algorithms and so on, to help us understand the molecular basis of life and understand the molecular basis of how these organisms work. And so one of the first model organisms to have its genome sequenced was Cinorhabditis elegans. It's a tiny worm that has been used extensively in many labs around the world to study the biology of metazoans and multicellular organisms more generally. And another genome that became available in 2000 was that of Drosophila. So having the genomes of these model organisms and all of the associated biological, biochemical, genetic, and functional information for these organisms has been highly significant for us working in the parasite field. Because this information has become available through databases, it's been publicly available, we can access this information, and it's become particularly important because these model organisms are actually related to the worms and insects that we work on. And so that has really underpinned a lot of the work that we've done. So with the advances in first, second, and third generation genome sequencing technologies, we've been able to make a lot of progress in defining the genetic code of a lot of these parasites that we work on. So these are some examples of papers that we've published that describe the genomes, but also the biology surrounding the parasites. And we've looked at some large roundworms of humans, hookworms, blood-feeding hookworms of humans, and some of these flatworms that we've been talking about, so schistosomes, but also the carcinogenic liver flukes that occur in people as examples. And we've generated a lot of draft genomes that then now have been deposited into these databases that are now available as resources to the community, the scientific community working on these beasts and studying the diseases that they cause. And so a lot of the initial work is done with second generation sequencing technologies. Things have advanced over the years quite really rapidly. And so we're now making use of third generation technologies to try and refine the assemblies and the annotation of these genomes to get a much better insight into the organisation of gene families, to try and work out how these genes function, and importantly, to try and discover and characterise some of the unknown genes that occur in parasites that likely represent those molecules that give parasites the particular mode of existence that they need to survive in host organisms such as animals and humans. And so with these resources available, you can see here on the left, there's a lot of genomic information available for nematodes, for instance, but there's also information available for their RNA, proteins, sugars, and lipids that occur in these worms, as well as metabolic pathways and so on. And as time progresses, we'll be able to complete this table here and fill out all of these areas to then be able to use all of these resources to study in a holistic way by integrated genomics, is study the fundamental molecular processes and pathways that occur in parasites, and particularly try and focus on the way that these parasites interact with their hosts to try and find ways of blocking this interaction or blocking biological pathways, finding the Achilles heel in these worms using advanced technologies. And more recently, we've seen the emergence of artificial intelligence approaches with machine learning and so on that allow us now to find new ways of looking at the structure of molecules, but also looking at the way in which these molecules function in the parasites. And we're in the process now of trying to translate these discoveries using machine learning approaches to try and identify vaccine and drug targets. And combined with laboratory work, we can now try and work towards new drugs and vaccines. So this is a really exciting area. So we have a bright future ahead with being able to integrate the use of these advanced digital technologies, particularly machine learning, to work towards new interventions against parasites, and then using those to contribute towards some of the main sustainable development goals of the UN to try and control parasites, particularly neglected tropical diseases.
Please welcome now Professor Robin Gasser. Congratulations, Robin, and thank you so much for your presentation.
Thanks so much, Paul. I'm very, very humbled.
There's been so much focus, rightly so, on COVID-19 globally, but you mentioned in your presentation 1 billion people around the world who are affected by the sorts of parasites that you're studying. Why do you think that the development and commercialisation of vaccines to treat parasites has been so slow?
Yeah, it's a great question, Paul. I think the main issue really is that the developing world really doesn't have the funds to cover the costs to pay for a lot of this. So a lot of the pharmaceutical and biotech companies have not invested heavily, but philanthropy has. You know, for example, Bill Gates and co have done a lot to really support some of the philanthropic arms of these ventures, where there's a focus on trying to, you know, find better ways of controlling some of these really persistent parasites, which cause chronic disease. I mean, the issue is that they're very chronic, you know, and so quite often drugs don't really give you the sustained effect you need over time, because often the complexity of life cycles leads to this chronicity in populations, you know, in human populations, and sometimes even in animal reservoirs. So I suppose really to answer the question briefly is there just hasn't been enough investment in these diseases that predominantly occur in disadvantaged parts of the world, in the tropics and subtropics quite often.
Yeah, and really those images that you shared are really quite obviously disturbing and shocking, but hopefully that help will increase over time. And I think the chart that you showed at the end in terms of the map of the work that lies ahead, but the path forward it was really compelling. Do we know the number of global annual deaths and even economic impact to the world because of parasitic infections?
It's very hard to estimate, Paul, because you know, often it's not recorded accurately, you know, but there are estimates, and they give them in the score, you know, in what they call a daily score. So it's the adjusted life years impact and the burden of disease. I can't give you an accurate figure, but it is very high, and quite often it gets claimed that it's much higher than, you know, than cancer and a lot of other diseases, you know. But it is very substantial, and the problem is it's chronic, you know, so it affects populations over a longer period of time. If you look at situations like the northwestern part of Thailand, where you have, for example, carcinogenic liver flukes that live in the bile ducts of people, they're continually perpetuating from snails to humans, or from fish to humans, and because of the induction of cancer, quite often, you know, the prevalence of disease is quite amazing, not only of the parasite itself and the impact that it has, but also the complications, namely the induction of cholangiocarcinoma. And so these are examples. Sometimes they're endemic, you know, there's pockets of these problems, but on the other hand, there can be quite vast impacts in particular communities as a consequence of, you know, the presence of the parasite, but also the factors that allow the parasites to keep perpetuating and transmitting.
Robin, I've got so many questions I'd love to ask you, but I will only have time to ask you one more for now, which has just been sent in by Professor Francis Saparovich, one of our greatest contributors of questions. So I'd encourage everyone else to continue to ask questions as well. Francis asks, what role improved hygiene could play for reducing parasitic diseases, akin to what's happened with other microbial-based diseases?
That's a great question. I mean, you know, when you look at parasites, there's two sorts really, if you think about it. There's those that transmit directly without the involvement of an intermediate host, such as a fly or a vector of some description. But there are those that might involve a range of different intermediate hosts. Sometimes it's one, but it could be multiple. So it really depends on the parasite as to how you go about control. But for a lot of them, hygiene is really important, particularly those that have direct transmission. So, for example, if you look at ascarid roundworms, so common roundworms in people, hygiene is one of the key factors. And not only sort of in the household, but also in the environment. So contamination of foodstuffs and water and so on will, if you can prevent that cycle, that will disrupt the transmission of the parasite. On the other hand, where you have more complexity and you have intermediate hosts, it becomes harder because the intermediate hosts have their own sort of environment as well, their own, and so it's much harder to control parasitism and parasitic disease when you have the involvement of other invertebrates, for example, or even vertebrates. So it can become complex. But for those that have direct transmission, hygiene is a critically important part of control, management and hygiene. Yep.
Robin, it's a field possibly less glamorous than the study of Paris, but far more impactful, the study of parasites. Thank you so much for your presentation and congratulations again.
Thank you, Paul.
Now, please welcome back Professor Helene Marsh to introduce our next fellow.
Next, we'll hear from Professor Barbara Nowak. Barbara's research on the effect of pollutants and environmental factors on fish diseases has advanced understanding of fish health and contributed to sustainable aquaculture. With her Norwegian collaborators, she discovered a new type of lymphoid tissue in salmon gills, redefining directions in fish immunology. She's developed methods used worldwide to assess fish health and investigate fish diseases. Now we'll hear from Barbara.
Imagine if fish disappeared from the world. Imagine life without fish. No more fish on the menu, no fish on supermarket shelves, no fish markets, no more fishing trips, no fish in public aquaria, and no fish in pet stores. Fish are essential for at least 5 UN sustainability goals. If fish disappeared from the world, we would have to find another source of protein to replace the 17% currently contributed by fish. Fish-based industries such as fisheries and aquaculture wouldn't exist, so there'd be massive loss of jobs. No fish would result in a collapse of many food chains. Fish are an important component of ocean as a carbon sink, so no fish would mean reduced carbon sink. Fish currently contribute 52% of all vertebrate species, so if fish disappeared from the world, it would result in loss of biodiversity. As a recreational diver, I often admire this biodiversity. But sometimes I can't stop myself from noticing health issues. For example, Melbourne skate, shown on this slide, has little black spots on its skin, which on the closer look are obviously marine leeches. Other parasites which can be easily seen underwater with naked eye are isopods, like here on the globefish, or copypods, sea lice on the lingcod. Most of the time, I can't say if the fish is healthy or not just looking at it underwater. And to ensure the health of fish populations, we need to be able to measure the health of individuals. So how to make sure fish are healthy? My research group has been working on fish health for many years, and we developed lots of different methods. You can also either focus on the health of the fish and look at its normal immune response, normal hematology, normal histology, or normal gene expression or other variables. Or you can see if the fish is sick, and either if any of those variables are different than the baseline, or we can look at presence of lesions or presence of pathogens. I would like to share with you a few examples of our research. So to be able to measure pathogen load, we need to identify pathogens first. And one of our main contributions to fish health was the identification of Neoparamyba purulans as the causative agent of amoebic gill disease in farmed Atlantic salmon worldwide, as well as in some other fish species farmed in marine cages. This allowed us and other researchers to focus on this pathogen when we work on treatments and try to develop vaccines against this disease. Another disease I've been working on is epitheliocystis. And at the time when I started working on it, it was believed it was caused by the same pathogen in all different fish species it was affecting. However, our research showed very quickly that each fish species had different pathogen causing the same condition. That has implications for disease management because we know that other fish species are not reservoirs of the pathogen for the fish we are interested in. Another contribution we made in this area was development of molecular methods to detect blood fluke Cardicola orientalis, which at the time was only known to be present in Japan. We showed that it was not only present in around southern bluefin tuna, but also using archival samples that was present before we detected it first time. Another blood fluke affecting tuna is Cardicola forsteri. Blood flukes need two hosts to complete their life cycle, fish and an invertebrate. And we did not know what the intermediate host was. We only knew that tuna was the final host. Knowing intermediate host can sometimes allow us to avoid infection if we can avoid the intermediate host in the farming environment. A joint collaborative effort, including Queensland University, Queensland Museum, SARDI, and tuna industry, showed that the intermediate host was a small marine polychaete, Longicarpus modestus. And while it wasn't possible for the whole industry to avoid it because it's widespread in the ranching area, those tuna which were farmed further offshore were not affected by the blood fluke. Another tuna parasite was sea lice, Caligus gyastos, which was associated with blindness and loss of condition. Sea lice have a simple life cycle, so they need only one host. They have 4 living stages, including nauplius and copypodid, and parasitic stages, calamus, per-adult, and adult. What we noticed was that the sea lice on tuna were almost only adults. And for example, out of 5,000 sea lice we looked at 1 year, there were only 3 early life stages. So few individuals of early life stage suggested that there was another reservoir of this parasite, which was bringing the parasite into ranching area. A large survey of wild fish species showed that these parasites were common on wild Degan's leatherjacket, and new management practices, excluding the Degan's leatherjacket from ranching areas, resulted in a reduction in the sea lice infection, so it's no longer a problem for the industry. So these were some examples of looking at pathogens, but we can also look at fish itself and measure its health, either using traditional methods like hematology or histology, or more recently developed gene expression, protein expression, or presence of microorganisms, in particular bacteria, on gills, skin, and gut, where they usually colonize them, and comparing the normal microbiome to microbiomes from diseased fish. Here are two examples of how we applied our results to establishing biomarkers of pollution effects. One is for sculpin in Greenland, and the other one sand flathead in Tasmania and Victoria. For sculpin, the best biomarker was determined histologically, and we're looking at densities of mucus cells on the gills. Whereas for sand flathead, the best health indicator was gene expression. Depending on the type of pollution, we looked at different genes. Determining fish health can be very challenging, though, because of the high biodiversity, which is illustrated here in the phylogenetic tree. So the normal baseline values are not only species specific, but they can be also life stage specific and also different under different environmental conditions, for example, changed by temperature or salinity. Because of that, they can be only determined for the species which are economically important. And for other fish species, we have to find other ways to determine their health. Fish disappearance would be catastrophic. So it's important to be able to measure health of individual fish so we can predict health of populations and ecosystems. And fish health measurements can be used as bioindicators of effects of pollution and other environmental changes. It is also important to know biology of pathogens and parasites, so we can use them as biomarkers, indicators of fish health, but also importantly, that allows us to control and manage infectious diseases in aquaculture. Behind every successful scientist, there are lots of people who contributed to this success. And I couldn't fit everybody on this slide, but I would like to thank all my students, honors, masters, and PhD, postdocs, visitors, collaborators, and in particular mentors for supporting me on this journey. And thank you very much for your attention.
And we're delighted to have with us Professor Barbara Nowak. Congratulations, Barbara, and thank you so much for your presentation.
Hello from La Terravita. I would like to acknowledge traditional owners of our land, and thank you very much for the introduction and for congratulations. I'm really honored to be here.
Barbara, could I start off just by asking you what led you to study fish health in the first place?
That's a very interesting question. I was always really interested in science. I had fantastic science teachers, even in primary school. My parents were very supportive of science. So that was a really big part of it. But the movement to aquatic science was partly through my recreational interests, including sailing and diving. And watching fish underwater was really something quite fantastic. I guess when I studied fishery, and I always thought that it would be really good to contribute more to developing new sources of protein for people. And somehow the fish health appealed most to me because it's almost like a detective work and investigating cases all the time. And it is very essential for not only farm fish, but also for wild fish.
Barbara, I have a question here from Professor Helene Marsh, who says, thanks very much for an interesting talk, Barbara. Have there been any examples of disease in wild fish leading to pollution controls in Australia?
In Australia, the main impact of disease on wild fish was huge mortality of pilchards in the 90s, but it wasn't related to pollution. It was related to a virus, and we never found the source of that virus. It was suspected to be introduced with fish imports from other countries, and that was really quite a massive fish kill. I've been involved in quite a few fish kills which were due to either pesticides overspray with investigations followed by court cases where the pollutant got into the water, but most of the time by accident, but sometimes also as an effluent from a particular company. And they usually caused mortalities. My research is more into what happens before the fish die. By the time the fish die, it's a bit late for those fish to do anything. But obviously, fish mortalities are very often one of the signs that there is something wrong going on with environment. They're not always caused by toxic substances. They can be also caused by environmental changes. but the environmental change is also important to understand.
Another question from our audience. Could you briefly comment about the relative health of farmed versus wild fish, especially with regard to parasites?
This is a really good question. So I talked a little bit about parasitic life cycles in my presentation, and fish which are farmed are fed manufactured feed, which means that they don't get any parasites through feeding. So in the wild, a lot of parasites they have complex life cycle, they infect the fish because the fish is eating another host of the parasite which is already infected. So farmed fish will not get those parasites because at least most of them will be feeding just on manufactured feed, which doesn't contain anything like this. But the parasites which have simple life cycle, which only need one host, have a really good place to proliferate because they have a lot of fish which are the same species, and in sea cages in particular, they're confined to place, so they can't go somewhere else where there are no parasites, they have to stay in a cage. In Tasmania, Atlantic salmon has problems with amoebic gill disease, which I mentioned, which is caused by amoeba. And this is really the only parasitic problem in Australian farmed Atlantic salmon. But in Norway and other countries, sea lice is a huge issue, and that's affecting mostly farmed fish, but can also be present on the wild fish. And sea lice, while they have lots of life stages, they only need one host. So they're also another example of a parasite which can spread really easily on a farm. Because these parasites are external, there are lots of ways to manage and treat those diseases, either by putting the fish through a bath, or having even more sophisticated ways of treating them. In Norway, there have been trials with using lasers and thermal or magnetic projections to remove the sea lice from the fish skin. So there is a lot of research going on in industry to minimize, because obviously those parasites would have a significant impact on the industry, and the industry wants to reduce it. And there is a significant amount of research and progress going on with parasites. But interestingly, despite a lot of research going on developing vaccine against those parasitic diseases, we have no commercial vaccines against any of parasitic diseases in farmed fish.
Extremely interesting, Barbara. I wish I could ask you more questions, but that is all that we have time for now. Thank you so much, and congratulations once again.
Thank you very much. I hope to see you in Canberra one day.
We're looking forward to that indeed. Now, please welcome Academy Council Member, Professor Bob Graham, to introduce our next presentation.
Thank you. Our next presentation is from Professor Peter Currie. Peter is a world-renowned developmental, evolutionary and stem cell biologist who studies the genetic basis of skeletal muscle stem cell action during development, evolution, regeneration and disease. His key discoveries utilize several animal models to define the genetic and evolutionary basis for muscle formation and growth. Please welcome Peter.
My name's Pete Currie, and I'm Director of the Australian Regenerative Medicine Institute at Monash University. And I want to thank the Academy for finally allowing us to make our presentations, and I extend my congratulations to all the fellow inductees. So my interest is in regeneration. I'm a regeneration scientist, and I share this passion with a classical figure from history. This is Hercules, one of the original regeneration scientists. And here he's conducting an experiment on the Lernaean hydra, and he's lopping off the heads of the Lernaean hydra and learning to his horror that two can grow back. But what is interesting about this regenerating system and all the systems that we study is that they go back in an exact copy to repair the regenerating tissue back to homeostatic conditions. So I'm interested in what stem cell systems are deployed to generate this repair and how that process is regulated. Of course, the hydra that we study these days is not so confronting as the one that Hercules had to dissect. But nevertheless, it has the same amazing property that if you lop off parts of it, it can regrow and form the original tissue just as it once was. So this is the process that I'm interested in studying. This particular attribute, regeneration, does not really occur in the phylogeny in a way that we can fully understand. It's present in a range of organisms and systems and tissues, but rather randomly in the phylogeny. However, we can illustrate the fact that non-mammalian vertebrates show a greater regenerative ability, and therefore I study this end of the phylogeny using teleos and basal gnathostome species, fish. I study regenerative capacities and their origin. And I study this particular tissue, skeletal muscle. Skeletal muscle is pro-regenerative and a highly conserved tissue across the phylogeny. And if you're going to remember one thing about this particular talk and who I am, I'm that fish guy that studies skeletal muscle. So what does the lab do? Over the course of the last 3 decades, researchers settled into 4 major themes within the group. We study the regenerative capacity of skeletal muscle in terms of human disease. We also study the mechanisms of repair using the zebrafish system, the small freshwater teleost. We try to understand how other stem cell systems are deployed during growth, and we study this across the phylogeny using non-model systems. But our chief workhorse is this rather beautiful creature below, the zebrafish larvae, which is like a wet mouse, in which we're able to study the stem cell dynamics of living tissues, in particular skeletal muscle. In this particular animal, we're charting the clonal contribution of muscle stem cells to growth, with each muscle stem cell generating muscle in different colors. So what sort of discoveries or what sort of attributes of stem cells are we interested in? Well, we try to answer questions that aren't available to other systems to study. So in vivo dynamics of muscle repair, skeletal muscle repair, was largely uncharted waters for regeneration scientists. So we developed transgenic animals, and here we're just showing the result of one experiment after injury, where we've made different stem cell populations different color in green and yellow, and we're able to show that one stem cell population gave rise to the other to affect repair and generate this through asymmetric division. And this became a first principle of repair of skeletal muscle, that asymmetry was the way that the stem cell systems operated. We're able to go on and chart the clonality of the repair system using that ZebraPo system I introduced, and chart which stem cell systems give rise to which repair, and show here in the same 2 larvae prior to and after injury that regeneration is generated by rare clonal stem cells that generates clonal repair. We're also able to understand, using transgenic zebrafish larvae, the in toto wound repair environment. So by labeling the cellular milieu in different transgenic colors, we're able to show which particular cellular phenotypes are evoking or controlling repair. And here we're showing one of the first responders to regeneration, the innate immune system, here in yellow, a particular macrophage. And what these images show, that the blue guys here, which are the stem cells, are enveloped and actually controlled by the macrophage, which secretes specific signals which stimulates their repair. So the macrophage in this context generates a transient stem cell niche that was absolutely obligate for repair. And by sorting these particular cell types from individual larvae and then profiling them by sophisticated techniques such as single cell RNA-seq, we're able to identify a particular subset of macrophages that express specific genes, and we're able to isolate which of those genes are required for the regenerative process and which are secreted from the macrophage to stimulate the proliferation of the stem cell, and then take that molecule and stick it in a mouse, which is not a very regenerative system, a particular injury model that was not regenerative, and show that the fish molecule was able to evoke a very high level of repair in this normally non-regenerative system. So this is just one example about how understanding the in vivo dynamics of stem cell systems can lead to the isolation of novel observations, which could have therapeutic relevance. The other area that we're interested in is growth. We think growth is one of the great remaining frontiers of development of biology. We know a lot about how to make the first forms of organ systems through the embryology that's been going on for decades, to the point we can make little avatars or organoids of most organs in the body in the dish. But we struggle to understand how we grow those organs to scale to form the homeostatic condition that is insulted by injury or disease. So we've used our system, the zebrafish system, to label muscle stem cells that are required for growth. And our system, zebrafish, has one advantage, is that the organ keeps growing continually through life. And we were able to characterize an organ system or stem cell system called the external cell layer, which formed an eternal or a hyperplastic growth zone of stem cell systems that drove the growth from the outside of the animal to form stratified hyperplastic growth, here marked in green and in white. The growth of the organism is controlled by specific stem cells. And we're able to show rather simply using our clonal system that this growth was random to begin with. Different stem cell colors contributed equally and different clones contributed equally, but over time a process called stem cell clonal drift, where one uber stem cell came to rule them all and drive growth of the entire organ system, such that the adult organ system was comprised nearly entirely of the progeny from a single stem cell. And this clonal drift mechanism has come to be universally recognized as one of the mechanisms that derive growth of a number of organs, in barcoding of individual embryonic rudiments and understanding how different tissues are derived has led to the understanding that clonality is a feature of most organ systems. As I said, we're also very interested in about how the properties we describe in our model system, zebrafish, are deployed across the phylogeny. And we use particular species. Here I'm showing you an embryo of the living fossil Callirhynchus milli, or elephant shark, which we have a capture program for over time, have developed over time. And we use these animal systems at the base of the vertebrate phylogeny because they are the earliest or most basal species that have a modern vertebrate body plan with the organs that we know and love and can study. So we studied two of the basal chondrichthyans, chimeras, and a true shark. So we developed systems for capture of this living fossil Callirhynchus milli, which luckily only comes in to waters off Western Port Bay, or lucky for us, that is, about 4 miles from my holiday home. And so we line capture these animals, bring them into a large holding facility in Rosebud. The females lay their eggs because they're fertilized from males offshore, and we collect the eggs, grow them in these cages and generate embryos. And we also developed a model shark system for in laboratory study using the developing husbandry techniques for the epaulet shark, which is a true shark. We can obtain embryos from paired eggs that are laid every 2 weeks. We can generate sophisticated embryology of the animal, and we just finished the genome, so all the modern genetic techniques are available for the study of the embryology of this model shark system. And using these two model systems of basal gnathostomes, we've used all the markers of stem cells within these embryos. These are embryos of Callirhynchus milli and of the epaulet shark, and sections of these animals marked with the stem cell markers. And what we rather dramatically have shown in this study is that sharks use a different system than in zebrafish and teleos, which is much more like amniotes, us, birds, and mammals, and very different to the ones that are deployed in zebrafish. So through the phylogeny, growth cell systems vary greatly and are regulated by the different molecular signals. And we think this will be a rich area for study in the future. So thank you for listening to my fishy story. I want to thank the legions of honours, masters, PhD and postdocs and techs that have worked with me over the decades to make these discoveries. I showed work from David Gurevich, from Fong, and from Danny and from Catherine through the talk, ably enabled by current lab members, and these collaborators generated some of the data that I showed you today. So thank you for listening.
Joining us live now is Professor Peter Currie. Welcome, Peter. Congratulations.
Thank you very much.
I wanted to start off by asking you, how might your work be applied to human muscle or tissue regeneration?
Thanks, Mark. Well, we try to study broadly phylogenetically so we can understand general basic principles of stem cell function. And I picked a story at the front of my talk to really illustrate how we think this can work, that if we can find molecules that are pro-regenerative in one system, the fish and apply them in mammals to stimulate repair. We call it the fish tank to bedside approach. And we think that has capacity to generate therapeutic outcomes for us.
Yeah, it's a really exciting area to be delving into. What other areas of study in the future are likely to build on your discoveries?
Yeah, so we are trying to build an in toto map of regeneration in the larval fish using the sophisticated imaging techniques that we showed. So we'd like to know what every cell is doing over the time of the repair and what that cell is contributing to repair. So what I showed to you today is just the start of that scientific journey with a couple of the cell types. And we hope that we can make discoveries of equal impact for every different cell type that contributes to repair. And there are many, from the vasculature to fibroblasts to other cells that aid repair. And so if we can generate an in toto map of this particular tissue and organs repair, it could be broadly extrapolated to other regenerative or non-regenerative settings.
I love the videos and the images of the sharks that you gather near your holiday home, as you mentioned. Tell us a bit more about the process there and how do you gather their eggs and are they released again at the end?
Yes, so the females are, Calliorhynchus milli is a chimera, which is the oldest or most basal species with paired appendages and a head on the planet. And it just so luckily happens that this living fossil, which I'd sort of observed fishermen catching in the bay as a child, is actually a really important species for understanding the evolution of all kinds of organ systems. So the process was very simple. We do two methods of capture: line fishing with specially guarded lines to restrict line whip on the animals and keep them as healthy as possible. We bring them into the holding facility in Rosebud. For 3 months, they lay the eggs, and we have a release ceremony where we take the females and release them back into the bay. And we also work with trawler fishermen who do trawler fishing up and down the coast. They frequent the coast along various areas. And the skein net fishing is a really nice way to catch the females because it doesn't mark them in any way. And we actually pay fishermen to go out and skein net fish for them.
You showed a beautiful image at the end there of your postdocs and the other people who've supported the work throughout your career. One of the strategic priorities of the Academy is to increase the participation, particularly of women in STEM. What would you say to encourage women considering a career in science?
Well, I think science is one of those careers that's a very well-kept secret. I mean, you get to travel the world and meet great people. I mean, the pay is not that great, but if that's not what's of interest to you, it's extraordinary. But, in the life sciences, there is an incredible wealth of female scientists who we lose along the journey. And I don't think we have a problem engaging female scientists. We have a problem in keeping them and promoting them. So that's something that at our institute and across the Academy, I know is a great passion. And so we just have to do better at doing that.
Yeah, it's very well said. Now, just finally, does your research involve working with researchers from many other disciplines?
Yes, indeed. So a lot of the techniques that we involved in the light imaging are engineering based and developing of new techniques. Because I also span evolutionary biology, I work with paleontologists, and that is a fascinating science that I'm a dabbler, but really keen to know more about the phylogeny, which we can't see. And Australia has some incredible paleontologists and I think incredible paleontological resources at its disposal. And particularly in the basal nathostome area, there's an area called the Gogo fauna, which harbours uniquely preserved soft tissue specimens of placoderms, which are the most basal of the nathostone species. So I really love going to paleontological meetings and being completely confused, but it's an amazing science.
Well, Peter, thank you so much for your presentation and your time this morning. Congratulations. Now, please welcome back Professor Bob Graham to introduce our next fellow.
Thank you. Our next new fellow presentation is from Professor Glenda Halliday. Glenda is acclaimed internationally for her research on neurodegeneration. She established the Sydney Brain Bank and applied her expertise to clinico-pathological correlations revealing the anatomical, biochemical, molecular, and genetic characteristics of several neurodegenerative diseases, particularly Parkinson's disease and frontotemporal dementia. Her work has changed international diagnostic criteria, provided differentiating characteristics for neurological diseases, and developed disease staging schemes. Now to Glenda's presentation.
I mainly study the human brain and would like to tell you some of the things I've learned. Collaborations are key and the human brains are different. And by that I mean each brain is unique. It's just like a face, slightly different. It's regionally heterogeneous. Brain tissue can only be assessed at one time point, at death, and there's no other way to assess changes at the cellular and molecular levels. Diseases affect certain regions more than others and select cells within regions. So how did it all start? It's not something that you think you'd be doing. I did neuroanatomy and mathematics as my majors in the undergraduate, and during this I wondered how the human brain compared to those of the laboratory species I was studying. The brain itself is so much bigger, as you can see here, a human brain compared to a monkey brain compared to a rat brain. So to do this, I established widespread collaborations with neurologists to obtain full clinical histories for donated brain material. This culminated in 2009 by the establishment of the Sydney Brain Bank. I could then assess different species and look at the cell structures and composition of the brain, and highlighting here just the area of the that contains dopamine neurons. Here I can stain the cells by different species using the same marker, and then we can map in the serial sections single neurons in different regions of the brain. And you can see here, I think that you can see that as the brain gets bigger up to humans, you can actually see an increased density of these cells. So is it that you actually just have a bigger brain that's got more cells, therefore they're all arranged in the same way, or are there subtle differences? And to do this, we assess the brain size compared to the cell numbers. And if you plot these, you can see that they're not parallel lines, which would suggest that some species have more cells in a different size brain compared to more or less cells in their brain compared to the size of the brain. We can do this throughout the brain, and that can give us an idea of how the different circuits are functioning. And here I'm illustrating this by the major basal ganglia circuit, which regulates the cerebral cortex. It has a number of important structures, and you can look at the volume of these structures and the numbers of cells in these structures and the type of cells. And in doing this, we can build up a picture of the different structures in the different species. And interestingly, the humans have less substantia nigra dopamine neurons that are innervating a larger striatum, and indicating that there's indicating that the cells in the substantia nigra that are dopamine would be more highly active in humans to regulate a larger structure. And then humans have a larger circuit, the globus pallidus and subthalamic nucleus, compared to other species, suggesting that this arm of the circuit is more active in humans, and therefore greater regulation of the basal ganglia through this pathway. And that's been recently shown in functional MRI studies of human brain. It was known that there's some unique properties to humans, and here I'm showing the pigment that you can see by eye if you just cut the brainstem of a human compared to all the other animal species that we studied. And we wondered whether or not how you're born with this pigment, or how it actually how it occurs. And so we studied brains of different age groups, and found that you're not born with pigment, that the actual pigment starts to occur around the age of 3, and that's when you're acquiring motor skills. And it actually, you have a full complement by the age of 25, when you largely developed you have largely finished your development of motor and cognitive skills. So this suggests that they have increased activity of these cells because the less cells in the human that are innervating the larger basal ganglia circuit, and therefore has greater propensity to oxidize and form this pigment over time. And that this is actually the activity in this circuit is greatest as you're developing the skill sets that you need. By studying disease mechanisms, we could do that by concentrating on longitudinally followed cohorts, assessing people at different clinical stages and looking at regions that are primarily affected or less affected, and looking within the regions at cells that are primarily affected versus less affected, and then within the cells, looking at regions, at mechanisms, pathways that are affected versus not affected. This builds up a concept of the disease over time. And then you can look at the different regions that are affected early in the disease and then follow them over time. And I want to illustrate some of the studies that I've done by looking at the dopamine cells that are actually got the pigment and their accumulation of alpha-synuclein pathology. So how does these Lewy bodies, accumulated Lewy bodies form? We know that this is a pigmented dopamine cells, mainly has monomeric alpha-synuclein. And then the first thing that happens is that the alpha-synuclein forms punctate material inside the cells. And then these coalesce into loosely packed filaments. And then the loosely packed filaments ubiquitinate, which is an indication that they should be going into a lysosome. And over time, these filaments compact into a really ordered structure, a bit like a pincushion of filaments through a compaction. There's a number of other biochemical features that happen inside the cell. For example, alpha-synuclein phosphorylates before it ubiquitinates, and there's a number of other proteins that are actually abnormally ubiquitinate and accumulate in the cell in addition to alpha-synuclein. The changes in the cell occur very slowly by doing patients at different time scales. We could actually tell that the average duration for Lewy body formation is about 10 years in Parkinson's disease. The neurons are not the only ones that are accumulating alpha-synuclein. The glia are, and particularly the protoplasmic astrocytes, a specific type of glia. And over time, there's more protoplasmic astrocytes that are actually accumulate alpha-synuclein than neurons. The other thing that we found is that the older the age of onset of clinical disease, the more likely you are to have a much more rapid disease course and a more rapid spread of pathology through the brain. So there's an age association with the impact of these inclusions on the brain with an older age causing a much more rapid disease. Just want to show you some of the other work we've done, which is to look at the brains of patients with frontotemporal dementia. We defined a staging criteria based on atrophy. And you can see here the frontal lobe and its atrophy over time. And here's the temporal lobe, particularly the hippocampus and its atrophy over time. We found that it takes about 5 years on average to go from stage 1 to stage 3. And then you can have a long languishing period. That the actual clinical phenotype is rapid to go from stage 1 with very little symptoms to severe dementia by stage 2. Unfortunately, the survival in these patients is much less than the survival in Alzheimer's disease on average. So I hope I've shown you a little bit about the work looking at the human brain. We can compare the brains of humans and animals to identify functional differences in the anatomy, and I've illustrated that by illustrating that by basal ganglia circuit that modulates cortical activity. That some human brain cells have obvious unique characteristics. I've shown you about the pigment. The development of pathological occlusions actually takes time and has a set course. And for example, I showed you that alpha-synuclein fibrillizes into insoluble Lou bodies over about a 10-year period. That there's unique patterns of atrophy and spreader pathologies occur over the course of the different diseases. And I've shown you that by looking at the rapid atrophy, which is a feature of frontotemporal dementia. I'd like to thank and acknowledge the research participants and their families who committed to the longitudinal studies. That includes brain donation, all the funding bodies, and the research is a real team effort over many years, and I've just highlighted some of those that have been involved. Thank you.
And please welcome live with me now Professor Glenda Halliday. Congratulations, Glenda, and thanks so much for your presentation.
Thank you, and thank you to the Academy for it's very exciting, very exciting.
Now, I've got a question for you to kick off with. Which comes first, the brain stem pigmentation or fine motor skills? For example, if an older person stopped using their motor skills, would that decrease the basal ganglia activity and reduce the pigmentation?
It doesn't reduce the pigmentation at that time. It looks like the pigmentation is more associated with the development of the skill set rather than the perpetuation of it. So over time, you can get decreased activity, which doesn't impact on the pigmentation. After the age of about your mid-20s, the pigment, you don't get less pigment, but the pigment continues to oxidize. So you get an increase in the iron and the oxidized forms of the pigment rather than the amount of pigment within a cell.
Okay, thank you. I've got a question sent in by one of our fellows in the audience who asks, how do atrophied cells get removed from the brain and how often are they replaced?
Good questions. The cells get removed from the brain through the immune system. There's a specific cell types in the brain. There's specific immune cells, effector cells, which are the microglia, and they phagocytose through very similar immune reactions to your peripheral immune cells that phagocytose things, so like the macrophages of the brain. And so there's a single way that they are actually cleared. Even if they have, there's a number of ways that neurons die, including by apoptosis, but the apoptotic bodies are still cleared through the immune system by the microglia. How are they? So what was the other part of the question? Sorry, what was the other part?
Yes, it was how do atrophied cells get removed from the brain and how often are they replaced?
Yeah, the replacement is a more interesting question. I mean, in animal brains, for example, they've found that the hippocampus, you remove cells and that's over time you get more cells, but there's actually been a lot of fighting in the literature, contention about whether or not that's the same in a human brain. And there's a number of different studies which show that you have a lot of new cells, and there's other studies that show that you have very few new cells. So the difficulty, I think, is in you can't manipulate the cells to know what's new or old in any single brain. And so it's still hypothetical rather than knowledge from actually knowing experiments.
Yeah, well, possibly interesting to see where that contention leads as we look at the issue further. Just finally, Glenda, has your research on neurodegenerative diseases such as Parkinson's disease allowed for earlier detection and better treatments, and if so, how?
At the moment, it's not allowed yet for earlier detection, although there's those are things that we're working on, and that's because it's It's hard to actually do the screening for degenerative disease in the general population, to actually try to find who of the age population or a non-age population would actually have a degenerative disease. So the screening techniques that we have at the moment are too crude at the structural level. And so we have to go to the molecular level, which will take a little more time because that's a surrogate marker rather than a marker of looking at the brain itself.
Understand. Well, Glenda, it is a fascinating area of research. Thank you so much for your presentation and time today, and congratulations again.
Thank you.
Now, we're going to take a short break and be back for session 2 at 10:45 Australian Eastern Daylight Time. Welcome back to Science at the Shine Dome 2021. And don't forget, you can ask our fellows questions throughout the event by using the panel on the right-hand side of your screen. Now, on to our next presentation. Please welcome back Professor Helene Marsh to introduce our next fellow.
Associate Professor Lee Berger. Lee determined the global cause of catastrophic declines and disappearances of amphibians in pristine areas by discovering a novel pandemic disease caused by a chytrid fungus. Her research has made a major contribution to the recognition that spread of exotic disease is a threat to biodiversity and a serious consequence of globalisation. Now we'll hear from Lee.
Hi everyone, I'm Lee Berger. I'm a Principal Research Fellow at Melbourne Uni. I've spent most of my life working on chytrid fungus in frogs, but I have a broad background in wildlife disease research. I wanted to start by introducing our One Health research group. So we aim to find solutions for wildlife health issues to benefit humans, animals, and the environment. So we work on conservation diseases, zoonoses, and also diseases that spill over to livestock. Because few people work on wildlife disease, we often have large problems to ourselves, and so we need to be multidisciplinary. And so our group includes vets, ecologists, and molecular biologists. We tackle infectious and non-infectious disease in a large range of wildlife species. So starting with the overview of amphibian declines, this map shows where declines have occurred due to different reasons. So in the green, we have areas where losses have occurred due to habitat reduction. In the blue is areas of overexploitation or overcollection of frogs. And the red are these so-called enigmatic declines that occurred mainly in Eastern Australia, as well as Central and South America. So these were declines that occurred suddenly in high altitude protected mountain streams, mainly since the 1970s. And for a long time, these declines were quite mysterious. There was no obvious cause, but we now know that they were due to the introduction of the chytrid fungus that caused mass die-offs as it arrived in naive populations. So this is the fungus for Trichochytrium dendrobatidis, which I'll call BD. So we described it as causing the declines in 1998, and it was described as a new genus and species in 1999. So we now know that it originated in Asia and it escaped from there in the 70s. It belongs to this quite obscure phylum of true fungi, the Chytridium mycota. So there's been over 1,000 species of chytrids described, but a lot of them are environmental biodegraders. There are a few serious pathogens of plants, insects, and algae, but BD was the first to be found causing disease in a vertebrate. So this graph is a result of a global review led by our postdoc Ben Shiel, which quantitated the impacts of chytrid. And we found that 501 species had declined globally, including 90 extinctions. So that makes chytrid fungus the worst invasive species known. So this graph shows the number of species that in the year they first declined. And so you can see there's a peak in this pandemic in about 85. And by the time the cause was diagnosed in 98, we were getting towards the end of this epidemic curve. And so it was a very long time to diagnosis for the frogs. Early on, we did a lot of mapping. So this shows where the fungus occurs in Australia. It's mainly the coastal areas where it's wetter and cooler. The fungus doesn't survive in the arid inland areas. By looking through museum samples, testing archived specimens, we could work out the dates that it first occurred in different areas. So we think it arrived in 1978 near Brisbane and then spread north and south from there. And these dates of first occurrence coincide with the timing of the frog declines in those areas. So to look at whether it was going to keep on spreading, we had Chris Murray, a student at the time, make a MaxEnt model which predicts where the other areas of similar suitable habitat. And there was only a few areas where it looked like the fungus could spread to, and one of those was up on Cape York, and the other area was in southwest Tassie. So both of these areas are still free of the fungus, but do contain endemic frog species that might be susceptible. So these are areas of continued concern. This is what the fungus looks like in a section of frog skin. So it just occurs in those outer epidermal layers. Here we have a solid immature stage, and as the fungus matures, the cytoplasm divides up into more infectious zoospores, and these are released through the discharge tube. And so you can see the timing of that development of the fungus really matches the development of frog skin. And by the time the fungus has been carried outwards into these outer shedding layers, you only have the old empty stages of the fungus left. And so that suggests it's really well adapted to living in the dynamic tissue of the epidermis. So this is a cool scanning EM photo showing the surface of frog skin, and almost every cell here is infected, and you can see the discharge tubes poking to the surface. And on transmission electron microscopy, you can clearly see the fungus is occurring inside the cells, the frog cells. You can have 2 or even 3 sporangia inside a cell. And again, we see the immature stages in these deeper, more viable skin cells, and then the old empty stages in the shedding layers. So a big question was, why do frogs die from a superficial skin infection? And the answer is that frog skin is really important in osmoregulation. And so during infection, the sodium and potassium pumps are damaged, leading to loss of electrolytes across the skin, and they end up with very low sodium and potassium levels. So this graph shows that in a sick frog, potassium can get down to half what it should be in a healthy frog, and at that point, the frog undergoes heart failure. But yeah, you don't see any changes in internal organs by histology or biochemistry. It seems like it's just the skin damage that is enough to kill the frog. So Australia's had this threat abatement plan that was driven by Rick Spear. Initially, it was there was a large focus on ensuring we didn't cause spread to new areas, but we're now really focused on reducing impact in infected areas. So we know that 43 of Australia's frogs have declined from the fungus, 6 have gone extinct. We now have 6 critically endangered that really need intervention to survive. So many species can persist, but they often have reduced abundance and smaller distribution than they used to. There's been some clear evidence that 11 frogs have started recovering. So our current research, it's really aimed at those critically endangered frogs, hoping to reduce mortality in the wild. And so we aim to improve host immunity. We think that this is the most sustainable and feasible approach. So we're collaborating with experts in aquaculture who have proven methods that they can use to improve resistance. And so we want to firstly detect resistance genes by using a genome-wide association study. Normally resistance is polygenic. Normally it's due to a lot of genes of small effect. But when we have results from that study, that will help decide whether artificial selection or genomic selection is the most effective way to go forward. We also have studies looking at the role of increased reproduction in population persistence. So we've seen that some species in the wild continue to have very high mortality rates, but the populations are surviving due to very high recruitment. And we also have continued work looking at pathogenesis and trying to get a better understanding of the mechanisms of pathogenesis in this disease. So I just wanted to finish by bringing up the fact that wildlife health in Australia has been neglected for a long time. It tends to fall in the gap between health and conservation. And in the past, national research and training has been ad hoc. It's generally driven by short-term opportunities rather than a strategic plan for the country. So at the moment, the Melbourne Vet School is leading a number of initiatives to establish an Australian Wildlife Health Institute to try and get long-term and increased resources for this really important issue. And that work's being led by Lee Skerritt, and we have a lot of support from the new head of school, Anna Meredith, who also is very keen on improving wildlife for Australia. So I'm really grateful to a lot of collaborators, students and staff over the decades. I've just listed here our current group of wonderful students and also our current funding. So thank you.
Joining me live now is Associate Professor Lee Berger. Welcome, Lee, and congratulations.
Yeah, thanks very much.
Now, I wanted to start by asking you, why isn't chytrid fungi so much of a problem in Asia? Have frogs there developed resistance to it?
That's right. We think it evolved there, and it's going back. People can find it back for over 100 years in museum specimens. So just think it's been there a long time, and the frogs are resistant. If we just looked at those frogs, if it had been found in those frogs, it just looked like a harmless skin parasite. There was no indication that it was a deadly disease. If you just look at Asia.
Professor Helene Marsh has written in and says, congratulations, Lee. Do you think that the recent mass deaths of frogs across eastern Victoria, New South Wales and Queensland is likely to be due to chytrid?
Yeah, there's a lot of investigation still underway. So that does look like, the frogs that are dying are generally have heavy infections with chytrid. And we're not sure why it's worse this year than previous years, whether it's an environmental change, just a colder weather after some bumper seasons for frogs, or if there's been some change in the fungus or in there's a lot of people trying to work out why it's got worse. But I can't answer that at the moment.
Work to continue. I'm sure it's a range of things too. Now, Francis Saparovich has also written in, lovely work, Lee. Is there evidence that frogs are changing the range of antimicrobial peptides from their skin secretions to adapt to these environmental changes?
That's a great point, Francis. There is actually. One of our former students has spent the last 10 years in Panama, where they were able to study populations when the fungus first arrived, and then 10 years later they've had this very intense monitoring. And one thing they noticed when the frogs, some of the species have started coming back in the rainforest, and they think the reason for that was probably the evolution of stronger peptides. So they also looked at the fungus in those areas. They isolated fungus early on, and 10 years later, and the fungus hadn't changed, hadn't reduced its virulence. It looked like it was the host that was coping better.
Could importation of the fungus have been avoided? Did we know it was a possible threat before it came?
No, it had never been found. It's spread for 20 years before it was even discovered. And I think that's the thing with wildlife diseases. It's impossible to predict. And even if we had known about it, as I said, it just looked like a harmless skin parasite. So there was no way to predict this was going to happen. It really relies on keeping our borders strong and having good biosecurity and being ready for the unknown pathogens to come. Yeah, you can't just rely on testing for known diseases with wildlife. The only thing you can do is to have very, very strict quarantine.
Yeah, and it really highlights the breadth of measures that are needed to monitor and identify these sorts of threats, isn't it? Now, are there other fungi that cause diseases in vertebrates?
Other chytrid fungi. There's since this BD, the frog chytrid was found, another one emerged in salamanders. So that was in about 2013. It was discovered in Europe, and it was same. It also originated in Asia, arrived in Europe, and started killing the salamanders there. But yeah, they've discovered that one much earlier, and there's been a lot more action with that one. And so America is still uninfected with this salamander chytrid, but they have a huge diversity of salamanders, and so they've very quickly stopped importing salamanders, and yeah, hopefully they'll be able to keep it out. It's hard. It's hard to do.
Yeah, and just finally, can you tell me anything about perhaps the 6 frog species you mentioned that we've lost, but even also the sorts of species that you're perhaps particularly focused on in your research now?
Yeah, so the ones that have gone extinct were all rainforest frogs from Queensland. So they crashed very quickly when the fungus arrived. Yeah, the forest just went quiet, and within a few months the populations had gone. But the alpine frogs have had a much slower decline, and so some of these areas the fungus arrived 20 or 30 years ago, and they're only at the point now where they're going to go extinct without our intervention. And so yeah, it's a lot of the two crobbery frogs, the other alpine frogs are there's a lot of attention in captive breeding, and they're the ones we're focused on with our work, trying to improve their immunity.
Well, thank you so much for your presentation today, Lee. Congratulations again.
Yeah, thank you very much.
Please now welcome back Professor Helene Marsh to introduce our next presentation.
Next, we'll hear from Professor Stephen Chown. Stephen is a preeminent authority on the biodiversity and conservation of Antarctic systems and the threats posed by environmental change. He's revolutionised the way in which information on the environmental responses of animals is understood at global scales, unveiling not only the nature of adaptation, but also the way in which its limits constitute problems for life in changing circumstances. He's a leader in science and conservation policies for the Antarctic. Now, to Stephen's presentation.
Well, thank you. It's a great pleasure and privilege to be with you here today to share elements of my work. My work is about biodiversity variation, the conservation of that variation, and the fundamentals of biodiversity and its conservation in the Antarctic region. I'd like to dwell on 4 examples with you today. The first is about physiological adaptation at large spatial scales, a field my colleague Kevin Gaston and I have called macrophysiology. Second, the consequences of that large-scale physiological variation for conservation, and in particular during times of climate change. Third, what those consequences are for the Antarctic region. And finally, the implications of our discoveries for Antarctic policy and how those have come to be accepted by decision makers and to alter the way we do science. Before I begin this, just a reminder that it is about life's natural diversity, the species with which we share the planet and for which we're responsible, making, of course, meetings such as COP26 that's coming up not only important for ourselves, but also important for the species with which we share the planet and which are facing a very changing set of extinction probabilities. So let's start with physiological variation at large spatial scales. And in this case, thermal tolerance, the ability to survive either high or low temperatures. Of course, much had been known already by the 2000s or thereabouts about animal physiological tolerance, and especially of heat and cold. We looked at insects right across latitude and discovered, amazingly, that upper thermal tolerances show much less variation than lower ones. You can see that the patterns found in the insects themselves look very similar to environmental temperature variation across the globe. This is in the case of the Americas here, indicating very low temperatures in the Arctic region. The Antarctic is not illustrated here. And a decline in upper temperatures, but very much less variation in insects, but a reasonably good match. This was the first time demonstrating that there are these real differences between upper and lower thermal tolerances across latitude in insects. And they have some surprising implications, especially on the upper end. So when we think about environmental temperature variation and how that's changing with climate change, and we think about insect and other ectotherm, that is frogs, reptiles, and so on, tolerances, then it turns out that in the tropics to about the horse latitudes or 30 to 35 degrees or thereabouts, there's huge risk. There's not much tolerance of warming, either current warming or future warming in these regions. In fact, some populations seem to be already below the amount that they can tolerate. And it turns out that there's indications of extinctions in these areas already for insects and for reptiles. Towards the polar regions, there's much more tolerance of warming. So as climate change proceeds, if we don't get it right at COP26, we can expect to see extinctions across these regions outpace those elsewhere. Of course, this is for indigenous species. We also know that we've introduced other species, either intentionally or accidentally, to the places which we live in. So in Australia's case, we have significant biosecurity to prevent this, especially the prevention of accidental introductions, which can cause huge problems. But as it turns out, the species that have already made it here are really quite different to indigenous ones. So these are small soil organisms known as springtails. And it turns out that if one measures their thermal tolerances right along the east coast of Australia, then one finds that the invasive alien species are much more tolerant. They have greater warming tolerance by 5 or more degrees than their indigenous counterparts. And that's right across latitude, from the tropics to the temperate areas. So the expectation is that as climate change plays out, we'll see that the invasive alien species prosper relative to their indigenous counterparts. Now, these kinds of large-scale patterns can also be seen in the Antarctic. We, for example, looked at what seeds people were bringing in with their luggage or their clothing, either scientists or tourists were bringing in to the Antarctic, and what the properties of those propagules or seeds actually are. And it turns out when you do the analysis that there's a high risk already of the establishment of invasive alien species in the Antarctic Peninsula region. Now, we undertook this work during the International Polar Year of 2007 to 2008. And when we published it in 2012, that year, the invasive grass Poa annua was discovered in this region. And we've seen a growing number of non-Indigenous species establish on Deception Island, on King George Island. So our forecasts were correct, and biosecurity has now been enhanced for this area. And I'll demonstrate that in a moment. Our work has also demonstrated that these significant interactions. So with greenhouse gas emissions, we not only warm oceans, they become acidified, there's increased risk of invasive species, of course there's increased transport with tourists and scientists, and there's a great many interactions that can play out. And together with colleagues such as Stephen Toole and others who are working in the physical sciences side, we've shown that if we don't meet our obligations under our agreements, such as the Paris Climate Conference agreements, if we don't actually get to net zero by about preferably 2040, then we're going to change this region significantly, not only through melting ice sheets, which will then affect our own coastlines, but a complete change to the ecology of the area. But we still have the opportunity to do so, emphasizing the significance of COP26 and the decisions we make there. Of course, this information will go nowhere if we don't take it to policymakers, such as those that will be making decisions at that meeting. And we've not only conveyed this information to the international arena, but also to the Antarctic Treaty parties who are responsible for the implementation of local conservation actions in the region to either mitigate or adapt to some of the change that's coming. And this work has largely been done through bringing the outcomes of our research and others to the Antarctic Treaty Parties at their meetings. So I've attended these meetings for the past couple of decades, all the while ensuring that the best available evidence from our research, from others' research, makes it to the treaty parties so they're well informed to take their decisions, which will be based on a range of factors, but really require the very best science of the time coming to them. And an example here on the right-hand side is illustrated by this question of where are unvisited areas of the Antarctic continent? The Protocol on Environmental Protection to the treaty actually requires that these are conserved. To date, nobody has known. And where's wilderness, which is also required to be conserved under the protocol? And our work looked at where humans have been. The red dots you see here are in fact not land, although some of you may think it looks like the Transantarctic Mountains or ice-free area in the Antarctic Peninsula. The red dots are human visits over the last 200 years, including very large traverses through to the South Pole, as you can see. And the purple areas indicating where humans, as far as we can tell, have not been. There's not much left of the continent that hasn't been explored. But on the positive side, negligibly impacted areas, areas that have been very rarely visited, are still very abundant. And many of these encompass ice-free areas where most of the biodiversity is located in the Antarctic. In consequence, there's a very great opportunity still to conserve the wilderness of the Antarctic region. Indeed, it remains one of the largest wilderness areas on Earth. Of course, the work we do is not only designed to have an influence and to inform decision makers, but it also has practical utility. So, for example, our work on biosecurity was translated into a very simple checklist for supply chain managers of national Antarctic programs, working with a council of managers of national Antarctic programs. Very simply, what do you have to look for on ships traveling to Antarctica, for aircraft, for stores, for food, for clothing? And this simple brochure has been remarkably effective. Here's an example of it posted at Scott Base in the Ross Sea region. This is a New Zealand station, indicating what might be required also for transfer into the field. So it's not only propagules that are coming into the Antarctic itself, seeds and insects and so on, but also how we may transport them during our field research. In turn, our work has also indicated that the policymakers have some priorities for science. So they're not only fundamental questions that remain to be resolved for the Antarctic and for which the Antarctic can be used to answer, for example, through astronomy, but there are also some very specific questions that are about biosecurity, about our human presence, about changing ice sheets. And as a consequence of the work I've done, I've been involved in ways in which we can, with the community and listening to both the science community and the policymaking community, set priorities for Antarctic science into the future. And these efforts have translated into a whole range of science plans for Australia, for example, and elsewhere. So the work I've done has covered these areas, moving between science, policy, and the way we can set about ensuring that we have a livable world. And with that, I'd like to thank several colleagues, including my partner, Melody McGeoch, my colleagues at Monash University and elsewhere, funding agencies for support, and Monash University, and particularly those who invited me to come to work here. And thank you finally to you for your attention.
And I'm delighted to have with us now Professor Stephen Chown. Congratulations, Stephen, and thank you for your presentation.
Thank you, Paul. It's a real pleasure to be with you here today.
I wanted to start off with COP26 because you mentioned it during your presentation. Have any of the outcomes so far given you any hope?
I would say that the outcomes are not as powerful as they should be. I think they're a little disappointing. Of course, I'm more inclined to think that we should be concerned about what civil society and business does, because I think that we're seeing that real decision making and action is being transferred actually into the hands of civil society and business, and that our reliance on the majority of politicians, not all of them, that are at the more senior levels has to wane in this effort. I think we're going to be really reliant on what we do rather than what we expect our leadership to do.
Yeah, and you also mentioned during your presentation that ideally net zero would be reached by 2040, whereas commitments have so far been centred around 2050. Can you give us some idea from your perspective on the difference that makes?
Well, I think the best place to go and have a look for that is through the special report on 1.5 degrees of warming from the IPCC. And it's pretty clear from that report that there is a substantial difference. I don't think that if we push it out to 2050 or 2070, as for example has been stated by India, we're going to see ourselves able to keep warming below 1.5. And that has catastrophic implications for us. It has sea level rise implications, but in Australia, we've seen it through the 2019 fires and the predictions that Ross Garneau made in the report that came to pass in 2020. I think well illustrated also through Bronwyn Adcock's book, Currie. And if we don't get to things by 2040, I think we're in real trouble.
We've got some questions coming in. Francis Aparovich says, wonderful work, Stephen. Having visited Antarctica with 100 women, I came away inspired, but also feeling a bit guilty. Do you think we should further limit Antarctic tourism?
I think we have to consider the extent to which our visits not only promote the Antarctic, in other words, develop an appreciation for its significance in the Earth system, but also have a direct impact at the same time. And I do think that work that's exploring cap and trade schemes, for example, is really important to understand the balance. So my answer to that is I think we have to, but we have to do it in a sensibly informed way. And that work is currently on the go to decide how best to do that.
And Peter Currie has asked, given the invasive nature of species you mentioned and the proliferation of human occupancies in bases on Antarctica, should this be more highly regulated internationally?
Yes, I think the Antarctic Treaty Parties are pushing forward biosecurity as one of their primary, it's in fact the #1 goal of the Committee for Environmental Protection. But I do think there's additional emphasis required to be placed. Most of the regulations in this respect are currently through resolutions of the treaty, and those resolutions are actually encouraging, they're hortatory, they're actually not mandatory. And I do think there's some room for the regulations to be more mandatory than they currently are.
A couple of questions about your presentation as well here. Why is there more tolerance for warming in some regions compared to others? And why do alien species have more tolerance than indigenous species?
The first question has to do with the difference between what species are capable of tolerating and the maximum temperatures found in any area. So it seems like losing high temperature tolerance is not very costly, and so maintaining it doesn't have much of a cost. So you find high temperature tolerance very similarly across latitude, whereas of course the maximum temperatures are lower in the temperate and polar regions than they are in the horse latitudes. They actually decline a bit in the tropics too. So that certainly explains what's going on there. Why there's this difference between indigenous and invasive species is a current research topic of ours. And in part, I think it has to do with many invasives come from disturbed areas, and disturbed areas often have high exposure. There's little vegetation. It's eroded quite frequently, either, for example, through fire or through flooding or through other forms of disturbance. And that may mean they're exposed to high temperatures more frequently. They're also capable of tolerating difficult conditions. They're weedy, so to speak. That makes them more easily transported. But that at the moment is a working hypothesis. And I think there's a bit of research required still to go into that, including the mechanisms as to why it seems to be so evolutionary labile in some groups, but not in others.
Well, Stephen, thank you so much for your work in this area and for your presentation today. Congratulations again.
Thank you, Paul. Great pleasure.
Now, to introduce our next fellow, please welcome back Professor Helene Marsh.
Next, we'll hear from Professor Catherine Lovelock. Kath studies the impacts of climate change on coastal wetlands. She also looks at the role of coastal ecosystems in mitigating climate change or blue carbon. Her research has focused on mangrove ecology, on the impacts of sea level rise on mangroves, and on carbon storage in mangrove ecosystems. Kath contributes to national and international policy to implement conservation and restoration of coastal wetlands in order to enhance the resilience of coastal communities. Now to Kath's presentation.
It's a great honour for me to be able to present my research to you today. And I thank my colleagues and the organisers to make this possible. About 2.4 billion people live in the coastal zone, and they live embedded in about 1 million square kilometres of coastal ecosystems that include mangroves, salt marshes, seagrasses, coral reefs, and rocky reefs. Together, these natural ecosystems and the human societies form a complex socio-economic system that is the focus of my research and also inspires my research. So both human societies and the ecosystems that they're associated with on coasts are exposed to climate change. And these climate change pressures include increasing intensity of cyclones, increased frequency of coastal flooding, sea level rise, and marine heat waves. In addition to the physical impacts of, for example, extreme storms and climate change, this is also occurring on a background of intense human activities in the coastal zone. So agriculture and aquaculture are practiced on the coasts often at the expense of coastal ecosystems. We have intense urban development. For example, you can see in the lower picture of the Brisbane airport that's embedded in a system of coastal wetlands. And we have industry on the coast. And the picture on the right is of salt production in the Pilbara in Australia. These intense human activities on coasts have resulted in degradation to coastal ecosystems where they've been converted, and the example here is the widespread conversion of mangroves to aquaculture in Southeast Asia. They've been damaged by overexploitation, for example, this mangrove forest that's been harvested for timber and charcoal in Madagascar, and they've been removed, for example, salt marshes in the south of Australia that have been converted, to example, for rubbish tips and then filled. The reason why we care so much about what happens to coastal wetlands is the enormous amount of ecosystem services that they provide and the importance of these ecosystem services to the maintenance of healthy and resilient coastal communities. Coastal wetlands provide cultural services. They're important for recreation and tourism. They support fisheries and biodiversity. They're important for forest products. And this picture on the right here is a woman in Myanmar selling mangrove timber for cooking because people there have no other options to support their daily lives. They're important for coastal protection and flood mitigation. And coastal wetlands improve water quality by trapping nutrients and sediments, incorporating them into their biomass, undergoing biogeochemical change. And then finally, they're important for for carbon sequestration in the coastal zone. And this has been increasingly acknowledged over the last decades, and I'll return to that topic in a moment. So one of the parts of my research that has been important in understanding the impacts of climate change, and also how coastal wetlands are going to respond and can adapt to climate change, is monitoring of their resilience through monitoring how they maintain themselves in the tidal frame relative to sea level. So if you want to have the same kind of coastal ecosystems, or coral reefs for that matter, those systems have to be maintained in a very similar zone relative to sea level rise. And the way coastal wetlands manage this is through vertical accretion and also landward migration, so changing their distribution over time. So with a network of collaborators in the Indo-Pacific region and globally, we use a common methodology to monitor how the surface of coastal wetlands is changing over time. And the bottom panel basically describes what these systems look like. They involve a whole lot of stainless steel rods being placed down into the sediment to form a benchmark, a firm benchmark from which we can measure millimetre changes in the elevation of wetland surfaces over time. So the instrument looks a little bit, when it's being measured, looks like the picture in the right lower panel. So these monitoring systems, these data have underpinned a range of models. What they've done is really help us understand that there's variable levels of vulnerability of coastal wetlands globally, and that sediment supply, which comes from the catchments often and is altered by things like damming and modification of catchment processes, but sediment supply is key. And the other factor that is really important is avoiding coastal squeeze. So basically allowing the space for wetlands to migrate landward with sea level rise. So the kinds of models that this data have underpinned, there's a recent one shown in the lower panel here, and that is a model of how our ecosystems will change with sea level rise over time, and how well our reserve systems basically will capture those changes. So the other element has been really, and it's highly linked, that this element of my research is that of blue carbon. And hopefully you've all heard about blue carbon. So blue carbon is the carbon that's stored in the soils of vegetation of coastal wetlands. If you look at the picture, you can see it's not blue, but brown. And that's just because of the high level of organic matter that is within these systems because of their high level of productivity, and also the fact that inundated sediments slow decomposition. So carbon accumulates in these systems over thousands of years. So blue carbon also describes this imperative to conserve and restore these ecosystems for their ecosystem services like carbon, and for reducing greenhouse gas emissions to the atmosphere, but also for fisheries, coastal protection, and all these other ecosystem services that they provide. So prior to 2013, no nations were asked to account for changes in their coastal wetlands over time. So they weren't asked to account for the emissions that result from the disturbance and conversion of these systems, or the good work that was happening with restoration, which enhances carbon sequestration within the coastal zone. But with the IPCC guidance in 2013, this has provided incentives for further investment. And in our own country, Australia has supported the International Partnership for Blue Carbon, and also is in the process of developing the first national blue carbon method for a carbon market. So it's this, these incentives that blue carbon provides that we hope will really incentivize and accelerate restoration and rehabilitation of the coastal wetlands that have been lost from the world. So we are in the business of looking for climate smart restoration opportunities, for characterising the benefits from restoration, and also really trying to understand the enablers for restoration. So with that, I'd like to thank the University of Queensland, the Australian Research Council, ARC, and CSIRO for their support, and also for the numerous colleagues that have joined me in these research endeavours over the years, and finally to my family and friends.
And Kath is currently on her way to COP26, so she's unable to join us today for her Q&A session, but we are so glad that we could hear her presentation, and we wish her well in Glasgow. Please now welcome Academy Treasurer, Professor Marilyn Anderson, to introduce our next presentation.
Thank you. Next, we will hear from Professor Barry Pogson. Barry has revealed how communication between different parts of a cell influence growth, development, photosynthesis, and tolerance to drought. He has enabled new insights into the evolution and function of retrograde signalling networks in gene expression, photosynthesis, leaf development, and even water loss during droughts. Barry has actively sought the translation of his basic research into beneficial outcomes and mentors upcoming leaders in science, policy, and industry. Now to Barry's presentation.
Hello, and thanks so much for this opportunity to speak to you about my research. You know, there's 6,500 ways to say hello, and most of them I do not understand. And that's for one species. So what about plants? How do the different compartments within a plant cell communicate with each other? And how are those different languages integrated across time and space? Do they vary? Are they conserved? Our languages evolved over time and space. Words take on new meanings, words diverge, and there's an increasing complexity of languages just within one species. How does this work in plant kingdom? And why do we care? We care because we need high yielding and more resilient crops. In the next 50 years, we'll need to harvest as much cereal grain as being produced since the beginning of agriculture 10,000 years ago. And that's against the impacts of increasing temperatures that will bring about more severe climatic events such as droughts. But the problem here is resilience and yield are in a trade-off. Increased resilience typically decrease yield and vice versa. The challenge here is to break the paradigm and increase both. So these are 4 questions that have driven my career. What are the languages of chloroplasts? How are the languages integrated across time and space? Are there crop-based solutions for food security? And how do we prepare the next generation of leaders? But first, why do we even care about communication within plants? Because of the potential to regulate thousands of genes within the nucleus. 30,000 genes in the typical nucleus. Between 3,000 to 5,000 of those are either directly targeted to the chloroplast or involved in chloroplast function. And they need to be able to respond to dynamic and sustained changes in the environment. Just look at my solar panels across 3 days. Bright sunny day, risk to the plant is oxidative damage. Cloudy day with sun spikes, risk to the plant is these rapid adjustments will again lead to damage. Followed by a rainy day, risk to the plant, not enough light to grow, and it can't just plug into the grid. It really has to ramp up its ability to capture light in a day like that. So how's this going to be coordinated? This image shows the hundreds of chloroplasts within a given cell, all communicating back to the nucleus. The nucleus needs to, as the CPU, needs to integrate that and control the signals coming back out and regulate the expression of genes to enable rapid and sustained acclimatory responses. So how have we gone about doing this? Is a project that's involved just about everybody in my lab over the last couple of decades, first starting with Bart Roussel and Paulsen, and then moving on to so many people I can't show the photos on the slide. The initial screening was to look for mutants that disrupted this signaling pathway. And we did that with a stress sensing promoter and looked for mutations that affected aspects of chloroplast signaling. I'm only going to talk about one pathway today, but it's worth noting that these retrograde signaling pathways, as they're called, are multifaceted with many components and many different signaling mechanisms. So we found a protein called SAL1, located in the chloroplast, that when it's inactivated, constitutively activates stress sensing. It's a negative regulator of a retrograde signaling pathway. It accounts for about 1/3 of the thousands of genes regulated by high light in a typical plant. It itself is a sensor of the high light. It's inactivated by the production of reactive oxygen species, forming an inactive dimer. Once it's inactivated, the substrate, it's an enzyme, accumulates. The substrate that accumulates is a phosphoadenosine called PAP. It's AMP with an extra phosphate. PAP can move between the chloroplast and the nucleus, and in the nucleus it binds to a class of proteins called exoribonucleases. And in binding to those proteins, it promotes a novel mechanism of gene regulation whereby there's upregulation of a downstream proximal gene. I won't cover that today, but happy to talk about it with anyone who's interested. So retrograde signaling is typically being viewed as bilateral communication between the chloroplast and the nucleus. But I wanted to test the hypothesis that the chloroplast is actually acting as an environmental sensor for the entire cell. And if that was the case, then we might expect in different cells that the chloroplast has taken on different functions, and these retrograde signals have different effects. We tested this by looking at the guard cells of stomates, stomates being the pores that control water loss within plants. You can see the addition of PAP is closing these guard cells. And it's doing it by activating a reactive oxygen species burst and stimulating calcium signaling networks. This is analogous to the classic ABA stress hormone that regulates guard cells, but it's operating in parallel and it's acting different components of these networks. So there's something quite unique and interesting here where we've got the classic ABA signaling network and in parallel, this chloroplast sensing network that ends up in a point of convergence on regulating stomatal function. And it gives us this view that, yes, it is functioning as an environmental sensor and doing different things in different cell types. In leaf cells, it's quenching the active oxygen species. In guard cells, it's producing them to activate a signaling network. Significantly, this pathway is conserved across all land plants that we've tested. Any plant with a functioning guard cell closes in response to PAP and has this ROS and calcium regulation that we've seen in Arabidopsis. And that includes crops like barley. So it evolved a long time ago, probably around the emergence to land, which is around the same time as a lot of the major hormone pathways. Let's move on to the next question then. Can we break the yield resilience trade-off in crops? This is our knockout of SAL1, where we get constitutive activation of retrograde signaling. You can see it's a very happy plant without water, but equally, it's a very abnormally growing plant. It's slower growing, it's late flowering, and it's got changes to morphology, many of which are not good traits in a crop. And this is the problem with the yield resilience paradigm, is quite often people have just simplistically taken a model plant gene and tried to phenocopy that effect in a crop. The reality is we've got to think a lot smarter and clever about this. One of the things we need to think about is titrating the activity of that gene. So it's a full max during stress, but wound back when the plant's under stress. One way of doing this is to do synthetic biology or directed evolution. We have this crystal structures, we know the active site, we know how the protein works. So we're starting to fine tune that protein to see if we can come up with new ways where it'll be a better regulator of stress signaling. The second way, and simpler way, is just to take advantage of the fact that there's 7 SAL1 genes in wheat. So if we just knock out different combinations of them, we can also titrate back the activity between min and max and end up with a normal growing plant. We've done that and we've started to grow these lines across field sites across Eastern Australia. This is just to remind you the scale of an experiment is a lot bigger when you get into crops from model systems. We found that, as we'd expect, if we knocked out too many SAL1 genes, we got increased resilience and decreased yield. But in at least one of our lines, we got increased resilience and increased yield. And that is an interesting interaction between SAL1, again, having functions in stress resilience and broader functions in developmental biology. A promising start, a lot more to happen, and a lot more to figure out if it's going to carry forward. The final thing I want to cover in this talk is to talk a bit about my passion for mentoring and what I see as the global challenges needing a new generation of researchers. Playing in the background is a video former members of my lab produced a few years ago, talking about the need to create global networks of scientists and talking about different mentoring strategies that they found valuable. In addition to thinking about just the individual strategies of one lab and the networks we can build, we've thought more broadly about that. And we've now got funding from the Australian Research Council for a training centre for future crops. This training centre is really quite exciting. It's a partnership between ANU, University of Adelaide, and 19 other partners, including many, many industry partners from across Australia and internationally. The students will be taught cutting-edge technologies in gene editing, such as CRISPR, to produce the crops of the future. But they'll be doing it in partnership with industry to make sure they're working on traits that industry cares about. And we have a social science team that will be working alongside us to look at the economics, societal expectations, and regulatory frameworks for all of these products. Finally, I just want to thank the people who have made this possible. My lab brings me joy every day I go to work, and without them, I wouldn't be here. Many of my former lab have gone on to their own careers, and it's been a pleasure to work and engage with them. Many, many collaborators. I've just listed a few here. And again, I would not be here without the mentors. Again, some of you are listening in. You've been an incredible source of insight and wisdom. And finally, can't do anything without my family. Thank you.
I'm delighted now to have with us Professor Barry Pogson. Congratulations, Barry, and thanks so much for that presentation. Well, you might be on mute there, Barry. If you could just click off the mute button.
The universal syndrome of leaving yourself on.
It has to happen at least once. So congratulations again, Barry.
Thank you. That's great.
Now, I wanted to just start off by mentioning those increases in grain production that are required over the next 50 years. It's pretty confronting when you look at the chart that you showed us. Can you tell me a bit more about the kind of grain increases that you're seeing in your field trials?
Look, these field trials aren't going to solve world hunger, right? You know, what we're seeing is around an average increase of 6% across multiple sites and seasons. Sometimes it's up to 20, sometimes it's 0 for that particular line. And that's one variety of wheat grown in southeastern New South Wales, right? It's what we really need is stuff that's global. And really all of us are working on global things. The speaker following me is going to talk more about different ways too that will impact yield, right? So it's a global thing and it's going to require a hell of a lot of effort, just like all the things we're having to deal with on climate change. Feeding the world is a global challenge.
Oh, it's a fair point, but still really important work, and great to see that the piece that your work is playing in answering that question. I've got a question that's just been sent in by John Patrick, who says, "Congratulations, Barry. Do you suspect that the Australian flora has evolved yet to be discovered retrograde signalling pathways that may confer higher levels of tolerance to impacts of climate change? And could these traits be transferred to crop plants?"
Yeah, look, I think there's a fascinating idea there about starting to investigate Australian flora, right? And the extent to which Indigenous agriculture has influenced both their selection or at least the ways in which they were grown and can be grown. So I think there's a huge potential to start to think about learning from our Indigenous colleagues and forefathers and First Nations peoples about what they farmed, how they farmed it, and how they modified the landscape to optimize the farming procedures. And then when that in itself could be a great source of new grains and nutritious grains. Some of the Indigenous crops have far higher protein content than the mainstream grains. Some has as much protein content as chicken. So there's nutritional benefits as well to start to think about bringing them into our diet, as well as the point John Patrick raises about moving traits from one species to another.
Yeah, and you make a really great point there, referencing what we can learn from First Nations people. I wanted to ask you a bit about leadership and mentoring. They're clearly extremely important to you. Can you offer any ideas of how perhaps the system could be improved to better attract and support both women in STEM, but also Aboriginal and Torres Strait Islander researchers?
Well, I'm part of the problem. I'm a white male, right? So we have to get better at it. And I think we're only going to get better at it when we start to put real programs in place. You know, we've got to put in proper gender equity programs. We've got to put in support for childcare, for conferences that enable women to travel with or without their family. They need support for those sorts of activities. The horrible loss of talented female scientists as they move through the progressions from, we have in biology, we have equal numbers around the time of a PhD, and it just gets further and further away from 50-50 as the careers progress. So there's got to be active measures and active investment. And a lot more decisions and leadership have to be done by people other than white males too, right? We really need to make sure that there's women in the room who are informing us about what they need and how they can advance equity.
Thank you so much for that, Barry. I wish I had more time to ask more questions, but that is all the time we do have for now. Thank you once again for your presentation and congratulations on your election to the Academy.
Cheers.
Please now welcome back Professor Marilyn Anderson to introduce our next presentation.
Thank you. Our next presentation is from Professor Andrew Harvey Millar. Harvey's discoveries underpin our understanding of respiratory damage during plant cell aging and disease. He discovered key biochemical mechanisms through which plant respiration is adapted to cellular processes and harsh climates, and identified how damage by lipid, oxygen, and nitrogen radicals lower the efficiency of respiration. Please welcome Harvey.
My name's Harvey Millar. I'm at the University of Western Australia in Perth, and I'd like to use the time available to me to introduce my research to the Academy on mitochondria and respiration in plants. Plants capture carbon dioxide and convert it into triose-phosphates and ultimately into carbohydrates through the action of photosynthesis, releasing oxygen. But plants, just like us, also need to break down some of those carbohydrates through respiration to make chemical energy in the form of ATP for the cell, reducing oxygen to water and releasing carbon dioxide. In the light, it's hard to tell that plants are actually respiring because photosynthesis dominates, consuming carbon dioxide. But when we measure respiration in leaves by turning out the light, we can watch the carbon dioxide release from the respiratory process in plants. But that tends to indicate that respiration is a rather trivial process, maybe only 7 to 10% of the photosynthetic rate. But when you consider the whole life history of a plant, the night when there's no photosynthesis, but respiration persists, roots where photosynthesis doesn't occur, shady days, and the development in plants, then more like 50 to 70% of the carbon dioxide that's taken up by photosynthesis is actually lost by respiration. It's important to step back and realise that we've only known about respiration and oxygen for a short part of human history. In 1775, both oxygen and animal respiration were identified and discovered, and only a decade later, plant respiration. But then there was a considerable period of time where respiration in plants was denied and the field languished, until ultimately mitochondria, literally the thread bundles in cells, were discovered, these bacterial-like structures in cells in both animals and plants. And then there was an active period where respiration was monitored and mitochondria were isolated to understand their role in the respiratory process. And then finally, more recently, a very large range of mitochondrial processes and the array of enzymes that make them work have been uncovered. But while humans only figured out mitochondria recently, in fact, they've been around probably since 1.5 billion years ago, when a bacteria entered another cell, set up house, and over hundreds of millions of years, gave away their DNA to the host of their cell, becoming dependent endosymbionts of the eukaryotic cell, ultimately their own energy factories for the cell. This transfer process means that while mitochondria today contain about 2,500 different enzymes and other proteins, these are scattered in the nuclear genome and need to be found in different organisms to assemble in our own minds what mitochondria actually are. So in my lab, we've been asking, so what are the proteins inside mitochondria in plants? My research group in Perth has been active in this area for the last 20 years, developing detailed maps of the proteome of mitochondria in plants, using peptide mass spectrometry to define the genes that encode mitochondrial enzymes in plants. Having made these lists, we've wanted to better understand how they influence the respiratory process. We ask, how often are mitochondrial proteins made and degraded? We've developed stable isotope labeling approaches so we could define the age of mitochondria and the age of the protein inside them. This has shown that plant mitochondria are not just a structure built and developed in the cell, but more like a much loved old car. They have protein complexes of different age inside them. They're constantly being maintained, updated, and repaired by the cell. Using these same techniques, we've been showing how protein complexes are put together, organized and assembled, and how these different parts of the respiratory process are involved and what assembly factors are involved in the process. Having made lists of composition and dynamics of their maintenance, we've wanted to better understand how they are integrated into the unique metabolism of plants. To give just 3 examples, we've recently been discovering the role of kinase signaling pathways in setting respiration rate at night in plants. We've been genetically dissecting the redundancy of pyruvate provision to plant mitochondria, showing 3 separate ways that support plant respiration. And lastly, and most recently, we reported for the first time the carrier that carries and allows mitochondria to exchange a malate for a citrate molecule, which is critical for plants to build amino acids using citrate as a building block. The point of all this respiration is not just to continuously consume oxygen and release CO2, but rather it's to make ATP for biosynthesis and growth. So we've wanted to know, what is the ATP exported from plants actually used to build? One of the largest sinks of ATP in cells is making proteins in other parts of the cell. In fact, 25 to 40% of all ATP is used in plants and also in us. That's 5.5 ATP for every amino acid attached to a growing polypeptide chain. Using the stable isotope techniques that we developed for looking at mitochondria, we've started to map and explore the rate of synthesis of many different proteins in plants to determine the energy use that's involved in building the proteome of leaves, looking at the way in which this varies in the day and the night, and most recently, how this process defines the upper limits of protein content in wheat grains. This is all aimed to embed respiratory research into the energy biology of plants, from mitochondrial maintenance to respiration rate, to how this provides the energy needed by plants in harsh and changing environments. Right now, we're at the cusp of a new era in biology, where we don't just document plant mechanisms and delete genes, but we build systems to optimize their performance. It's been said that you don't really understand something until you can build it. For respiration, this is about predicting processes that seem inefficient or odd and redesigning how they could work to see if we can build the mitochondria we want rather than the one evolution has provided for us. In 1775, when Joseph Priestley discovered oxygen and that air was only 20% good for breathing, because most of it was nitrogen, he concluded somewhat philosophically, the air which nature has provided for us is as good as we deserve. But what Priestley didn't know at the time is what processes actually provide that air. But in fact, our air is defined by plants and what they do. And it's a gift that we can give to the next generation, the quality of the air that they breathe and the knowledge of how to modify it for their own children. As a scientist, I'm also the child of some, and I also have my own scientific children. Here I especially note David Day and Fraser Bergerson, both fellows of this academy, and my PhD supervisors, who introduced me to mitochondria and respiration. Without them, I would have nothing to talk to you about today. My scientific children are the past and present members of my laboratory who have undertaken so much of the research I presented today and will take it on into the future as they start their own scientific journeys. But I can't give my PhD supervisors and the environment they built all the credit. It's also genetic, because I'm a third-generation scientist. My father, a computer scientist, and my grandfather, an industrial chemist. They both instilled me with a Christian faith as a way to live, as well as a scientific method as a way to explore the world. I also couldn't have done my science without the love and support of my own family, my wife, Glenda, and daughter, Jessica. Thanks for your time today.
And we have with us live now Professor Harvey Millar. Welcome, Harvey, and congratulations.
Thanks very much.
A truly inspirational presentation. Can I start off by asking you a bit about your research? I wanted to ask you, do plant mitochondria, fungal and animal counterparts have any genes in common?
Well, historically, they have nearly all of their genes in common, but each of these organisms have also adopted a whole range of other processes, which are kind of unique. So there is things that plant mitochondria do that fungal mitochondria or human mitochondria don't, and vice versa to a degree as well. But plants tend to be accumulators, so they tend to take everything that everyone else has and just add some more things to it. Partly, I guess, because plants live in so many different environments that they need these different capabilities in certain cases. But there's a great amount of research that's done tracking through the connection between both the ancestral genes that all mitochondria have in common and the more recent eukaryotic genes that they've each captured from other partners who've joined cells in particular organisms.
John Patrick has written in with another great question. Says, congratulations, Harvey. From your discoveries of mechanisms regulating protein turnover in mitochondria, do components of these regulatory mechanisms present opportunities to improve tolerance of crop plants to abiotic stresses such as heat and drought?
Thanks very much. It's a great question. What we're finding is that the turnover of proteins is somewhat surprisingly varies really substantially. So probably varies over 100-fold differences in terms of how long proteins survive. We're only really just beginning to understand why that's the case. We imagine in our wisdom that things which break quickly, we could simply make them more tolerant and therefore maybe enhance plants in various ways. But of course, we really need to understand why plants are actually turning them over. So this is a complicated mix, possibly of accident and circumstance, but in some cases, particular proteins are actually damaged by what they do, or they're damaged by modifications that occur to them, chemical modifications that require them to be replaced. So we're taking a series of exemplar cases and seeing if we can actually modify and change these so that we can influence that process. Part of what we would like to be able to do is actually determine the timing of certain things being present in plants in a different way than they are naturally. And that may involve understanding and controlling both synthesis and degradation to enable that. And that may make greater efficiency in the system and connect what mitochondria are doing better to the environment that's around them.
Do we know if elevated levels of atmospheric CO2 enhance plant growth and plant respiration rates?
They certainly can. It depends on the plant species, and often it also depends on the temperature. So we have a complicated dynamic of the connection between carbon dioxide, which in theory should make plants grow better, certainly all of the C3 types, but the temperature rises that are occurring on Earth connected with that CO2 release aren't necessarily very positive for certain plants in certain environments. So it's a complicated mix. In a glasshouse environment, you could say CO2 is all good, but obviously we know in the real world CO2 is not all good, and that therefore this question of balancing is very important. But as we move forward and we actually see climates changing, we see temperature changing, and we see CO2 levels continue to rise, really it's very unknown yet exactly how this is going to play out in terms of what kind of plants grow in particular regions and how crops perform in different places. So this is a great experiment that we're doing in the world, maybe not what we intended to do, and certainly not one we have great controls for, but it is a large experiment. But it doesn't mean that there won't be some positives there. There will be some opportunities for particular crops in different places, and indeed for natural environments to regenerate, as well as others will probably decline.
Your last couple of slides showed a photo of your research group and, of course, your family, who both in the past and currently have a great interest and input into your science. I did want to ask you, though, about how have international collaborations played an important part in your research?
International collaborations have really been, I mean, they're vital, because not only do you bring different ideas which have come from different backgrounds, but you also bring the cultures of people. And many of those things influence the way people run their labs, the way they think about science, the way they think about the future. So for me, being a scientist has been about becoming a global citizen, understanding the circumstances of science in different countries, and seeking to find commonality of solutions and investments that make sense for that. So certainly the ARC funding I've received over the years has been really instrumental in driving those decisions to build partnerships, and then having the joy to actually go and visit people and do those. Obviously, over the last little while, especially from Perth, those opportunities to interact with the world in reality have been quite limited. But we have kept up many of our friendships and relationships online, and we hope that soon we'll be able to go back and visit people and have them visit us in Australia.
Well, Harvey, it was a wonderful presentation. Really a joy spending time with you now. Thank you and congratulations again.
Thank you.
Now we're going to take a short break and be back at 12:30 Australian Eastern Daylight Time for session 3, which will be hosted by Academy Chief Executive Anna Maria Arabia. During the break, we have a special presentation to share with you, an interview with another of our fellows, Professor Eddie Holmes, who last night was awarded the Prime Minister's Prize for Science. I'm Paul Richards. Good afternoon.
He's been at the forefront of the battle to contain COVID-19.
My kind of 30-year journey of working on virus evolution, how they jump species, it's kind of got me to this point looking at SARS-CoV-2.
Australian virologist Professor Eddie Holmes was one of the first people to publicly release the genome sequence of the virus.
I felt as a Western scientist under great obligation to get this data out there.
And he thinks the world needs to be ready for future outbreaks.
It's an obvious reflection of the way humans live. So it's going to happen more. And I think climate change and pandemics go hand in hand.
Professor Eddie Holmes, the Fellow of the Australian Academy of Science, is this year's winner of the Prime Minister's Prize for Science, and now sits down to discuss the year that was and what's still ahead with the Academy's Chief Executive, Anna Maria Arabia.
Eddie, congratulations on being awarded the Prime Minister's Science Prize. You must be thrilled about this honour.
Thank you, Anna Maria. Yeah, look, it's an amazing honour. Very unexpected, but it's, in fact, I'm almost still haven't quite come to terms with it, but it's a great privilege.
Eddie, look, you've been studying the evolution of viruses for more than 30 years. Can you briefly explain your work and what led to this award?
Yeah, so by training, I'm an evolutionary biologist, and over the years I've worked on a number of different things. Before I got into viruses, I started off working on primates. Then I worked on fruit flies, and I thought I'd get even smaller. So I worked on viruses for maybe sort of 30 years now. And what I'm most interested in is how viruses are able to jump from one species to another, which they do all the time. If you're a virus, your life's goal is really to get from one host to another, sometimes in different species. So much of my work has been trying to understand exactly how viruses do that.
And it couldn't be more relevant than it is today as we're fighting this pandemic. Let's go back to where this started. Why did you decide to study virology, and how did you become interested in infectious diseases?
Actually, my first degree is in anthropology, so I was actually really interested in human evolution, actually, so where humans have come from and our kind of evolutionary ancestry. And I'm still very interested in that story. The problem was human evolution is actually really slow, and you can look at fossils, but you can't really watch it in real time. You can't really watch it kind of in front of you. But viruses evolve extremely quickly. It's like 1 million times faster than human genes do. So it's like evolution on fast forward. So if you're an evolutionary biologist, that kind of allows you to kind of study them in more detail more quickly. So it really is like watching it in real time. So that's the kind of reason intellectually why I work on virus evolution, and why particularly I got into it was when I was working on fruit flies. That was in Northern California, quite close to San Francisco, and I spent all my days in the lab studying fruit flies and how they were evolving quite slowly. And then just down the road, like I say, was San Francisco. This was 1990. And that was almost like the peak of AIDS deaths in San Francisco at that time. So it was a terrible pandemic at that point. And there was no antivirals. People were dying in very large numbers. And then I saw a talk by a colleague from the UK talking about how the AIDS virus, HIV, was evolving. And I thought, actually, why don't I use the techniques that I'm doing on fruit flies? Why don't I use the same kind of ideas on HIV? Because it's evolving rapidly, so I can watch it in real time. Plus, there's a kind of practical outcome of this. I can, help understand how HIV evolves and maybe kind of help control it. So that's kind of, I pivoted my research, kind of changed in 1990 from flies to viruses.
Eddie, you were born in the UK and you've mentioned San Francisco, and now you're in Australia. What brought you to our shores?
I've been in the US. I've lived in the US I've moved a lot in my career. I was in the US for sort of 10 years prior to Australia. I wanted a new challenge. I thought, and I still do, Australia I think is a great place to do science. I wanted to be in the Asia Pacific. I thought that's kind of where the world was sort of shifting in focus, this region. I had wanted to work more in a kind of the Asian region, and moving to Australia kind of allowed that all to kind of happen in one place. So I moved here in 2012, I'm an Australian citizen now, and it's been by far the best move I've ever made, and I'm very happy to be here.
It's fantastic. It's so good to hear that. Eddie Holmes, you were one of the first people to publicly release the genome sequence of SARS-CoV-2, the virus which causes COVID-19. Can you explain why you did that?
Yeah, so what happened was I first heard about what is now COVID-19 on, I think it's December the 31st, 2019. And there's a wonderful website called ProMed that kind of posts daily updates of diseases. And I read this post of 4 cases of pneumonia in Wuhan associated with a market. And I've been to Wuhan, I've actually been to this market. So I thought that's very, what I've done, where I've been. So I contacted my colleagues in China on New Year's Day 2020 to say, Happy New Year, are you working on this? Can I help? And then my good friend and colleague, Professor Zhongzheng Zhang from Fudan, he said he was working on the virus because we actually had a project ongoing in Wuhan anyway on respiratory diseases. So he managed to get hold of some early samples, and they were sent to his lab on January the 3rd, 2020, and then 40 hours later he'd sequenced the complete genome. So 40 hours from sample arriving to find the pathogen. Go back to HIV, it took 2 years to show that HIV was the cause of AIDS. Here it took 40 hours. So we had the genome sequence on January the 5th. We talked on that day, and we decided that we had to get the data to the authorities as quickly as possible. So he wrote to the Ministry of Health in China on that day and said, look, it's a new virus. It's a coronavirus. It looks a bit like the first SARS virus. People should take precautions because this could be a respiratory pathogen. We kind of could see that from the genome sequence. So that's January the 5th. And on that day, at that point, there were very few cases in Wuhan, maybe small numbers of 10 or 20 or so. And then as the week wore on, although we told the local authorities, the case count was slowly increasing. And the Ministry of Health in China were in control of information, so we couldn't do anything without their approval. But as that week wore on, that second week of January, it became clear to me that the world wanted to know what the genome sequence was. The case numbers were rising. It was becoming more and more important to share this information. I never had the sequence myself. I'd helped analyze the data without actually having the genetic code of the virus myself. And the pressure rose, and I felt as a Western scientist under great obligation to get this data out there. And then I think it was like the Friday night, there was a tweet from Sir Jeremy Farrow, who's the director of the Wellcome Trust, and he basically said, if this virus is out, if the sequence is known, it should be released. And I thought, I need to do this right now. This has got to breaking point. So I contacted Zhang, Professor Zhang in China, and I kind of convinced him we had to release it. And so he was actually on a plane going from Shanghai to Beijing. So I called him on a plane, and the flight attendant was saying, "Put your phone down, sir." But he said, "Okay, we release." And so he contacted his lab manager. They sent me the genome sequence the first time I had it, and then I spent 52 minutes just running a kind of a little description of what the sequence was, running a tweet, and I put it out there as fast as I could on January the 10th Australian time. And that then, I think, kind of marked the scientific fight back against the virus. Having that sequence allowed tests for the virus to be developed and vaccines to be designed very, very quickly.
What an extraordinary story, and what a modern scientific story, Eddie. I mean, you know, it had all of the elements, didn't it? A genome sequence, it was critical, the importance of international collaboration, the importance of sharing information, and it was shared by Twitter.
And if I can say, Anne-Marie, I think that the one thing I've learned from all of this is that you know, the whole pandemic, sharing your data as rapidly as possible is absolutely critical. At the time we shared that, it wasn't clear this would be a pandemic. There weren't that many cases. It was probably not even on the news in most countries, but it was already beginning, you know, I sensed this had to be done. Now, even more so, the importance of that act and data sharing in general has become much more, you know, obvious to me and everyone else. And I think for going forward to how to prevent the next pandemic, it's absolutely critical. We build structures that allow scientists to share data in as freely, as openly and rapidly as possible. That's the biggest lesson I think I've learned from the whole pandemic.
In that too, Eddie, there's an enormous act of scientific integrity, and I personally thank you, and I know the world thanks you too, because without that commitment to transparency and scientific integrity, we may not be where we are today. Did you have any idea back then in January 2020 that this virus would spread and become the widespread pandemic that we now find ourselves in almost 2 years later?
When it first hit, so when I first heard about it first week of January, I thought, as case numbers started to go up, I thought, well, this would be like SARS-1. So SARS-1 emerged in 2002, 2003 in southern China, in Guangdong province. To me, it felt like that because the virus was very closely related. So I thought maybe this will spread to, you know, a few cities, a localized outbreak, you know, maybe a few thousand cases. And that first week in China, no one had died either. As time wore on, wore on pretty quickly, the case numbers started to increase and I started to get more worried. And by about the third week of January, the virus had got to other countries. And at that point, I think I flipped from this is going to be more than a localized outbreak. This virus is very infectious. It's going to be a big thing. And that, I think, was the big difference in the SARS-1 to SARS-2 and why my mindset changed, because by about the third week, it became pretty clear this virus was far more infectious than the first SARS virus. And in particular, there was growing evidence that people could transmit the virus asymptomatically. So they weren't showing any visible symptoms. In SARS-1, people who transmitted were ill. And that's why it was actually easy to control, because you could just kind of quarantine those people. SARS-2, COVID-19, your worst case scenario is the silent spread, the person you're sitting next to on the train or the bus you don't know is infected. That's what SARS-2 was. And as soon as that became clear and the virus started to leak out into other countries, I thought, okay, this is going to be a big thing. And by early February, the pandemic in my mind was clearly going to be a very major thing indeed.
What does your research indicate in terms of the trajectory of COVID-19? Did we predict the Delta variant, and what does research tell us might yet come?
Delta was a surprise to me. I thought, you know, if you asked me a year ago what would happen, I'd have said to you the virus will probably stay much the same as it is now. It probably won't get much more infectious, probably won't get much more virulent, and gradually we'll get on top of it by vaccination. Now that's not the case. The virus is now more virulent and a lot more infectious than it was when it first emerged in Wuhan. I think that certainly took me by surprise. I think looking at the trajectory of the evolution, this is what I kind of diagnose as what's happened. During 2020, I should just, before I answer that question, I should say one big thing about COVID is we are looking at this virus in the finest scale anatomy we've ever done before. Even viruses like influenza and HIV, our scale analysis has not been anywhere near as precise as it's been with SARS-CoV-2. There are now over 4 million genome sequences of viruses available for analysis. It's quite extraordinary. So it's a very, very detailed analysis. Given that, I think what happened was in 2020 and the start of 2021, you were seeing the virus evolve in populations where basically there was no immunity. Okay, everyone was susceptible. The virus could spread in an unfettered way. It's kind of like salad days of the virus. And selection, evolution at that period was simply for infectiousness, to make the best replicator, the fastest race car you could make. And Delta, without a doubt, won that race by a distance, right? By a clear distance. Much more than I ever thought. So Delta was the winner in that race in a population that's generally naive, haven't seen the virus before, for sheer infectiousness. Now, in second half of 2021, you're seeing is a different kind of selective landscape, evolutionary landscape. Now, many people in many countries have got pretty high vaccination rates, some obviously not, sadly, but many are pretty good. So the selection pressure, the evolutionary pressure on the virus is now going to be very, very different. It's not going to be simply the fastest race car that wins. It's going to be the one that's best able to get around that immunity, okay? So selection will favour strains that can reinfect people who already have been immunised against the virus. So that's what selection pressure is going to change. So then I expect we'll get different variants start to come up in the near future that can kind of get around immunity and reinfect people. Now, whether they'll be more virulent or more generally infectious is hard to say. Normally in evolution, there are trade-offs. So if you can evade immunity, if you're good at that, you normally can't do something else. So it's quite hard to say exactly what the future virus will, the Delta of the future will look like. But it will be different than we've got now because, again, the evolutionary pressure is changed. We're now in a world, now it's going to evolve in a world where people are immune rather than last year when people are not immune.
So is it correct to say there are at least two pathways to that evolution? One is to transmit in populations that have lower vaccination, and the other is to transmit and adapt to a vaccinated population, and that could lead to a multitude of variants across the world.
I think that's very well put, exactly correct. So in countries with low rates of immunisation, and some are actually on Australia's borders, obviously, evolution, the pressure on the virus will be very different than it is here with very highly vaccinated pressure. And so you may see different variants being favoured in different populations in exactly the same sorts of way as you're suggesting. The way I like to think about viruses, though, is that although they can do lots of things and they can adapt very well, there are constraints. They can't do everything, okay? So normally things that are very infectious are not very virulent, or the other way around. Normally things that are very virulent are often not very infectious, not that it's infectious. So there are kind of these complicated trade-offs. So if you try and favour one thing, you have to kind of reduce something else. So it's a bit like having a Swiss Army knife, right? So viruses are kind of compact and can do lots of things, but you can't do much more because you've got to fit into this one little knife. So for example, in a Swiss Army knife, you have a pair of scissors, right? That's kind of like a bit small, it can cut a bit of paper, but it can't cut anything big. To make a bigger pair of scissors in an army knife, you'd have to kind of like make it bigger, stronger. But to do that, you have to remove some of the other blades. So you can, if you want to favour one thing, you have to remove something else. So the virus is going to be kind of in this kind of complicated evolutionary trade-off. Its elbow room is limited to some extent. So quite how that will play out, though, is a complicated kind of set of nuances and trajectories, and it's quite hard to predict exactly what it will be like.
It's an excellent analogy, thank you. I'll never look at a Swiss Army knife the same again, I think. Eddie, there's been a lot of debate about where the virus originated. What does the scientific evidence show?
I think the scientific evidence as of today tells us this, that the most closely related viruses we have to SARS-CoV-2 are in bats, particularly these horseshoe bats. They're very common in Southeast Asia and southern China. And the closest we've found so far are actually from horseshoe bats in Laos, so northern Southeast Asia. And they're about 96.8% similar. So you've got SARS-CoV-2 and the closest bat virus, it's about 96.8% similar. Now that sounds very close, but in fact, in evolutionary terms, there's still a gap. So it's 3.2%. That's actually still, a decade or more, maybe 20 or 30 years even, of evolution. So we're missing the missing link. We're missing the direct ancestor. We know that bats are closest, okay, from Lao. Then there's a gap, and there's SARS-CoV-2. So the key thing is what's in that gap, where's in that gap. Clearly, bats do carry lots of these viruses, and I strongly expect if we sample more of these horseshoe bats across southern China, even in central China, Southeast Asia, we'll find more viruses that are close, probably ones even closer than they are to SARS-CoV-2. My guess is also that there are other animals out there that will have very, very similar viruses. We know that pangolins do, but I can't believe it's just bats and pangolins, and I strongly suspect the ecology of these viruses in nature there's other species involved there we haven't yet sampled. And so one of the key things is to try and sample more of the natural ecology of these viruses to see what viruses are out there. Going back to these bats from Lao, not only are they very genetically close, the critical thing is they have a bit of sequence called the receptor binding domain. You can imagine the virus is like a lock and a key. Virus in the cells like a lock and a key. So the cells that lock, the virus is the key, and the virus has to kind of get itself into the lock, turn it and get in. That key, that binding domain in these Lao viruses is remarkably similar to the human key. It's effectively the same thing. So that is the functional core of the virus. It allows the virus to do what it does to infect human cells. That core we know already is there in nature in these bats in Southeast Asia. So that tells you there are other things. There are two things. One is that if we look more, we'll find more of these viruses that relate. And two, that viruses like SARS-CoV-2 can emerge again because they're armed and equipped there in nature. So I think it's just a matter of sampling more and more animal hosts, bats and other things, to find the closest relative to SARS-CoV-2. That could take time, though, so it's not going to be a simple thing.
Eddie, you've discussed your idea of developing a global radar system. How would this work, and who would develop such a system?
So one of the other lessons I think the obvious lesson we learned from COVID-19 is that we have to be able to detect these pathogens as quickly as possible, as soon as they are emerging in humans. Okay, that's a critical thing to do. And we now have the techniques that can do that. We can monitor people who, and I think the key people to monitor are people who live at the animal interface. So who work in the wildlife trade or these animal markets, or who live near wildlife. They are at most risk. So what we need is a global organization or global mechanism that allows that kind of radar system to be put in place globally, and the data shared globally, so we can monitor for any unusual disease occurrences in human populations, again, particularly people who live at the animal interface.
How frequent do you think pandemics will occur, and what should we be doing to reduce our vulnerability to them?
Pandemics have increased in frequency in the last few hundred years, and it's an obvious reflection of the way humans live. We have bigger populations, we're more connected, we're more urban, we're denser, we interact more with wildlife species, we have deforestation. So it's going to happen more. And I think climate change is really going to be a key driver of this, because as climates change, what will happen is that animals will change their distribution too, because they're affected by climate change, and they're probably grouped together more, allowing viruses to jump more between them. Humans who rely on animal species will need to change their way of living, their livelihoods, their way of acquiring food, and they'll probably be exposed to new animal species and new pathogens too. So I think climate change and pandemics go hand in hand. Climate change will get more pandemics. So it's hard to put a precise number on the frequency that they're going to happen, but I think it's a racing certainty that we will get more and more of these. If you look at people who live near wildlife, who work in animal markets or live near bat caves, they are very commonly exposed to viruses. It happens all the time. Even people in North America are very commonly exposed, for example, to swine flu viruses. Okay, so they're continually, people who handle pigs, work in pig farms, go to pig fairs, their pig viruses come to humans. So this exposure is happening all the time. Okay, normally, luckily, they don't go anywhere. So there's one case and it burns itself out. Every so often, though, and increasingly often now in this modern world, you get a chance for the virus to get going, cause an epidemic or even a pandemic. So we need to kind of get to grips with the prospect this is going to happen again in the future. And the key lessons, I think, we need to prevent this happening again or help mitigate as much as possible. I think there are a few of them. One, we need new pandemic plans. So COVID, our response globally was based on flu. Flu was our model. That was a pretty good model, but COVID wasn't flu. There were some important differences. We need plans that can cope with a broader range of viruses. We need that global radar, as I said before. We also need basic research into vaccines and antivirals that are ready to go in the freezer, ready. So rather than waiting, waiting for a year, 18 months to get those vaccines ready, they need to be there on site. Go to freezer. Okay, that's the one I want. Now that's a massive ask. That really is like the Apollo project of virology and immunology. You need to have viruses that can recognize a broad range of, so vaccines that recognize a broad range of viruses ready to go. Okay, so we need vaccines to protect against a swathe of influenza viruses or a swathe of coronaviruses. That's a big scientific challenge. And as yet, we've not really made any universal vaccines for any system, but that has to be a key goal.
Eddie Holmes, looking forward to your own research, what are your scientific goals for the next few years?
I think I would actually like to play a key role in trying to do this, trying to put in place these structures that will try and prevent the next pandemic. And I think Australia is in a really good place to do this. I think we have a great scientific infrastructure, a very high quality scientists there. We have a lot of good biosecurity already in place, but I think it could be better. And I think what I really like to do is try and enhance our national biosecurity so that if viruses come in, and they will come in, okay, it's on humans and in other animals coming into this country and in plants too, we are able to respond to those in a much more, even more efficient way than we did before. In Australia and in most countries, there's actually a division between animals and humans. They're kind of dealt with different. The veterinary community deals with animal pathogens, and the medical community deals with human ones. But in fact, they're the same thing. There's this idea called One Health, which posits that humans and animals are part of the same ecosystem. We share the same pathogens. That's what COVID's taught us as well. And therefore, we need a unifying framework to understand disease in animals and humans. You can't separate them. And I think what I'd like to do for Australia going forward is help establish a new kind of national biosecurity that uses the kind of techniques that we've used that quickly identified SARS-CoV-2 to deploy that to look at other potential emerging threats to Australia and therefore help safeguard us against pandemics.
I can't think of a better person for the job. You've been awarded the Prime Minister's Prize for Science, and in October 2020 you were named the New South Wales Scientist of the Year. And at that time, the Sydney Morning Herald wrote in their editorial that you richly deserve that recognition because your story illustrates the bravery demanded of scientists in the current political environment. You have certainly been brave and worked tirelessly. I know that personally, having called you for scientific advice on a Sunday. But do you find it concerning that scientists need to be brave to undertake research for the public good? Is this just part and parcel of being a modern day scientist?
Science is now at the forefront of people's minds, and whether they think consciously they're talking science, people's understanding of science has increased dramatically. So you actually now ask, the average person, they'll know about genome sequencing. We know what the case numbers, we know what the R value is, okay? So our scientific literacy has massively increased as a nation, globally in fact, because of the pandemic. So that's great. The problem also is that scientists are much more in the public eye. I've always thought of myself as being a kind of backroom staff, right? I find the public eye slightly disconcerting, but it's thrust us into the limelight, which is something I was not expecting. And it did take some adjustment to do that. I think so now going forward, I think scientists need to understand that everything they do and they say, people are listening to, and there are consequences for that. So we need to be aware of that. Also in the modern age, so when I released that sequence, I used the internet. I posted it on a website and I announced it on Twitter. So I used social media, and that was an amazingly rapid thing to help respond to the pandemic. And social media is an amazing thing. It can allow you to And going forward for response to pandemics, we need to have tools like social media because they are so quick. And that's what the pandemics need a quick response. Unfortunately, there's also a downside, which is some people are very upset by things you do. So we have to kind of live with this duality where having the public access by social media is an amazing thing. It's a very, very powerful tool, but yet there's a necessary kind of downside. So quite how we steer our way through that, I don't really know, but it's going to be there for the future, and we need to kind of get used to it, I guess.
What about life outside of science? You're a dad, you've been in lockdown, you've had to assist with home learning. What have you done to keep balance, to unwind, and particularly at a time when your research is going at such extraordinary intensity?
Yeah, I think so. Like most parents in Australia during lockdown, homeschooling, online learning has been a challenge. I've learned a lot of French and Japanese feudal history, so that's been quite interesting. But it has been a challenge. So we all need to unwind. So I'm quite an avid guitarist, electric guitar. So I have a few of those, and I set myself a challenge before the pandemic started. Before lockdown started, I should say, of trying to learn all the guitar parts from Led Zeppelin 2. And I've done okay. Heartbreakers kind of still give me a bit of trouble. So I've just tried to kind of focus by my inner Jimmy Page coming out and failing. I haven't got the hair for it, but I've had a go anyway.
That's fantastic. I'm not sure if I've ever shared with you that I have this wish to bring together all the musically inclined and talented fellows of the Australian Academy of Science to jam together, and that would take collaboration to another level, I think. It's been fascinating speaking with you. I know you've still got a lot to contribute to science and to society, and to be honest, you're a hero to many scientists and to me personally. So please keep up your extraordinary work. We need independent science to inform our policies and future directions. Eddie Holmes, it's been my pleasure to talk with you today. Congratulations again on being awarded the Prime Minister's Science Prize, and we are so proud to call you a Fellow of the Australian Academy of Science.
Thank you. It's been a great pleasure talking to you.
Welcome back to Science at the Shine Dome 2021. I'm Anna Maria Arabia, Chief Executive of the Australian Academy of Science. Congratulations to Professor Eddie Holmes, a very deserving recipient of the Prime Minister's Prize for Science. If you missed that interview, it's available on the Academy website at science.org.au. Thank you too to Paul Richards for taking us through this morning's presentations from our fellows, and I'm delighted to be with you for the remainder of the afternoon. Thank you all for submitting your questions for our fellows. Don't forget you can ask questions throughout the event by using the Q&A panel on your screen. Now, on to our next presentation. Please welcome back Professor Helene Marsh to introduce our next fellow.
Our next presentation is from Professor Linda Blackall. Linda is a microbiologist who pioneered and championed procedures that reveal microbes and their function in natural and engineered systems. She discovered microbial contributions to practical aspects of water, wastewater, and solid waste treatment, leading to improvements in treatment and energy savings. We'll now hear from Linda.
My overriding area of interest is the microbiome, and due to its attention in recent times, I will call my short presentation today the microbiome zeitgeist. The zeitgeist can be seen from publications on the Web of Science since 2002, when the first publication using this term occurred. You can easily see the steady, consistent increase in publications to greater than 10,000 in 2020, and at present a somewhat lower number in 2021, which might be impacted by our current pandemic and reduction in ability of scientists to be in the lab at the same intensity as they previously were. In any case, this is a very hot topic of research and one that I'll focus on in my presentation today. To put the microbial community into a size perspective, you can see how they are in the hundreds of nanometers to micrometer size range, particularly relative to the larger cells of animals and plants, and also in relation to smaller entities like lipids, proteins, and atoms. To observe microbes, which live in these microbiomes. Typically, light and electron microscopy is used. So to see topics of my own work, I downloaded this from the Web of Science. Clearly, I'm a microbiologist, and the majority of my research findings fall into the broad microbiology category. Though I note the long tail of involvement that I have in many, demonstrating the involvement that I have in many areas. However, you can also see that I work very much in the applied aspects of microbiology, but also I collaborate closely with those in environmental science and engineering, as well as water resources and ecology. I've chosen to focus on just two areas of my work, and one that has occupied a vast majority of my research and development career has been around wastewater and its treatment. Though not covered today, I have over the last couple of years largely focused a lot of my work on exploring sewer contents to monitor microbes, genes, and chemicals. The treatment of wastewater involves numerous tanks where typically aerobic metabolism of microbes is promoted and the selected for microbial community grows on the wastewater components. Following this process, in the clarifier, the sedimentation or settlement of the grown biomass has to occur rapidly so that the treated water can be discharged to the environment. Therefore, the settlement of wastewater treatment plant biomass is a critical aspect of the overall process. In the aeration tank, microbes grow as flocks, which are loose assemblages of microbes in the order 500 micrometers in diameter, and they are three-dimensional in appearance. The microbes are held together by various forces, including the production of microbially produced extracellular polymers. This fluorescence in situ hybridization probed image shows that we have been able to visualize various microbial types, microbial species, using organism-specific gene probes. This third image shows an elaboration of the microbial community that has been generated by modifying certain features of the treatment process. The structure is called a granule, and it has microbes that are much more closely adjoined to each other relative to their association in the floccular structure. And the granular structure has valuable features that facilitate rapid settlement of the biomass. So I'm showing you this by way of a video in this particular slide. And we have two cylinders. On the left, the cylinder containing granular biomass, and on the right, floccular biomass. The material has been allowed to sit under static conditions, and therefore the biomass is settling simply by gravity. These images have been acquired over a period of time. So we see the granular structure, the granular biomass settles much more quickly, and the final material to which it settles, the volume to which it settles, is much smaller relative to the floccular biomass. And this ability to settle more quickly and to a more dense final material is highly important in rapid sedimentation within the wastewater treatment plant. And this has been a really important feature contributing to better wastewater treatment plant operation. The second component of wastewater treatment plant operation that I want to discuss that has also occupied a large part of my work has been in studying, understanding, and improving nutrient removal from domestic wastewater. The nutrients that I've explored include nitrogen and phosphorus, and a long collaborative process led to the discovery of the first phosphorus-removing bacterium in wastewater treatment processes. We called the bacterium Accumulibacter phosphatus, and it's shown in this fluorescence in situ hybridization image as yellow-coloured cells in comparison with green other prokaryotic organisms. We correlated phosphorus removal performance of sewage treatment plants with the abundance of Accumulibacter. We also were able to show using staining procedures that these Accumulibacter uniquely have polyphosphate inside of them, as shown by these purple spots inside the Accumulibacter. We subsequently went on and used metagenomics to understand their biochemical mechanisms and the ability that they have for selectively accumulating polyphosphate. Again, this was a major discovery and a landmark one for improvement in wastewater treatment plant operations. Changing tack now, I'm going to move on to another field of research that has occupied a lot of my time, at least since 2000s, early 2000s, and this revolves around marine microbial symbioses. To introduce the animals, I have images of stony or reef-building corals shown on the left there. In my research, I also use these other animals known as anemones from the genus Exaptasia. Both corals and anemones are comprised of materials, of tentacles rather, which they use in their heterotrophic metabolism. They also contain single-celled algal cells within their tissues, which are photosynthetic. And indeed, the vast majority of the organic carbon that is used by these animals is provided by these photosynthetic single-celled algae. Indeed, this makes these animals solar-powered. The prokaryotic communities that live with corals are very, very complex, and shown in this particular slide on the left side, the prokaryotes are associated with the mucous surface layer. They are also found inside of the coral cells, in particular the ectodermis and the gastrodermis. We can see also the single-celled algae belonging to the family Symbiodiniaceae are in particular associated with the gastrodermal cells. These Symbiodiniaceae cells also have their own microbial community, both around them in the phycosphere and internally. They also have prokaryotes, in particular bacteria, associated with them. Adjacent to and connected with, though, to the tissue-associated communities, microbial communities, are those that are found within the skeleton of these stony corals. Shown here is the massive coral Paragoniastria australensis, which has been sectioned through that white dotted line, and then the exposed surface shown in the image to the left. You can see the layers of different organisms within, or the coloured layers within the skeleton, which are comprised of photosynthetic algae, photosynthetic prokaryotes like cyanobacteria, and many other heterotrophic prokaryotes living in that skeleton ecosystem. So why are we studying these? Why are we studying the microbial communities associated with corals? We're interested in who they are, but we're very, very interested specifically in what are they doing for the animal? What is the role of the microbiome to the animal? An overarching feature that we study is the role that prokaryotes and other microbes could play in mitigating coral bleaching, which is a major a major feature that is occurring in corals around the world today. And I will show you that in this final slide here. Why do we call it coral bleaching? We have living coral in the upper panel, that is coral that's alive, the tissue is living. In the lower panel, the coral has died and the animals have sloughed from the calcium carbonate, which is the skeleton of the coral, exposing this white appearance, giving rise to this name, coral bleaching. And we believe that the prokaryotic community could play an important role in mitigating this major challenge that's occurring currently. Thank you.
Thanks ever so much, Linda. We now have live with us Professor Linda Blackall. Welcome and congratulations on your election to the Australian Academy of Science.
Thank you very much, Anna Maria. It's a huge honour to be elected and I'm extremely happy.
Fantastic. We have a question from our audience from Mark Westerboy, who asks, and Linda, you're going to have to help me out here. Could you comment on adaptive merit of accumulating surplus P? Question mark. So I hope that P means something to you. Do cells subsequently get to do a lot of multiplication without needing more P uptake? If so, how do they come to suddenly find themselves in a low P environment?
Yeah, good question. So these bacteria, the Accumulibacter phosphatus that accumulates polyphosphate inside of itself, does so because of a metabolic phenomenon. They're not using the P in their growth per se, although they do use part of it, but the majority is actually accumulated and indeed released. And that's an important aspect of maintaining various features in the metabolism of the organisms. So they're not really using all of the phosphorus in their metabolism per se. I think it would take quite a long answer to address fully that question. It's a complicated one, but the P stands for phosphorus, Anna Maria. So
thank you. I'm sure our fellows and others watching it's just me.
Sorry, Anna Maria, I do a talk where I say who's taking the P out of wastewater, and in that case, it's a double entendre.
Right. As long as that P is coming out phosphorus and the other variety, we're
right.
Fine.
Yeah.
Speaking of wastewater, phosphorus and nitrogen removal is something you've worked on. Can you talk us through the implications of that in terms of the treatment of wastewater? Is that something that has been applied to date?
Yes, so definitely, I mean, in relation to domestic wastewater, which is the wastewater that humans produce, a large portion of that is comprised of, well, carbon, which is able to be removed as CO2 eventually, but nitrogen and phosphorus both absolutely need to be removed from the wastewater because both of those would produce eutrophying conditions on the receiving waters. And so the removal is absolutely critical. And I was very impressed when I first started working in this field with the engineers who had read microbiology textbooks and designed the process around selection for, in particular, the nitrogen removing bacteria, that is both nitrifiers and denitrifiers, which require very opposite types of conditions. So I was impressed that they'd read the microbiology textbooks, provided the right conditions, and the nitrogen removing prokaryotic microorganisms, the bacteria, were selected for very successfully. However, understanding the microorganisms themselves led to much better and more reproducible removal of those nutrients. Phosphorus was more challenging. And so eventually we discovered who those organisms were and what was the motivation for their capacity to remove phosphorus. But they absolutely critically need to be removed to prevent eutrophication in the ecosystems.
Thank you. The other area you focused on in your presentation was marine microbial symbiosis. If you're able to mitigate bleaching, could this save coral despite the warming of the oceans?
Yeah, so that's a really good question, Anna Maria. And of course, it's a time issue, and we are not there yet. That is, we are working towards these strategies. Our results are quite promising in that we do see mitigation occurring. There's no question. And of course, we also were required to know why was bleaching occurring at the molecular level, and then develop procedures that exploited features of the bacteria that mitigated these conditions leading to bleaching. And so if it does come to fruition, we are working on probiotics for corals. All species can indeed use probiotics, and so too can corals. And so we would use them in this particular capacity to specific bacteria that have specific properties that can mitigate the coral bleaching features. If we can get our results to a better stage of development than they currently are, we definitely believe that there's a role for this approach in controlling bleaching, along with some other strategies that are used in the field as well.
All right. A question from Francis Saparovich. Are we seeing an increase in the proportion of corals that do require algae to survive in our reefs?
So the corals that require algae they have the algae as photosymbionts inside of their cells, so they're symbiotic associations, and we're not seeing more of them. Indeed, of course, we're seeing less of them simply because they're dying due to bleaching, but we're not seeing more of them without a doubt. But of course, because these coral species require the photosymbionts to be associated with them, they really exist where there's sufficient light for that to occur. And it's a relationship between the biochemistry of both the coral host and the symbiont, the photosymbiont, that leads to the production of the calcium carbonate. We heard from one of the fellows yesterday speaking about calcium carbonate and the issues around calcium carbonate. And one of the major impacts of climate change is at least ocean warming, which is leading to bleaching, and eventually, as was described by the fellow yesterday, acidification of the ocean waters, which changes the carbonate chemistry, the aragonite chemistry of the skeleton that is produced by the corals. So we're not getting more hard corals, but we want to maintain the coral species biodiversity that we currently have.
Thank you, Linda. Our time's run out rather quickly. We're going to have to end it there. Congratulations again, and we look forward to seeing you soon.
Thank you very much. Thank you. Bye-bye.
To introduce our next fellow, please welcome Academy Council member, Professor Jonathan Sprent.
Next, we will hear from Professor Ian Paulsen. Ian is a world leader in microbiology and a pioneer in microbial genomics. He discovered several novel classes of multi-drug efflux pumps that can make bacteria resistant to multiple drugs, and he showed that these pumps have natural roles in the cell and have been opportunistically co-opted by hospital pathogens as a resistance mechanism against antibiotics. Now we will hear from Ian.
Hi, I'm Professor Ian Paulsen from Macquarie University. I'm also Director of the Australian Research Council Centre of Excellence in Synthetic Biology, whose website I've got a screenshot here of, and I'll steal its tagline, inspired by nature, designed by science, is my title for today's talk. So my scientific journey started back in around about 1990. In fact, here's a photo of me as a PhD student in the early 90s with way too much hair and wearing a Violent Femmes concert t-shirt. And as a PhD student at Monash University and the University of Sydney, I was working on studying multi-drug resistant Staphylococcus aureus, we call them staph. And that's a very serious hospital pathogen that's, and here's a little toy version of it, which you can see is wearing a cape. And the reason it's wearing a cape is because it's a superbug. So it's a hospital pathogen that's resistant to most of the antibiotics that we have for treating it. And back as a PhD student, I was trying to work out how to become resistant to so many drugs. And so I was cloning and characterizing its drug resistance genes. And I found many of them were actually what we now know to be bacterial multi-drug efflux pumps. So these are proteins that sit in the membrane of the cell and then pump the drug out of the cell to protect the microbe from the antibiotic. And in characterizing these, I became one of the sort of co-discoverers of bacterial multi-drug efflux pumps. After I finished my PhD, I then went off to the US, where I intended to go for a couple of years and stayed 12 years, first at the University of California in San Diego, where I worked for a brilliant but highly eccentric professor, seen here, scavenging food out of garbage dumpsters behind shopping centres. After that, I then worked at TIGER, the Institute for Genomic Research. And here's me and my small group at TIGER. And the story of my career in some ways is also the story of how technology for reading and writing DNA has improved. So as a PhD student, it took me years to sequence one gene at a time, albeit it might have gone faster if I'd spent less time at the pub, but nevertheless, it was a slow process. By the time I joined TIGER, the technology had advanced, so instead of sequencing one gene at a time, we could sequence the thousands of genes that make up a bacterial genome all at once. And we could do it in a much faster time than it took me as a PhD student to sequence one gene. And so as one example here, I led the sequencing of the genome of the hospital pathogen Enterococcus faecalis. Now, we all have this bug living happily and harmlessly in our guts, in our digestive tracts, but it's also one of the more dangerous hospital pathogens, and like golden staph, is resistant to most of the antibiotics that we can treat it with. And so in sequencing its genome, it actually revealed how this had become a dangerous hospital pathogen. It had done so by acquiring genes from other organisms. So in fact, somewhere between 1/4 to 1/3 of its genome actually came from entirely different organisms, and that it incorporated that DNA into its own genome. And those bits of mobile DNA encoded the drug resistance genes and the virulence factors that made this a dangerous pathogen. I still retain my love for my interest in multi-drug efflux pumps and transporters in general, how cells move things in and out of their cells. And by having the complete genome, for the first time, we essentially had the blueprints of how these bacteria worked. However, I should say they're blueprints that when we look at them, we only understand maybe a half, maybe two-thirds of them. There are many genes of unknown function that we still don't know what they're doing. But nevertheless, it gave us enough information to start to build up how these bacteria really work. How do they, what are they transporting into the cells, and what are they metabolizing for in terms of nutrients and for energy? And this shows a simple model here that we've built of the sheep foot rot pathogen, Dichelobacter nodosus. And so we now started to have be able to put together a view of how life actually works. Then in 2007, Macquarie University recruited me to come back to Australia. And by this stage, our ability to read DNA had improved even more. It got faster and even cheaper. And so instead of sequencing one genome at a time, we could now go to any sample on the planet that we can get and sequence the thousands of organisms that live in that community all at the same time and get their near complete genomes. And this shows one of the weirder bacterial communities I've worked on. So this is a nullable plain, enormous piece of limestone. Underneath the nullable plain, there are limestone caves, and if you're in crazy cave dive, you can go exploring these. And for some of these caves, when they explored them, they found these weird, almost tentacular microbial cave slimes. And these are living in the dark, underground below a desert where there's essentially no input of nutrients or water from the surface. And so we went ahead and sequenced the genomes of this community to try and work out, well, how these microbes are making a living in this unusual environment. And in this case, what we found is that actually there's one dominant organism that provides energy to the whole community, and it gets its energy actually from oxidizing ammonia found in the water within the cave. And that's how that entire community is basically making a living down there. These new sequencing tools we had also let me revisit my first interest, and that is bacterial multi-drug efflux pumps. And so using these sort of sequencing approaches, we were able to discover several new types of multi-drug efflux pumps. And in the case of one of them, the ACI efflux pump from Acinetobacter baumannii, which is a different dangerous multi-drug resistant hospital pathogen, we were able to identify this particular efflux that pumps chlorhexidine out of the cell and makes them resistant to chlorhexidine. And you may not have heard of chlorhexidine, but it's actually the primary antibacterial agent in most hospital hand washes. Now, it's an entirely synthetic compound though, so it's only existed for about 100 years on Earth, but this particular efflux pump is found in organisms that are separated by hundreds of millions of years of evolution. So we hypothesized that these pumps must have some function other than pumping out chlorhexidine, some natural function, and they had just been co-opted by the bacteria to provide resistance to this. And in the case of ACI, we were able to find out that putrescine and cadaverine, which are short-chain diamines, are actually the natural substrates of these pumps. And that they're just providing resistance to chlorhexidine in hospitals by opportunistically. So I've talked about how DNA sequencing technology, our ability to read DNA has improved. More recently, our ability to write DNA, to synthesize DNA has also improved. And that's empowered a new science that's emerged in the last decade called synthetic biology. And in synthetic biology, the central concept is we can treat genes and DNA as modular standardized parts, essentially as like Lego building blocks from which we can build new things, new synthetic microbes, and do things that no natural microbes can do. And I led the formation of a new $50 million research centre in synthetic biology, which aims to take either agricultural biomass or waste from various sources and build synthetic microbes that can turn that into industrial biochemicals, fuels, and plastics, things that we currently get unsustainably from cracking crude oil. And I don't have time to go through a lot of our ongoing synthetic biology research, but I'll quickly tell two stories. So I actually first foray into synthetic biology, we joined the Yeast 2.0, which is a global consortium across 6 countries, looking to build the world's first synthetic yeast. So this is a yeast where we've designed its genome on a computer, chemically synthesized and replaced its natural genome with this synthetic genome. And at Macquarie, we built a facility, the only one in the Southern Hemisphere, capable of synthesizing complete chromosomes. And we've now built 2 yeast chromosomes, 2 synthetic yeast chromosomes, chromosomes 14 and 16, as well as some entirely new to nature yeast chromosomes. And yeast is an industrial workhorse, important for brewing, baking, winemaking, but also now for production of bioethanol to put in your cars and for many industrial biochemicals. But yeast is like a small child. It loves to eat sugars, which we have to get from essentially from agriculture, sugarcane or barley or wheat. So we thought we'd like to try and engineer yeast instead of to grow on sugars, can we make it grow on waste? In particular, we wanted to engineer it to grow on methanol, which we could obtain from various waste sources. And we engineered 3 different pathways into yeast for using methanol, and 2 of these worked quite well. But what we actually also found in doing this is that even though people have used yeast industrially for 5 to 10,000 years, no one had ever realized that actually yeast has a native ability to actually use methanol as well as a carbon and energy source. It doesn't do it very well, but we were able to also use evolution to help improve that, as well as doing our design of our synthetic pathways. So that's all I've got time for today. This is my team here pictured clearly in pre-COVID days, because there's no social distancing. And for those of you who are interested in the Centre of Excellence in Synthetic Biology, here's various ways to get to contact us or get more information about us. Thanks.
Now, Professor Ian Paulsen joins us live. Congratulations, Ian, and thank you ever so much for your presentation.
Thank you, and thanks for having me. It's a great honour and a great pleasure to be here.
You mentioned how far our understanding has come in reading and writing DNA. Are we just scratching the surface?
Yeah, that's basically true. I mean, we've made tremendous progress, but we've still only, sequenced the genomes of a tiny fraction of life on Earth, and even in our best studied model organisms, E. coli and yeast, we still don't know what somewhere between 1/4 to 1/3 of the genes do, and for most organisms, we only have the vaguest idea of what most of their genes do. So I guess one of the take-home messages for me from my career as I've got into sequencing genomes and then metagenomes is it's really revealed how much we don't understand.
So certainly an area that's ripe for discovery, for young minds to be attracted to, hopefully. Ian, there must be risks associated with writing DNA. What are they and how are they mitigated?
So all sort of, we've been doing recombinant DNA research since the 1970s. We've been doing synthetic biology for the last 10 or 15 years. All that research is overseen by university or other ethics committees. And in Australia, they report to the Office of the Gene Technology Regulator. So it's all fairly well regulated. And so in order to undertake any of this sort of research, you need ethics approval. The other, and I often get questions about, how do I know I won't destroy the world? One thing which particularly the Yeast 2.0 project has taught us is all of these microorganisms we're working on have survived 3.8 billion years of evolution of struggling against each other to survive. And so they're really literally ruthlessly honed survival machines. When we're making these changes, writing new things in DNA, virtually every change we make seems to make them more feeble, less fit. And we see this practically in industrial purposes as well. If you take, we make, I don't know, tons, maybe thousands of tons of wine yeast. This escapes through the process of making wine. But if you go to, even a few hundred yards from a vineyard, you won't detect any of those industrial strains surviving out in the wild. So all of these engineered strains really, they might be good at doing something in a lab, or they might be good at something sitting in a million-litre bioreactor in an industrial setting, but they're no longer capable of surviving very well in the tough outside world.
My last question to you is, where would you like to take your research next? I feel like there is a world of opportunity.
Yeah, so one of our strong interests essentially is in trying to make the chemicals and plastics and fuels that support our modern lifestyle in a sustainable way. At the moment, you probably don't even realise it, but most of the things around you, all the plastics, things like that, are all made from crude oil. Hang on, let me step outside a second. Are all made from crude oil distillation, or sourced from crude oil distillation. And this is clearly not something that's sustainable, and yet we don't want to give up the features of our modern lifestyle. So we need to find sustainable alternatives for them. And so we'd really like to engineer microbes to produce the chemicals, the plastics, the fuels that we need to continue our modern lifestyle.
Fantastic. Well, all strength to you, Ian. Congratulations again on being elected to the Australian Academy of Science. Good to see you.
Thank you.
Please now welcome back Professor Jonathan Sprent to introduce our next presentation.
Our next presentation is from Professor Brendan Crabb. Brendan is a pioneer of genetic technologies in human malaria and fundamental discoveries in malaria pathogenesis. His team discovered the malaria translocon, a protein machine that allows the translocation of malaria proteins into its host cell, a highly vulnerable point in the parasite's life cycle. He was the first to provide an order for the myriad of receptor-ligand interactions that mediate red blood cell invasion by malaria parasites. Please welcome Brendan.
My name's Brendan Crabb, and it's a great pleasure to be able to speak to you about some of the work that's got me here. And it's terrific to have in the room, I hope, listening to this, Professor Alan Kalman, with whom I've worked for the best part of the last 25 years or so, and who introduced me to malaria in the first place. And I'd like to acknowledge him right here and now. And I'd also like to acknowledge Dr. Paul Gilson, who I now run a group with. Paul used to be a postdoc with me, but he shared most of the science that I've done, certainly in the last 10 to 15 years. I have two careers, one as a virologist, which I'm not going to speak about today, but it's becoming very handy, of course, through the COVID pandemic. But my main scientific contributions has been in the area of malaria, and particularly of understanding the molecular basis of how malaria causes disease, some of its pathogenic processes. Malaria is a problem with a geographical side to it, as this world map shows. Everywhere in red is where there's lots of malaria. And it also has strongly associated with poverty. And of course, where malaria is, which is in red, are mostly countries that are very poor. And that tells you a story, a long historical story of the intersection of malaria and poverty. And it's one reason, a very big reason, why malaria as an infectious disease of humans is still so important in history and so important now. People who get it don't have many resources. The countries that those people are in don't have many resources, and that's an impediment to finding solutions. But it's not just a disease of poverty. It's a technically very tough challenge. As this very simple graph shows, the progress that's been made since 1990 on, for example, respiratory infections and diarrheal diseases is phenomenal. And that's underpinned tremendous improvements in life expectancy because of the survival of kids that are under 5. But when you look at malaria here, despite enormous effort, very similar effort to what's happened for respiratory diseases and diarrheal diseases, there hasn't been anything like as big a change. It has been improving significantly over the last decade, but it's got a long way to go. It's a technically very tough challenge. At the time my scientific career was starting and I was doing a postdoc with Alan Cowman, we were very lucky in our historical timing to be doing this work at a time when the 3 sort of intersecting genomes that relate to this parasite were being either published and, of course, being done before publication. The human genome, the mosquito genome of the main transmitter of malaria, Anopheles gambiae, and the malaria parasite itself. We had the genomes of all 3, and all 3 genomes have co-evolved. Malaria is one of the most powerful selective forces in the last 50,000 years or so, and even beyond that, of humans, which is why humans have red blood cell diseases as just one example. So the work that we were interested in doing was taking this new genomic knowledge and using that to better understand disease processes and try to find new tools that empirical approaches had not been that successful at discovering. My work really for all of the time since the mid-90s that I've worked on malaria has been interested in 2 key processes, the understanding of which we'd hoped could lead to vaccines, for which we don't have any robust vaccines, except for last week, of course. Just last week in the world, a licensed vaccine for malaria was announced, but it's not a very effective one. It's going to be tremendous to have, but it's more a first cab off the rank proof of concept rather than a robust malaria vaccine. And this understanding of how parasites invade their host red blood cells is a big part of trying to develop a rational basis for a malaria vaccine. And the second pathogenic area of interest was known as host cell remodeling. So once a parasite goes into a cell, it renovates that red blood cell and turns it into more like a normal cell that has certain characteristics. And the big characteristic it has is that red blood cell can be taken out of the circulation by binding to the walls of blood vessels. And that keeps the parasite from the circulation and from getting destroyed by the spleen. So there's a lot of interest in this process of host cell remodeling. And all of that was underpinned, all of our investigations were underpinned by genetic tools, molecular genetic tools that we helped develop. And here's the first gene knockout in malaria of a protein that was responsible for this rough appearance on the surface of red blood cells, and turned into, as you can see, the surface of the red blood cell becomes smooth. And in fact, these become non-adherent red blood cells. They don't have that sticky capacity anymore. And this work was done while I was a postdoc with Alan's lab, in Alan Cowman's lab. But the main point of showing it is those sort of molecular genetic tools were what provided the sort of breakthrough advances for all of the next 25 years or so for us. Here is another technology that was important, and this is real-time imaging of the invasion of red blood cells by a malaria parasite. Here's a malaria parasite. This is in real time. I believe the first movie published of real-time invasion by postdocs with me, but particularly actually a PhD student, Tana Taichelat Paisan. And here is this parasite going into a red blood cell where it's going to live for the next part of the best part of 48 hours or so. And it's this process that is highly vulnerable to vaccine attack and even drug attack. And that's why it interested us so much. And what we determined was that this relatively fast process is the 10 seconds here, it's another 20 seconds or so here, and a minute or up to 5 minutes here, was distinct phases. And the next part of 20 years or so was determining that each of those phases had molecular interactions that we could dissect and put the players down on both the erythrocyte side and the parasite side, known as the merozoite. And the order of events and the nature of the specific players has led to vaccine candidates that make logical sense being developed. And there's quite a few candidates in this phase here, this obligatory step that has been resolved through these molecular studies that obviously we've played a key role in. This is a summary of the work of many people around the world, but that we played a very substantial role in. And of course, where we're at the moment is saying, well, with that knowledge, can we exploit it as vaccines and drugs? And here's just a slide to look at current work that we're involved in, which is identifying new drug targets. And here's a drug that we've identified that blocks the motor that takes a parasite into a red blood cell. And here's one that blocks invasion at another step. Injecting lipid into the red blood cells is a very important part of invasion. So we identified these drugs by screening and then identified their targets in other ways. This is going to be a big part of us, of our future of using these drugs as functional tools to dissect this process further. And we'd like to actually make drugs with them. And here's back to red cell remodeling. Our real interest here is to identify how all of the proteins, the parasite proteins that are made here, get out into this red blood cell cytosol. About 5% or so of the parasite's genome encodes for proteins that go through into the red blood cell cytosol. And we discovered this complex here known as PTEX. And PTEX turned out to be a molecular machine. We published it back in 2009. And now another group has published this beautiful image of a cryostructure of this that completely validated that we were correct. And it becomes one of the most important drug targets in malaria for the future, as blocking this can interrupt the export of 5% of the whole genome. So with that, thank you very much. And here's my group that are currently working with Paul Gilson, who co-heads the group with me on the work that I'm doing now. And of course, I've mentioned those who've been so important to me over the last 25 years. Thank you.
Now we're delighted to have with us Professor Brendan Crabb. Welcome, Brendan, and congratulations on being elected to the Academy.
Thanks so much, Anna Maria. Great to be here.
I have a question for you. Well, what is it about malaria that makes it a technically tough challenge to tackle?
Yeah, good question. I mean, the main one is its plasticity. It is highly variable. It's a bit like trying to deal with HIV or even with influenza virus. We've got tools for both HIV and influenza, but they quickly become obsolete. In fact, we don't even have a vaccine for HIV. Same reason, their genomes are so plastic, including in the same infected person. You end up getting infected with a clonal form of the parasite and it changes its spots very quickly. And so, You try and make a vaccine and it avoids the vaccine. You try and make a drug and it avoids the drug. So that's the main reason. And, a big contrast to something like COVID. When COVID came along, that was our fear that would be the case. It turned out to be largely stable. And so we could make a vaccine against COVID. Of course, maybe we'll need more in the future. But stability has a lot to do with solving pathogens. And when they're variable, they're really tough.
There was news recently of a potential vaccine for malaria, and you've said that it wasn't that effective. What does that mean? What is it a first step? Is there promise?
Yeah, it depends whether you're a glass half full person or not, and I definitely am. I wasn't sure we would ever see a vaccine licensed for the reasons I mentioned, but one has come along, and it protects the main target population, which is young babies and young children in poor settings. The biggest trials have been done in Africa in more than 1 million children now. And it protects them about 50%, probably at best, from severe disease for the best part of a year. But between 30 and 50% protection for a year compared to COVID, which is protecting you by 97, 98% against severe disease, probably for 5 or 6 months, and then it's waning. So look, that's real. And when you, in the case of malaria, say, well, there's nearly 500,000 children who die every year. There's 300 or 400 million severe infections every year, severe diseases. That's a big public health benefit. But it's nowhere near the sort of holy grail vaccine that we would like. A child to get a couple of doses in there, before they're 6 months old and be protected for life. That would be our dream.
A final question for you from one of our fellows. Congratulations, Brendan. I noted you used the term membrane rigidity in your second last slide. Is the malarial membrane more or less fluid than the red blood cell membrane, which is ordered by cholesterol?
Yeah, it's a good question. My reference to that relates to the red blood cell membrane, not to the parasite membrane, and how the red blood cell membrane changes from being very plastic, when very malleable, when it's not infected, which it needs because normally in circulation, a red blood cell would squeeze itself through the spleen, the fenestrations of the spleen. That's the mechanism for the red blood cell to get tested every time it goes around the circulation. Once the parasite infects it, renovates the red cell, as I mentioned in my talk, and the red cell membrane becomes quite rigid for a whole host of reasons. It's not just because of the lipid composition changing. In fact, I'm not sure the lipid composition does change that much, but the architecture underneath the red blood cell membrane changes a lot, and it becomes very tough, very cell-like. And that's one of the reasons why the infected cell can't stay in the circulation, because if it did, it would find its way to the spleen, and the spleen would go, aha, that is not a normal red blood cell. I will get rid of that. Thank you. So it adheres then to vascular endothelium and stays out of the circulation. So that was my reference to rigidity.
Thank you ever so much, Brendan. Before I let you go, I know I'm speaking on behalf of many in thanking you for the very proactive work you've done in COVID-19. It's been a tireless effort and a magnificent one, and particularly around encouraging vaccination in low vaccinated populations around the world. Thank you ever so much, and congratulations again on your election to the Academy.
Thanks, Anne-Marie. It means a great deal to me. Thanks, everybody.
To introduce our next fellow, please welcome Academy Council member, Professor Bob Williamson.
Thank you. Our next presentation is from Professor Adele Green. Adele is an international leader in the epidemiology of melanoma and skin cancer. Her research provides the scientific basis for clinical and public health advice about sunscreen use for skin cancer prevention. Her significant contributions also include insights into risk factors for ovarian cancer and the burden of cancer on Indigenous Australians. Please welcome Adele.
Hello, everybody. I'm a cancer epidemiologist.
and I'd like to share with you some of the highlights of my career in scientific research. I've been particularly interested in melanoma and other skin cancers through my career, with a great emphasis on prevention. I've been involved with a wide range of descriptive studies as well as my central focus, and some illustrations of the investigations and findings over the years have been in looking at disparities in the cancer incidence and mortality rates in different countries and populations. That's included studies of cancers in Indigenous Australians. We were some of the first to describe the very high rates of lung and throat and liver and cervix cancer in Indigenous Australians in Queensland compared with non-Indigenous. I've had some exciting work in cancer of the gallbladder, which is due to liver fluke in northeast Thailand, very common there and yet extremely rare in Australia. And this gives us pointers for prevention, and also ranging as far as looking at brain cancer cluster in the Lake District in England and identifying if there could be some issues there for the population. I've had other focuses through my career, and a main highlight for me was a lot of research into ovarian cancer, in particular its causes. And again, we were carried out some of the earliest studies in the causes of ovarian cancer and identified the central role of oral contraceptive pill and confirmed that this is highly protective, as is tubal sterilisation, while on the other hand, perineal talc and cigarette smoking can be causative to certain types of ovarian cancer. But my main interest through my career has been the epidemiology of melanoma, starting at the incidence and mortality and charting its very rapid rise along with other Western countries of European origin in the 1960s to the 1980s. And then recently we've started charting now the decreases very gratifyingly in younger generations since the 1990s. And this is probably very much sheeted home to the success of SunSmart campaign in Australia. Other aspects have been the natural history of melanoma, in particular looking at some of the earlier studies of melanocytic nevi, which have a central role in melanoma genesis, and also looking at some of the very early studies of the causes of melanoma and showing that multiple sunburns are a significant risk factor, especially those in childhood, but also long-term sun exposure, and that's very easily illustrated by showing that per unit area of skin, the male ear uncovered and long sun exposed is the peak site of melanoma occurrence in Australia. I segued from epidemiology of melanoma into epidemiology of the other very common, more common skin cancers, basal cell carcinoma, which you can see at the top, and squamous cell carcinoma. And we've again charted the incidence not documented in the cancer registries around Australia, except in Tasmania. And so with Queensland having the highest rates, we've looked at the very high rates here, highest in the world, and also shown that even higher in a subgroup of our population are the immunosuppressed organ transplant recipients with unbelievable almost incidence of particularly squamous cell carcinoma. We've looked at the natural history of skin cancers, in particular the role of actinic keratoses, or known as sunspots, and also charted the even further premonitory signs of leading to skin cancer, and that is severe photo damage or premature photoaging. And that is seen, unfortunately, in Queensland as early as late 20s and 30-year-olds. And the main cause of all of these cancers and the other related conditions is overexposure to solar ultraviolet or UV radiation. And so that naturally one thinks this is preventable, and that then has become and been a mission for me through much of my career. 30 years ago, we knew that slip, slop, slap, such a wonderful slogan for protection, was protecting our population from sunburn, but did it protect against skin cancer was the question that was unanswered. And indeed, if you just look naturally in the community, you'll find that sunscreen use looks like it's positively associated with skin cancer for the very reason that the predictors of sunscreen use, such as having fair skin that burns and having a lot of outdoor activity, is precisely the same set of risk factors that leads to skin cancer. So they look like they are one and the same. And therefore, the only way that we can really assess whether sunscreen can prevent against skin cancer is to carry out randomized controlled trials. And that led naturally to the conduct of the Nambour skin cancer prevention trial, carried out in the community of Nambour, about 100 kilometres north of Brisbane in southeast Queensland. We randomised the community, who were representative of the community. 812 adults were randomly assigned to using daily sunscreen every day with which they were provided, and the control arm was assigned not to use sunscreen every day, but of course they could use sunscreen at their own discretion, which could include no use. The intervention sunscreen was an SPF 15 plus broad spectrum. Remember, this was carried out in the 1990s before higher SPF sunscreens became available. And participants were asked to apply the sunscreen to their head and neck, arms and hands every day, no matter what the weather, for 4.5 years. And we had excellent compliance. Unique evidence that came from the Nambour skin cancer trial has been instructive for all European-based and ancestral communities, and that is white-skinned communities, because it shows that sunscreen, regular application of sunscreen, can prevent squamous cell carcinoma, the second commonest cancer in white-skinned people. It can also reduce melanoma. And furthermore, the premonitory conditions, actinic keratoses, reduced by nearly 1/4 in the rate of increase, and similarly, premature aging in those under 55 are also 25% less likely the changes to occur. The commonest cancer, basal cell carcinoma, did not show any overall effect, but we did show that there was a decreased chance of multiplicity of basal cell carcinoma. So in all, what the long-term public health and clinical significance of these findings have been is to be able to tell Australia and the world that indeed we can include sunscreen, to slopping on some sunscreen, in one of the ways to protect against skin cancer. I'd like to finish with acknowledging what a privilege it has been to be a researcher so far in my career, and for the joys of collaboration. It's been marvellous to have the ability to carry out curiosity-driven research and have the thrill of discovery. Benefits of scientific knowledge for public health has been extremely rewarding, as has been helping to increase community understanding and self-help in controlling health and prevention and well-being. The role of sustained funding for the research that I've done has been absolutely crucial, through mostly the National Health and Medical Research Council. And I'm also indebted to my home institute, formerly the Queensland Institute of Medical Research, and now QIMR Berghofer Medical Research Institute, for decades of support and nurturing of research of my colleagues and myself. Nothing would have been achieved without the teamwork that I've enjoyed, research staff, clinicians, community members. Working with my doctoral students has been absolute career highlights. And finally, I've had the most outstanding collaborators, and they have been pivotal to my scientific achievements. Thank you.
Please now welcome, live with us this afternoon, Professor Adele Green. Congratulations, Adele, and thank you so much for that wonderful presentation.
Hello, thank you.
Adele, why is it that the male ear is the most common site of melanoma in Australia?
Men don't wear their hair usually over their ears, maybe not since the 1960s and the Beatles, but they've gone back to having that exposed. It's like the nose, one of the most easily accessible to direct ultraviolet radiation, and it's therefore receives long-term ultraviolet exposure, sun exposure. So in essence, it's really just a very easily identified cameo of how much sun it gets, and extraordinary that it's so prone to these skin cancers.
Yeah. Were you surprised by the high levels of compliance in the NAMBOR clinical trial over such an extended time frame? It was 4.5 years.
I think that this is actually an expression of the wonderful cooperation we had. This was a community-based trial, quite unusual really, and without the compliance of the community, and remember, it's not just the compliance that was putting on the sunscreen every day. You'll remember that there was also a group who were asked not to put on sunscreen every day, and we had to have compliance there as well. But the community were marvellous in terms of sticking to what they asked. We did have a few people whose spouse had been randomised to sunscreen and not them, and they were very tempted when they could see that even the wrinkly skin seemed to disappear, that they wanted to join in and not be compliant, but we had very good compliance, but it was all an expression of wonderful cooperation.
So the anti-aging factor was a good motivator.
You could see it very quickly, and it meant a lot, even to the men.
Yeah. And tell me, how has your research had an impact in other parts of the world where the climatic conditions might be similar to Australia, but there might be lower awareness in the population of the dangers of SCC and BCC and melanoma?
Really, the European ancestry is key to risk of melanoma and skin cancers. You said in similar climatic conditions, but really this has got universal transferability to other populations, even in high latitudes like Norway, where I've got many collaborators. Of course, they have darkness for a lot of the year, and yet knowing that protection and sunscreen can help because they're such addicts of the sun. So it actually goes right around the world to show that sunscreen is not just an acute protector, but a long-term protector, even in the sun in higher latitude countries like Northern Europe, Norway, New England in the United States, for example, or Canada.
Fantastic. Adele, I'm afraid that's all we have time for today. Thank you. We hope to see you in Canberra soon, and congratulations again.
Thank you very much.
Please now welcome back Professor Bob Williamson to introduce our next fellow presentation.
Thank you. Next, we will hear from Professor Andrew Roberts. Andrew has conducted groundbreaking research that has delivered venetoclax, a novel drug for blood cancer patients. Venetoclax is approved in Australia and internationally for chronic lymphocytic leukemia and for acute myeloid leukemia in the United States. Now we will hear from Andrew.
Hello, my name is Andrew Roberts, and I'm privileged to be presenting today on behalf of many colleagues about our research that's led to a new therapy for blood cancers. To help give some context for the listeners, blood cancers are a major challenge in Australia and around the world. Leukemias, lymphomas, and multiple myeloma account for about 10% of all cancer diagnoses in our country. That's about 15,000 people per annum. They also account for 1/6 of all cancer deaths in Australia and the loss of many productive life years. They also represent a major opportunity for improvement, and they're a leading area of the application of precision medicine to oncology care and research. In my career as a haematologist over the last 25 years, we've seen two uncommon incurable cancers now readily turned to cure in most people through the use of non-chemotherapy-based treatment. And the key to this has been the application of rationally designed targeted therapies. Indeed, most modern cancer therapy that is now designed is meant to be targeted, looking to identify the Achilles heel of individual cancers and then to target that weakness. However, to design new therapies, a priori, we first need to understand the biology of cancers in depth. As a young haematologist in the early 90s, I was absolutely inspired by members of this academy, the Australian Academy of Science, the late Professor Don Metcalf and Professor Suzanne Currie, as well as their colleagues at WEHI, because they understood the biology of the blood cancers I was trying to treat in desperately ill patients. I also saw that that knowledge was not yet being applied to the care of these patients. So the rest of this story is about one example how basic research has led to the development of a new treatment that is routinely used in Australia today. The discovery was by Vo, Currie and Adams. And in Nature, they described the function of BCL2, a protein of previously unknown function, but implicated in cancers. They revealed to the world that its function was to regulate cell survival and that high levels of BCL2 were associated with prolonged survival and avoidance of apoptosis. This research opened the whole field of apoptosis, and there's been a strong intersection with cancer research ever such. In the middle panel, I show what happens in normal cells where BCL2 is a pro-survival protein necessary to keep cells alive. When, however, they are damaged or meet a lethal stress, its activity is counteracted by the natural antagonists, BH3-only proteins that respond to the stress signal and lead to the appropriate apoptosis of damaged cells. On the right, you see what happens when BCL2 is inappropriately overexpressed in cancer cells. That same stress does no longer lead to apoptosis, and the cancer is allowed to continue to grow. So at an early time, once the discovery of the function of BCL2 was made, it was apparent that this could be a good therapeutic target. But how to target it was a major conundrum. And a key breakthrough was made in the early 2000s by the group at Abbott, now AbbVie, in the United States, where they developed small molecule inhibitors that bound tightly to pro-survival proteins like BCL2, and in this instance shown in the slide, BCL-XL, and by doing so, inhibiting their function. The first molecules were ABT737 and ABT263, and they inhibited 3 pro-survival proteins, including BCL2. We did a lot of independent work proving the mechanism of action with these early compounds. We identified targeted disease where we thought there would be major clinical benefit based on laboratory data. We also identified where the likely toxicities would be seen from targeting BCL-XL with molecules such as ABT737 and navitoclax. And I led the first phase 1 trial of navitoclax in patients with chronic lymphocytic leukemia. What we learned from this was that there was potential here, but that we needed to do better. We needed to target BCL2 specifically, both to increase the effectiveness of the treatment and to reduce the toxicity that was coming from targeting proteins other than BCL2. A commercial partnership between Genentech, AbbVie, and scientists at WEHI led to the development of the world's first specific drug specifically targeting BCL2, now known as venetoclax. We knew from our preclinical work that chronic lymphocytic leukemia was likely to be the disease which we would get answers in the clinic most quickly. CLL is an accumulation of malignant mature B cells in the blood, shown on the left-hand side here, and in the lymph glands, shown on the right-hand side. These cells accumulate in large numbers, and that is due to very high levels of BCL2 preventing apoptosis. So we thought this was the perfect scenario to test the concept that inhibiting BCL2 in patients should lead to apoptosis and death of leukemia cells. And that's exactly what we saw. And we saw it after the very first dose, in the very first patient, in the very first clinical trial of venetoclax in the world. And that was conducted at the Royal Melbourne Hospital. And in July 2011, we saw that there were very rapid responses in the first patients treated. Blood films went from that shown in the centre to that on the right, with the clearance of the leukemia cells within 8 hours and sustained for 24 hours after a single dose. And when we gave multiple doses over several weeks, and these are tablets taken daily, you see marked shrinkage of these abnormal lymph glands in the neck to a more normal appearance. So the effectiveness was immediately evident. Consequently, the further clinical development of venetoclax was very rapid through early phase trials in CLL lymphoma and myeloma, and then in combination with other anti-cancer drugs such as monoclonal antibodies, kinase inhibitors, and DNA-damaging chemotherapy in CLL, lymphoma, myeloma, and AML. The first approval for use as a routine was in April 2016 in the United States for patients with relapsed chronic lymphocytic leukemia. Our other work subsequently discovered the first mechanisms of resistance, and this is guiding further development. And I'll touch on the mature phase 3 trials in a moment. But before I do that, this slide summarises what we know about inhibiting BCL2 and the effect that it might have on blood cancers. There are many different types of blood cancers, but now we can divide them up into those that have a significant proportion of cells that are BCL2 dependent, where we expect a major effect of venetoclax indeed on its own, and those that are only partially dependent, where we're going to need to combine BCL2 inhibition with other anti-cancer drugs. And in fact, that is the majority of blood cancers. So what about the phase 3 trials? These are randomized trials testing whether venetoclax-based regimens are as good as or superior to the previous standard therapy. And for both chronic lymphocytic leukemia and acute myeloid leukemia, the venetoclax-based regimens led to marked improvement for patients over the previous standard treatments, both in terms of leukemia control and in survival of patients. That's most evident in patients with acute myeloid leukemia, where the survival at 2 years was more than double that observed with standard chemotherapy. So what is the impact of venetoclax in 2021? Well, it's now a standard therapy for CLL in Australia, USA, Europe, and many parts of the world. And it's an emerging standard of care for older patients with acute myeloid leukemia. Hundreds of Australians are benefiting every year from it now. But it's early days in its clinical development, and there are over 300 trials being conducted currently exploring its place in treatment for other blood cancers based on preclinical concepts. And the success of venetoclax has catalysed the development of other small molecule inhibitors of BCL2 and like proteins. And a successful story like this is dependent on the work of many. In fact, this is a team of teams. The multidisciplinary team from WEHI, shown on the right-hand side, with scientists associated with the development of venetoclax. And the stars of the show are shown in the middle row, particularly my collaborators, David Wang, who co-led the translational and preclinical research, and John Seymour, who co-led the clinical research with me, with the work done by a very talented group of young hematologists and scientists as students, postdocs, and many of our colleagues. We received generous funding from philanthropy and NHMRC for the public good research, and the commercial research was funded by AbbVie and Genentech. Thank you.
Please now welcome live with us Professor Andrew Roberts. Congratulations and welcome.
Thank you very much, Anna.
Thank you for a fascinating presentation. I'd like to ask you, how does it feel when you first discover that the drug you've produced and had a significant part in developing is effective in clinical trials and it's saving lives across the world?
Well, it's an absolute joy to know that this drug was working. And while we were the first people to give patient this drug, the first person in the world who realised it was working was actually the first patient who said to me, "Doc, I think this is working." I was a little sceptical and had to wait till the next day, but they were right. It was a pretty impressive moment for my colleagues and for myself.
It's tremendous. And going forward, what are the ongoing challenges for blood cancer treatment now that venetoclax is being used and given that most cells are partially BCL2 dependent?
There's a number of challenges. I think the development of new treatments in haematology has taught us that there's always more work to do. A drug like venetoclax is making a major impression for people with chronic lymphocytic leukemia. We're aiming to do better. Ideally, we'd like to be able to get rid of that disease for good, i.e. Cure this. And the only way that will happen will almost certainly be combinations of treatment. We're working on combinations of treatment that include venetoclax. In terms of the other blood cancers, some of these have very poor prognosis. And acute myeloid leukemia, particularly in older patients, is a devastating disease for which we need much better therapies. I'm hoping that venetoclax will be part of the solution. At the moment, it looks like it's giving advantage that we couldn't achieve previously, but there's a long way to go. So that's going to require laboratory research, clinical trials, and then roll out into the clinical space.
A very exciting discovery journey ahead. Can venetoclax and similar drugs target other types of cancers, not blood-related cancers?
Yes, so these belong to the class BH3 mimetics, and venetoclax is the first in class to be used in routine practice. But targeting MCL1, another pro-survival protein, or BCL-XL, another pro-survival protein, or these in combination with BCL2 may well be effective in solid cancers and potentially in other diseases. Certainly, prototype drugs work well in vitro and in preclinical models, and MCL1 inhibitors are in clinical trials now, early phase clinical trials, and we await their results with interest. We've gone first with the blood cancers because of the pivotal role of these pro-survival proteins in those diseases, but it's not restricted to the blood cancer.
Andrew, thank you ever so much. That's all we have time for today. It's been a pleasure to speak with you, and we look forward to welcoming you in Canberra as a newly elected Fellow of the Australian Academy of Science. Thank you and congratulations again.
Thank you very much, and I can't wait.
To introduce our next Fellow, please welcome back Professor Bob Williamson.
Thank you. Our next new Fellow presentation is from Professor Arthur Christopoulos. Arthur is a molecular pharmacologist who has made seminal discoveries of spatially distinct allosteric drug binding sites on G protein coupled receptors, the largest class of all medicinal targets. His research at the interface of chemistry and biology, with a particular focus on neuropsychiatry, cardiometabolism, pain and addiction, is transforming the way these medicines are discovered in academia and industry. Now to Arthur's presentation.
I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Monash University Parkville campus stands, as well as acknowledging the traditional custodians of the various lands on which you're all joining me from today, and the fact that sovereignty of these lands was never ceded. I pay my respects to their elders, past, present, and emerging. I, of course, also want to thank the Fellows of the Academy for bestowing me the honour of electing me into your ranks. Over the next few minutes, I hope to provide you with an admittedly whirlwind overview of an area that has fascinated me since I was an undergraduate pharmacy student, and that is the versatility and malleability of G protein coupled receptors. GPCRs are a superfamily of proteins that act as the major conduits by which we sense our external environment. They recognize an extraordinary array of activators ranging from ions to nutrients, lipids, neurotransmitters, hormones, large proteins, and even light. Activation of GPCRs leads to a conformational change that engages intracellular transducer proteins, the best characterized being these heterotrimeric G proteins, and hence the name, to then further trigger a cascade of biochemical reactions that ultimately lead to an integrated cellular response. the significance of GPCRs can be further appreciated when we consider that just a relatively small subset of the entire GPCR repertoire nonetheless accounts for at least a third, if not more, of all current drug targets. Now, nonetheless, GPCR-focused drug discovery suffers from the same issues that plague all major discovery programs when it comes to clinical failure rates. As shown here for phase 2 and over here for phase 3 clinical trials, we can quite strikingly that no matter what we thought we knew in the preclinical and translational phase of a drug development program, more than 50 to 60% plus of all medicines still fail to make it to the market due to a lack of efficacy. Now, there are many reasons for this, the most obvious being the polygenic nature of most global health burdens. But for me, I've always felt that the drug discovery process has failed or had failed to fully appreciate and capture novel paradigms of drug action based on what we've progressively learned about protein behaviours over the years, and often from other protein classes. And for me, pharmacologically, that lens was defined by the phenomenon of allostery. Now, this slide is going to summarise at least 25 to 30 years worth of research. The binding site for endogenous receptor activators to antagonists is defined as the orthosteric site, and it's akin to an on-off switch. Over 90% of all drugs in the market target this site to either activate it or block it. However, we and others have shown that all GPCRs, and that's a lot of proteins, possess spatially distinct allosteric sites, which can change the receptor conformation to modulate the activity of orthosteric ligands akin to the properties of a dimmer switch or a rheostat. Now, there are at least 4 major advantages of allosteric drugs as medicines. First, allosteric drugs can be much more selective than orthosteric drugs because these sites have not necessarily evolved to accommodate a common agonist activator between subtypes of the same receptor. Second, and in my mind, the most important molecular differentiator of allosteric drugs, the direction and magnitude of the drug effect is titratable. It approaches a ceiling. It's saturable. It's completely governed by the conformational communication between the 2 binding sites in the protein, such that it can be very subtle or it can be very large. And this bidirectional communication is often referred to as comparativity, and it is thermodynamically reciprocal, meaning that what the allosteric ligand does to the orthosteric ligand, the latter can do the same to the former. That is, they sense each other. And this means that we can literally titrate the allosteric drug to its target, that it's going to be safer in terms of on-target overdose scenarios, and that's going to be sensitive to disease context, and I'll come to that later. Third, the promise of allosteric drugs is that they can be designed to do nothing on their own in the absence of an orthosteric activator, meaning that the medicinal effect is more natural because it's only expressed when and where the endogenous chemical transmitter is released. And finally, because allosteric drugs change the receptor conformation, they can also change the normal rules by which the receptor signals. That is, they can bias signaling towards certain pathways at the expense of others. Now, although much of the work in GPCR allostery has been driven by pharmacology, there's been an explosion in the last decade and a half in the field of structural biology, which we summarised here a couple of years ago, and it's already out of date. But it confirms the fact that these dimmer switch regions do indeed occur anywhere on these receptors, from the extracellular environment all the way through into the intracellular space. In essence, because the architecture is similar but highly dynamic for these proteins, one type of receptor's orthosteric site can be another type of receptor's allosteric site. So the realization that allostery is a universal phenomenon at the largest class of drug targets meant that we had to develop methods to pharmacologically quantify allosteric effects in a manner that can drive medicinal chemistry and drug discovery programs. So this slide summarizes one such example of where we developed analytical pharmacology methods and applied these to understand the chemistry of positive allosteric modulators of the M1 muscarinic receptor, an important target for cognitive disorders. At a minimum, allosteric drug discovery programs need to be able to differentiate at least 3 properties: the ability to bind, the ability to activate, and the ability to modulate. It's very common to introduce chemical modifications to make one property go up, the other one go down, so they cancel each other out. Standard drug screening approaches cannot pick this up. So it's likely that a lot of promising preclinical programs for decades were scrapped for such reasons. Now, we've subsequently shown how combining analytical pharmacology with medicinal chemistry, structural biology, and preclinical disease models can inform GPCR drug discovery for a range of disorders, including neurodegeneration, schizophrenia, diabetes, obesity, and heart failure. But I'd like to conclude by showing you an example of this based on our most recent published work, specifically aimed at identifying novel, non-opioid analgesics for treating chronic neuropathic pain, because I think it's a nice example that brings all this together. So, shown here is a diagram of the dorsal horn of the spinal cord. The nucleoside adenosine is one of the body's most important cellular protective molecules, and previous work has established that its actions on the A1 adenosine receptor subtype are important for dampening neuronal activity. Now, following the development of neuropathic pain, there's actually an increase in adenosine release in the dorsal horn from primary afferents and from astrocytes. So we thus hypothesized that positive allosteric modulators of the A1 receptor should not only be able to boost this effect in the spinal cord, but that the allosteric drugs would actually work better under conditions of neuropathic pain than under conditions of no pain, because the former's got more adenosine in the spinal cord, in essence showing target tissue and disease-based selectivity. So to test this hypothesis, we investigated a series of 2-amino-3-benzoyl-thiophene modulators that we synthesized as part of a previous medicinal chemistry program. So the compound on the left, VCP-171, binds with a higher affinity than the compound on the right, which is 521, but the latter over here has substantially higher cooperativity than VCP-171. In essence, this boosts adenosine by a factor of 6, this boosts it by around a factor of 60. So does it make a difference? Well, first of all, using rat spinal cord slice patch clamp electrophysiology, we found that both modulators were able to reduce evoked excitatory postsynaptic currents that were mediated by the A1 receptor, and this was confirmed by blocking the effect with the antagonist, the A1 antagonist DPCPX. Now, interestingly, when we determined the dose-response relationships of each of the modulators to boost the inhibitory effect of adenosine, only MIPS521, shown here in red, exhibited a significantly greater effect in rats that had undergone partial sciatic nerve ligation as a model of neuropathic pain relative to sham-operated controls. So this was confirmed in vivo, shown here, using a model of rat paw withdrawal threshold, where you can see a very modest effect and only the highest dose of 171, whereas a clear dose-dependent effect of 521. So in essence, this study provides proof of concept that the unique molecular property of cooperativity of allosteric drugs can be used to dial in activity that will be sensitive to existing endogenous tone in a tissue and disease-specific manner. And excitingly, we also use cryo-electron microscopy to solve the first high-resolution structure of the activated A1 receptor bound to both its cognate ligand, adenosine, shown here, its G protein, shown here, and our novel positive allosteric modulator, shown here. So this is essentially a molecular snapshot of the whole shebang in action, and to allow us to identify a novel extrahelical lipid-facing allosteric site that can now be used to drive new structure-based discovery of more potent and optimized allosteric modulators. And in case you're wondering, this is both a biological and pharmacological concept that can be applied to any drug target. Receptors are proteins, and we should not forget that. So this figure is taken from a review that I was fortunate enough to write with the pioneer of the field of allostery, Jean-Pierre Changea, highlighting how all major receptor families have the potential for allosteric drug targeting. Now, we all know that it's impossible to appropriately acknowledge all the collaborators, mentors, mentees, and funders in a brief presentation such as this one. And I'm only listing on this slide the names of the key folk that have contributed specifically to the studies I shared with you today. However, it would be remiss of me not to highlight a couple of key individuals. First and foremost, Patrick Sexton, who's been my dear friend and collaborator for over 2 decades. Our friendship and collaboration is the very definition of the whole being greater than the sum of the parts. Peter Scammels, for nearly as many years, collaborator in the medicinal chemistry space. Celine Vallant and David Thal. This is pretty much their lab now. And importantly, Fred Mitchelson, my PhD advisor, who introduced me to the world of pharmacology and allostery. And of course, my wife, Tracy, our children, Sarah and James, and my wonderful parents, Chris and Willa. And thank you for your attention.
Please now welcome live with us Professor Arthur Christopoulos. Congratulations, Arthur, and what a wonderful presentation.
Thank you, Anna Maria. Thank you. It's wonderful to be here. I'm delighted.
If we have questions from our audience, please pop them into the Q&A on your screen, and I'd be very happy to ask them on your behalf. May I ask about where you think your work could take healthcare and the treatment of disease in the future? You did mention a range of conditions. What sort of pathways are you seeing?
I think the conditions that I mentioned all represent huge global health burdens. So whether it's neuropsychiatry, which is really making a comeback, whether it's cardiovascular disease, which remains number one, or chronic pain, for instance, and addiction. I believe that in terms of therapeutic areas, we're already trying to hit a couple of the really big ones. But the important thing, I feel, is bringing industry and academia along with us on the journey to actually translate these molecules into actual medicines. I mean, if you look at the presentation just before me, what Andrew did with venetoclax, for instance, I feel we're poised to rationally translate these types of allosteric discoveries onto the market as rationally designed medicines.
Absolutely. And of course, you mentioned the importance of cross-disciplinary research in medicine to get to drug discovery and application in medicine. That's particularly important. How much has your work been influenced or continues to be influenced by cross-disciplinary research?
99+%. None of this would have happened if we didn't apply cross-disciplinary research. I started with pharmacology, but I quickly realized that you need chemistry. Okay, chemistry is king. But not long after that, we realized we need in vivo appropriate animal models. So that came on board as well. And so we've essentially progressed along that pathway. And most recently, you see, we've now got into the field of structural biology. So we're the only group in Australia to date that's been solving both originally crystal structures and now cryo-EM structures of GPCRs. So you never stop learning and you never stop bringing in new tricks.
Yeah, speak to me a bit more about that. It's obviously an area that you've developed in structural biology. There were some terrific slides in your presentation that showed how you've pieced things together in that direction. What sort of applications do we see in that field of study?
Well, the first thing I want to do is acknowledge Patrick Sexton, who I did at the end of my presentation, because Patrick, we've been together for over 20-something years here.
Yeah.
Patrick was really the one that really drove us to move into the structural biology space. So yes, it has been a collaboration. So we started with crystallography, which is far more well established. But and we had a collaboration with Brian Kabilka, who's a Nobel laureate who won the Nobel Prize in 2012 for his work with structural biology GPCRs. But we realized that cryo-EM really is the way to go in terms of being able to capture more natural active states as well, more dynamic states. And you tend to not need to engineer the proteins as much as you do in the crystallography space. So that's why we've moved into cryo-EM, because we think the closer you can get to a native-ish state of the protein, especially under, if the next step is trying to understand these things in under disease conditions, the better.
Yeah, fantastic. Tell me, Arthur, can your work be applied to the treatment of animals as well?
Absolutely. I mean, it's interesting. We have a friend and colleague who works in the veterinary space, and one of the areas that they've been very much interested in is also in the application of nanobodies to treating disease. But the animal receptors, of course, have allosteric sites. So in fact, there's no reason you can't do that. Intriguingly, though, because of species variability often between humans and animals, you could be using an animal model to discover an allosteric drug for a human that won't translate into the human space. But yes, if you're after the animal, you can do it right there and then.
Look, thank you ever so much. We're out of time, I'm afraid. Congratulations on your election to the Fellowship of the Australian Academy of Science, and we look forward to welcoming you in Canberra in the near future.
Thank you. I definitely want to see you all soon.
Thank you. Now we'll take a short break and be back at 2:15 Australian Eastern Daylight Time for session 4. Welcome back to Science at the Shine Dome 2021, where today we have been hearing from the newly elected fellows to the Australian Academy of Science over the years 2020 and 2021. Don't forget, you can ask our fellows questions throughout the event by using the panel on the right of your screen. Now back to the presentations. To introduce our next fellow, please welcome back Professor Bob Graham.
Thank you. Our next presentation is from Professor Ryan Lister. Ryan is a genome biologist who has made major advances in our understanding of the epigenome, the molecular code superimposed upon the genome that can regulate the readout of the underlying genetic information. His work is driving advances that will provide benefits to agriculture, human health, and medicine. We will now hear from Ryan.
Good morning, good afternoon, or good evening, everyone. I'd like to begin by saying thank you to the Academy. It really is a great honour to be elected. Also, thank you to everyone who supported me along the way, starting with my nominators and supporters for election. I've been really lucky to have had many great opportunities and amazing mentors along the way, in particular Joe Ecker, Jim Willen, and Harvey Millar. I've also been fortunate to work with so many talented people, great students, early career researchers, and fantastic colleagues. And it's really only through such collaborative work that I've been able to achieve any of this. And finally, thanks to my ever supportive family who have underpinned it all. So the process that's really fascinated me in my career is how the enormous diversity of cell and organism forms and functions, the hundreds of distinct cell types, arise from the control and selective use of the information that's encoded within a genome. How is this information selectively accessed and correctly read in order to orchestrate the hugely complex processes of life? Besides a few exceptions, the cells of our body contain essentially the same genome sequence, yet we start out as a single cell and through the process of development become an organism with tens of trillions of cells and many hundreds of distinct cell types. And it's the same throughout multicellular life in diverse species. So the question is, how is the information in the genome differentially and dynamically used to give rise to this vast complexity? Well, it turns out that in addition to the A, C's, T's, and G's of the DNA sequence of the genome, there are additional layers of molecular information that are added to the genome or to the proteins around which the DNA is wrapped. And these can instruct the cell on how to use the underlying information that's encoded in the DNA sequence. These other layers of information are collectively called the epigenome, and essentially they act as minuscule chemical signposts that can signal to the cellular machinery to use a particular underlying sequence of DNA and produce RNA from it, or to wrap up this other region of the genome tightly and archive it away and not use it. And really key chemical modification, one layer of the epigenome, is called DNA methylation, which is simply a CH3 methyl group that's added to cytosine bases, the Cs, within the genome sequence. And this is what a lot of my work has focused on studying. When I began working in this area, a major outstanding question was, where exactly are these chemical modifications located in the genome? Which cytosines have them and which don't? Up until then, it hadn't been possible to comprehensively map where all these modifications were throughout a genome. So we took advantage of a chemical conversion process and a revolutionary new DNA sequencing technology that was emerging at the time and developed new techniques to identify exactly where all the cytosine bases that had DNA methylation added to them were located throughout an entire genome, as well as sequencing the RNA output of the genome. We first did this in the model plant Arabidopsis, shown here, because of its really compact genome, small genome, and great genetic tools. And this provided us with the first integrated base resolution maps of gene expression and DNA methylation sites and small RNAs that can direct these chemical modifications. Following on from this, in plants, we've been able to study various aspects of DNA methylation, such as how it can change in response to stress conditions, the patterns that we see in different cell types, and even how these parasitic transposon elements that can jump around the genome can leave lasting footprints in the form of methylation changes. With these mapping tools in place, we then set out to take on a bigger challenge, which was mapping the sites of DNA methylation, which we otherwise refer to as the methylome, throughout the human genome, which is 25 times larger than the Arabidopsis genome. We first compared two very different cell types, these highly specialized fibroblasts and embryonic stem cells, and generated for both of these the first comprehensive single base resolution maps of DNA methylation throughout the genome, throughout the human genome, revealing huge differences in the DNA methylation patterns between these different cell types. So in this work, we were comparing cells that represented very different cellular states seen in human development. These non-specialized embryonic stem cells that have the potential to give rise to all the cells of a mature organism, versus highly specialized and differentiated fibroblast cells. But we wondered, what would we see when we used cell reprogramming technologies to basically wind the clock backwards, to reverse this process and reprogram a specialized cell type back into a stem cell-like state? So we set about making the first high-resolution maps of the DNA methylome for human-induced pluripotent stem cells, which are cells that very closely resemble embryonic stem cells and that can be made by forcing the reactivation, the expression of just 4 genes. Overall, what we saw was that this reprogramming process almost completely reset the specialized fibroblast epigenome patterns back to a state that looked like an embryonic stem cell. However, when we looked closer, we saw that there was actually a molecular memory lingering in the induced pluripotent stem cell genome, a memory of the specialized fibroblast cells that they began as. And this memory was in the form of DNA methylation patterns that in some areas of the genome are like the patterns that are seen in the starting specialized fibroblast cells. But does this matter? Well, there's now evidence that this epigenetic memory may affect the plasticity of induced pluripotent stem cells and perhaps bias them towards turning into cell types that are similar to their starting cell type. So we can now use this information and our understanding of how the epigenome is naturally reset to come up with methods that might erase this epigenetic memory and make more predictable pluripotent stem cells. But overall, what this demonstrates is how these epigenome modifications added to the DNA can act as forms of molecular memory. Given these dramatic changes that we observed in the epigenome during development that happen as cells divide many times and become specialized, we thought an interesting question next would be how malleable the epigenome was in cells that live a very long time but no longer divide, the neurons of our brain. So we set about mapping the DNA methylome in the human prefrontal cortex from gestation through to adulthood and discovered really extensive reconfiguration of the epigenome during this process. What we saw was a non-canonical form of DNA methylation that rapidly appeared and accumulated in the neuronal genome after birth, in particular at the same time as the processes of synaptogenesis in childhood. And we also saw widespread changes in DNA methylation at the regulatory switch regions of the genome that are important for controlling gene expression. So this non-canonical form of DNA methylation we found is now thought to play important roles in controlling gene expression in normal brain development and in the experience-dependent plasticity in gene activity control in the brain. Again, this demonstrates the critical role of these chemical modifications in the genome and controlling cell functions. So the ability to comprehensively map the epigenome provides these new insights into this dynamic process of controlling information and its usage in the human genome. However, humans are, of course, extremely challenging or impossible to study in various contexts, such as embryogenesis. So to look at this process, what we instead did was to take advantage of a comparative genomics approach where we compared different vertebrate species, studying epigenome reconfiguration at the most conserved point of vertebrate development, which is called the phylotypic period, where embryos from many diverse vertebrates look really similar, which happens when the vertebrate body plan is being established during embryogenesis. So through epigenome mapping throughout this process in mice and fish and frogs, we discovered an ancient conserved process shared between these species that are separated by hundreds of millions of years of evolution, whereby the genome regulatory regions that control essential genes linked to development lose their DNA methylation marks, and which is associated with these regulatory switches being used to control gene activity. So this likely also happens within us as we develop during embryogenesis. We also noticed that these regulatory switches seem to remain unmethylated throughout the body, even in adult cells. So the cells of the adult body contain a memory of the critical events that happened way back in early development in the form of these DNA methylation modifications. So from this, it's clear that these chemical modifications on our genome are really dynamic and important for our development. And they can tell us simultaneously about both the current state of a cell, but also the past history that the cell or its predecessors in development experienced. Also, we know that DNA methylation marks show many changes in disease states. So in a way, we can use these DNA methylation sites as the equivalent of tens of millions of molecular antennae that cover our genome and that are sensitive to the local regulatory events that take place. And in some instances can store that information about those events in a really stable manner that can be read out through DNA methylation mapping approaches. Mapping DNA methylation in cell-free circulating DNA in blood is now a leading approach for both detecting the presence of cancer cells in the body through their distinctly perturbed DNA methylation patterns, as well as identifying their cell type of origin through the unique memory of their originating cell type that's stored in their DNA methylome. And finally, we don't have to just read these patterns, but through using genome editing technologies such as CRISPR, we and others have now shown that it's now possible to start specifically and precisely editing DNA methylation patterns in the genome, deliberately where we want, providing new ways to explore and use these molecular markers on our genome. So with that, thank you again to all my colleagues and supporters along the way that made all this possible. And thank you very much once again to the Academy.
Now, Professor Ryan Lister joins us live. Congratulations, Ryan, and thank you for your terrific presentation.
Thanks very much.
Ryan, we have some questions for you. I want to start right at the beginning. Who or what inspired you to pursue a career in this area?
Yeah, so actually my interest in this, I guess, started some time ago, back when I was a teenager, and actually, it was from a fiction book reading Jurassic Park, the Michael Crichton book. And I guess just being given an evocative example of how the information that was in a fictional setting able to achieve the de-extinction of those creatures just contained the entire information, patterns, instructions for building these complex organisms. And then just from then on set me forth in thinking about the complexity of what lies within this repository of information in the genome and the incredibly complex processes that must regulate that so that, for example, starting as a single cell, we can unfold into this enormously complex organism. And really just then trying to understand and better understand, unlock some of those puzzles of how that happens.
It's a fascinating and very fast-moving area. Let me ask you, what are the potential impacts of your evolving work on the epigenome in understanding neuroplasticity associated with neurodegenerative diseases such as Alzheimer's, Parkinson's, and Huntington's disease?
Yeah, that's a good question. So what's really exciting nowadays is that the tools that allow us to characterize these states, the patterns in the epigenome, are really advancing at a rapid pace. So a few years ago, we were only able to look at these in mixtures of many thousands or millions of cells. But now what is occurring is that we're able to move these profiling and mapping activities to be able to look at individual single cells and to be able to look at thousands and so millions of individual cells in parallel. So that then gives us the ability to take a really complex tissue, such as the brain, and to look at these patterns and how they change throughout processes, whether that's development or throughout aging, and to focus on individual cell types. So not just a complex mix of many different cells before, but focus on each individual type of cell and study how these molecular patterns are changing, whether we're seeing specific alterations in certain cell types, in various pathways that may be relevant to altered functionality of those cells. I think studying this in neurons is particularly interesting as essentially we have in the neurons of our brain those that basically we had in our early development shortly after birth. So we have then these cells that are living for decades, but at the same time remain plastic, respond to particular changing conditions, form new memories. And so a great interest then is how these molecular marks may allow a level of information that can provide a fixation of particular molecular states, but also then reversibility so that the cell could enter a period of flexibility and remodeling at a later state. So I think that there's a great opportunity now with the emerging technologies to really explore how these patterns are changing both in normal developmental or aging processes, but also how they might be altered in disease disorder contexts such as neurodegeneration.
Thank you. You spoke about epigenetic memory. Experimentally, how would you go about removing that?
Yeah, so there are multiple ways. So the epigenetic memory that we have been studying in the past has been in the process of cell reprogramming, where you can take this revolutionary technology where you can take a specialized cell, reactivate just a small handful of genes, and turn a subset of those cells into the equivalent of embryonic stem cell states, highly plastic, and they retain this memory. So one option is to try to enhance that reprogramming process, either altering different sets of genes that are turned on or altering the conditions under which the cells are reprogrammed, for example, media conditions, and to explore that. Another opportunity is to, and I think one of the really exciting opportunities is to use these genome editing technologies, such as CRISPR technologies, which are we and others have been repurposing so that you can go into a cell and then very specifically modify and engineer these epigenetic states wherever we want in the genome. So this is not changing the genetic sequence, but in a very specific place we want changing the epigenome marks. And so these tools are under development, in various forms are working now, and groups around the world are working on making these work much more effectively. So in that way, we'd be able to go in and engineer these layers of information that we've had no ability to alter before. So this gives us the hope of being able to both correct aberrant states, but also in terms of biotechnology or cellular technologies, I think opens up a whole new area of work where we can explore how this information can be used to perhaps reach cell states that have never before existed, by being able to unlock certain patterns of activity in cells that normally don't occur due to the organisms behaving in a way that has been sculpted by evolution. But with these tools in hand, we'd be able to start to change regulatory processes in cells that hadn't been possible before.
Ryan, one last question from our audience. Peter Price asks, what are the relative roles of histone epigenomics and DNA methylation?
That's a good question, and one which there's been a great body of work on, and which requires a lot more work as well. So it's an incredibly complex system. There are more than 100 known chemical modifications of the histone proteins around which DNA is wrapped and DNA itself. And there are a variety of known interactions between these modifications of various histone marks and modifications influencing DNA methylation, which can influence where the nucleosomes are positioned, how the cell interprets these, so we understand a variety of these different interactions, the proteins that can read these and how they can alter how the cell's interpreting the underlying information. But there are a lot of these interactions that we don't yet understand. So we are hoping that having some of these tools in hand that allow us to very precisely go and alter one of these marks in isolation, or indeed multiple of them simultaneously, will allow us to have a greater ability to mechanistically dissect what these particular modifications are doing and how they affect other marks on the DNA, the organization of the genome with other proteins, and how the cell is reading and utilizing that information.
Thank you, Ryan. That's all we have time for. Congratulations again on becoming a Fellow of the Australian Academy of Science.
Thanks very much.
Please now welcome back Professor Bob Graham to introduce our next Fellow.
Thank you. Next, we will hear from Professor Mark Dawson. Mark focuses on the biological mechanisms by which epigenetic regulators facilitate the induction, maintenance, and progression of cancer. He has made several paradigm-shifting discoveries into epigenetic mechanisms of cancer, stem cell maintenance, resistance to anti-cancer therapies, and regulation of anti-cancer immune surveillance. His research has directly contributed to the development of several first-in-class epigenetic therapies that have been studied in clinical trials across at least 20 countries. Now we'll hear from Mark.
I am both humbled and honoured to have been elected into the Academy, and it is now my great pleasure to present a short overview of my area of work. We're surrounded everywhere and every day by examples of the importance of epigenetics. However, few examples illustrate this better than a child's wonder at witnessing the metamorphosis of a caterpillar into a butterfly. These two distinct physical states emerge from the same genome. And similarly, I have for many years been driven by an insatiable curiosity to understand how a single genome could give rise to complex multicellular organisms. As a hematologist, much of my career has been dedicated to understanding how access to our DNA template is regulated in blood cells and how this process drives the cell fate decisions that gives rise to this rich diversity and function. In fact, it is these insights that have been critical in understanding how epigenetic regulation is corrupted in cancer. As humans, if we were to stretch out our DNA content end to end, we would be able to walk to the sun and back nearly 100 times over. This is because unnucleated cells contain about 2 meters of DNA, which is packaged into a nucleus that is no more than 10 micrometers wide. This amazing field is accomplished because DNA is packaged as chromatin. In fact, chromatin provides the context in which all our DNA templated processes are regulated. Broadly speaking, there are 2 major disparate chromatin states. Euchromatin, which is an open configuration, which is often transcriptionally active, and heterochromatin, which is more tightly packaged and condensed, which is relatively transcriptionally silent. The basic functional unit of chromatin is the nucleosome, which contains a histone optima around which DNA is wrapped. Over 2 decades of research in chromatin biology has now demonstrated that all of the components of the nucleosome are covalently modified, and these chromatin modifications are integral to the dynamic regulation of chromatin. We've learned a great deal about the major protagonists and basic principles involved in epigenetic regulation. Broadly speaking, the enzymes that lay down histone or DNA modifications are termed epigenetic writers, and they include enzymes such as histone methyltransferases, acetyltransferases, and kinases. Chromatin modifications are dynamic, and the enzymes that have evolved to remove these modifications are called epigenetic erasers, and they include enzymes such as histone demethylases, deacetylases, and phosphatases. One of the most important discoveries that we have made is that these chromatin modifications serve as an instructive platform for the recruitment of proteins that have specialized domains that recognize these modifications. These epigenetic readers then recruit in larger macromolecular complexes that modify and remodel the chromatin template, leading to the processes of transcription, DNA repair, and DNA replication. The advent of next-generation sequencing technologies really brought to light that many developmental disorders and human diseases are underpinned by mutations in epigenetic regulators. This is most pervasive in cancer, where proteins that regulate the processes and function of chromatin are some of the most common abnormalities seen in cancer. In fact, the dynamic regulation of the epigenome has now raised the prospect that we can manipulate it for therapeutic gain. And over the remainder of this talk, I would like to highlight the work that we have contributed to this to this field so far. My path to chromatin biology started during my PhD when I was studying the role of signaling kinases in cancer. Whilst it had been appreciated that signaling kinases activate transcription factors to mediate gene expression changes in response to exogenous stimuli such as cytokines, here we describe how these kinases can directly alter chromatin structure and function. We described the first tyrosine phosphorylation on a core histone and showed how this chromatin modification facilitates various processes, including transcription. The Wellcome Trust then invested in me as their inaugural Wellcome Bait Fellow to pursue my ambition to therapeutically target chromatin. And working closely with GlaxoSmithKline, we developed and characterized one of the first examples of a drug that targets a protein-protein interaction at the chromatin template. These drugs against an epigenetic reader domain called a bromodomain within the BET family of transcriptional regulators provided some of the first proof of principle studies that disrupting the way epigenetic readers are localized to chromatin to facilitate oncogenic gene expression was a viable therapeutic approach in cancer. It, along with other studies, really laid the foundation for over 40 ongoing clinical trials in over 20 different countries. At the time that we had started our work on the BET proteins, they were known to be key transcriptional regulators. But immediately following our seminal publications, the field exploded and continues to grow exponentially. However, despite our initial progress in targeting these transcriptional regulators, many fundamental biological questions remained. For instance, why are there 3 BET proteins that have structural and functional similarities, but are ubiquitously expressed in all cells? Why do each of these BET proteins have 2 bromodomains that are virtually identical? And is there any biological rationale to target these domains individually? We set to work to answer some of these key questions, and using chemical biology approaches, we developed tools to monitor the effects of our drugs at subcellular resolution, including at the chromatin interface. We were also able to monitor the distribution of these drugs in both malignant and normal cells in various different tissues. And this work really led us to realize that it is the first bromodomain of the BET protein BRD4 that was required to tether it to chromatin to maintain gene expression. And whilst this gave us the impetus to develop domain-selective inhibitors, we were still unclear about the precise role of BRD2 and 3 in all cells, and what did the second bromodomain do? Again, working with GSK, we now developed domain-selective inhibitors, and with these tools, we were able to show that it is the first bromodomain of BRD4 that is required for the maintenance of established gene expression programs in all cells, and it is the second domain of all BET proteins, BRD2, 3, and 4, that is needed for the induction of gene expression. This also brought about a new therapeutic opportunity to divorce the targeting of established pathological programs, such as in cancer, from the induction of pathological gene expression, as in inflammation. In studying how resistance to BET inhibitors developed, we also established some other important paradigms in our field. As our work demonstrated, that resistance to anti-cancer therapies can emerge without any further mutations in our cancer genome. Instead, this non-genetic resistance can be mediated by transcriptional processes that evade the therapeutic challenge. It was also one of the first examples demonstrating that these adaptive processes occur in cancer stem cells. In fact, we now know that in cancers like acute leukemia, these non-genetic mechanisms of resistance account for up to 40% of therapeutic resistance seen. And this is an increasing area of research in a broad range of other cancers. We've gone on to illustrate two different types of non-genetic resistance. The first case, the therapy selects for cells with the right transcriptional program. And in the second case, a cell undergoes an active rewiring of the transcriptional program. Both these processes are amenable to therapeutic manipulation, and these manuscripts have spawned new clinical trials to overcome these adaptive mechanisms. We've also now honed in to try and uncover unique epigenetic dependencies in cancer stem cells. This work helped uncover a dependency in leukemia stem cells on the histone acetyltransferase HBO1. And this work, along with others, have helped facilitate the commercialization and clinical development of first-in-class drugs against this family of epigenetic writers that are now entering clinical trials. Finally, in my lab, we have also been investigating how we might be able to leverage epigenetic regulation to increase anti-cancer immune surveillance. Using unbiased genetic screens, we have recently uncovered master regulators of key immune checkpoint molecules and therapeutic targets that increase antigen presentation and increase anti-cancer immunity. Whilst I'm incredibly grateful to be the recipient of this wonderful recognition, it is really important for me to emphasize that this is a team effort, underpinned by a number of funding agencies who have invested in our scientific vision. It is particularly important for me to recognize Sarah Jane Dawson, my wife, who runs an independent cancer genomics laboratory and is my main scientific collaborator. It is true to say that very little of what we have achieved would have been possible without her. I want to end there and am now happy to take any questions that may arise.
Thank you ever so much, Mark. We are now we're delighted to have Mark with us live. Congratulations on your election to the Australian Academy of Science.
Thank you, Anna-Maria.
If the audience members would like to ask any questions, please pop them into the chat on the right-hand side of your screen. Mark, can I commence by asking, it's been a tremendous time of discovery over your career. What's been the most surprising or impactful insight gained from your research in the field of epigenetics to date?
I guess, I guess there's a number of ways to answer that. The first thing I'd say is, when I first started in the field of epigenetics, its importance, I guess, to cancer biology was largely underappreciated. That the major fundamental insights came from sequencing the cancer genome, because it rapidly became apparent that actually nearly all cancers, the most prevalent genetic mutations are mutations in proteins that regulate access to our DNA template, that regulate the processes of DNA repair, DNA replication, and transcription. And really that, has harnessed not only, the attention of the academic, of various academics around the world, but also industry and pharma. And we've also got on board a number of clinicians who previously would have been largely unaware of epigenetic gene regulation, to now become, far more integrated and interested in understanding what this might bring to the table in terms of therapeutic gain.
Thank you. A question from Andrew Roberts. Which diseases do you think will first be transformed through targeting epigenetic changes?
I think the short answer to that is both. I think there's opportunities in terms of autoimmune diseases as well as in terms of cancers themselves. In terms of cancers, I think certainly potentially the more genetically simple cancers, pediatric cancers that are often driven by either single chromosomal translocations or single gene mutations. They clearly, despite having almost an identical cancer genome, have very different cellular behavior. And, if we go back to the original description of what epigenetics is, it's the heritable state of cell behavior that cannot be accounted for by the genome. And we really see this play out in very many pediatric cancers. So I think understanding that process in greater resolution, that will give us the best therapeutic opportunity there. And understanding the contribution of inflammatory gene expression may give us, an opportunity to intervene in autoimmune diseases as well.
And finally, how far along is your research in epigenetic regulation for anti-cancer immune surveillance?
Yeah, I'd say really at its infancy. It's been rapidly enabled by genome editing technologies. The ability to go and ask an unbiased question by surveying the whole genome through an experiment has really lent itself to make discoveries of key regulators that we now need to go and work out the precise molecular mechanism and find ways to therapeutically enable those pathways. So I think really at its infancy, but certainly a major area of interest and ongoing focus in the lab, and hopefully good things to come in the years ahead.
We look forward to seeing what's yet to come. Mark, congratulations again on your election to the Fellowship of the Australian Academy of Science. Thank you for being with us today.
Thank you, Anna-Marie.
Now to introduce the next presentation, please welcome Academy President, Professor John Chown.
Thank you. Next, we'll hear from the Honourable Dr. Annabelle Bennett. Annabelle is an internationally recognised leader in intellectual property patent law, specialising in scientific, medical and information technology issues. As a federal court judge, also with a PhD in biochemistry, she delivered nearly 500 decisions concerning intellectual property, including patent matters, trademarks and copyright. Now it's a pleasure to hear from Annabelle.
I thought that I would talk about the thread of science in my life. When I was informed that I was to be admitted as a Fellow of the Academy, I was at the same time deeply honoured and somewhat quizzical. Why me? My immediate reaction was that there was a mistake, as I was not recognised as a scientist. However, I managed to overcome that concern and simply enjoy the moment. Well, so far, so good. However, it has come, as they say, home to roost when I was told that I had to present this talk about myself as a scientist. What to do? I have no research to describe, no litany of publications in reputable journals, no great contribution to research and the advancement of science that immediately sprang to mind. Then, when I thought a little more about it, albeit in panic mode, I realised that science has been a constant and ever-present during my life and in my career. It may have commenced when I was only 8 years old and my father started an industrial chemical company. I used to spend my holidays at the factory, in the office, doing filing and putting invoices and receipts into envelopes and reading all of those chemical names. It can well be said that chemistry supported me through school, adolescence and university. I was the first year of the so-called Wyndham scheme that introduced a 6th year into high school. Without going into the details, I was told about the Sydney University matriculation exam just before Christmas of my 5th year, which would otherwise be my final year. So I spent the holidays studying the leaving certificate subjects and did the exam. I couldn't do any science because I didn't have the right prac books, but I did have to do other different subjects, of course. So I did all the new English texts, different modern history, a third of the maths 1 syllabus, 2/3 of the maths 2 syllabus, because I had been doing combined maths, and French. I passed, but what now? My father persuaded me out of law because he thought that being a woman would make it too hard to succeed. Medicine seemed too long, and I couldn't see what I would do with an arts degree. So I studied science almost by default. I was fortunate that my lack of science in the matriculation exam did not preclude entry to the faculty. Well, to cut the story short, I did science, and then I did histology honours in mitochondrial populations, and then a PhD in the Department of Veterinary Physiology. I will always be grateful to Professor Cliff Emmons and to my supervisor, Dr. Ian White, for my success in my research, which was on phospholipids in sperm, looking at the integrity of sperm membranes, and it was fascinating. The trouble was that I wasn't really cut out to be a researcher. The thrills didn't seem to occur often enough, and I was still very attracted to law. So while I did my postdoc, I studied law part-time, but I had to stop my research when I had my first child at the end of that first year. My second child was born 19 months later, and I had a third subsequently. I finished my law degree when my first two were 2 and 3 years old, and I went straight to the bar. There was no such thing as science law in those days. Indeed, people looked at me with astonishment when I said that I was changing from that one field to the other. The most common observations were, well, you've wasted 8 years of your life. And my favourite was, well, how can you do both science and law? Don't they think differently? The concept of logic as a common denominator was apparently unheard of. Furthermore, in those days, you were not allowed to advertise, and it was not considered the done thing in Sydney to use the title doctor. So my science simmered in the background, unutilised for my first dozen years at the bar. Then there was an article in the Good Weekend about women barristers, as we were still a somewhat unusual phenomenon. And it mentioned that I had a science degree, and the doors opened. A patent attorney saw the article and briefed me in a patent case. I still remember it was all inorganic chemistry and nomenclature, but I was, or perhaps more importantly seemed to be, the most qualified person at the bar to deal with it. I was technically qualified in a sea of those who were almost universally boasting of their lack of knowledge and skill in such matters. That combination of my science and law led to my entry into the world of intellectual property, endlessly fascinating both scientifically and legally, and increasingly a field that went from being a somewhat esoteric backwater to one of substantial commercial importance. So why was the science so helpful? The answer is not straightforward. First, my scientific knowledge helped me to understand some cases more easily. An example was the newly emerging area in Australian litigation of molecular biology. The first case in Australia in this area was about recombinant erythropoietin, and I appeared for the patentee. Well, at least I knew about transcription and translation, although obviously I needed to be upskilled for the purposes of the case. After all, I had to cross-examine experts on the subject. This need to be brought up to speed occurred regularly, as knowledge had advanced a long way from my undergraduate days. I remember, for example, a case which demonstrated to me how much knowledge had developed from my studies in immunology at university to what was happening there and then. But what was the key and what was the fun? Well, the key was that, of course, I didn't know everything, but having studied science and having been exposed to scientific methods, I wasn't frightened. I wasn't scared of the technology. It can fairly be said that some lawyers and judges were afraid of exposing their lack of understanding of some of the complex technologies that were developing. I just asked the questions. Perhaps the fact that I had a degree in science gave me a veneer of protection from seeming to be feeling stupid. The other wonderful aspect was that I was paid to have private tutorials with some of the world's leading scientists, right up to Nobel Prize winners. And how good is that? But that was not all. While I continued doing cases as a barrister and as a judge, specialising in patent cases, I learned about subjects ranging from pharmaceutical developments and new treatments for a vast range of maladies to telecommunication standards and the intricacies of smartphones. I've also engaged with judges from all over the world who are themselves working on these cases. What is interesting is that there is still only a very small minority in the law who have dual qualifications. For many years, I was the only technically qualified judge on the federal court in Australia. Apart from my work on the court, my science background has most likely been a factor in opening other doors and exposing me to other experiences. I was able to feel part of the research world during my time on the council and then as Pro-Chancellor of the ANU. I also met and worked with the wonderful Ian Chubb. I chaired the NHMRC for a term and was actually thankful that I did not have the skill to determine the outcome of grant applications, as it was hard to see some only miss out by a whisker, and not for want of value and importance, but for want of sufficient funds. I was asked to be an arbitrator with the Court of Arbitration for Sport, most likely because the number of cases there involved chemical substances of one sort or another. I've become Chancellor of Bond University. I chair ANSTO, where I'm learning about a whole different area of science, engineering and research. And I'm now regretting that I dropped physics at the end of first year, but even that basic level of teaching has helped. I sit on the board of the Garvan Institute of Medical Research, and so I'm still exposed to advances in medical research, while I keep one eye open to the intellectual property ramifications. So if anything, science has re-emerged in its various forms, once again to become the basis of so much that I do. I suppose that I'm an example of the benefits of an education in science. I can't imagine where my life would have been had I chosen to do another course of study. And this was before talk of STEM or of girls in STEM, but was probably the product of the approach when I was at school that science was compulsory and important for now and for the future for everyone. They were the days of Harry Messel and Julia Sumner Miller. We knew that it was exciting and that it would provide a stimulating and rewarding career, even if you went on to another career. That background was of inestimable value, directly and indirectly. It's crucially important, in my view, to bring back that enthusiasm and excitement to the schools. Somehow it is so often not adequately conveyed. I remember the headmistress of my daughter's school say to the mothers, "None of you would boast of being illiterate. I do not see why you boast of being innumerate, and I will not have you pass that on to your daughters." It should not have to be said. There should be an understanding that STEM subjects are of basic importance, and as those basic skills are acquired, a whole world of potential opens, the potential to boldly go where no one has gone before. The Academy Fellows cover the spectrum of the sciences and demonstrate the contribution that can and is made from the knowledge acquired, the advances made, and the skill applied. I am deeply honoured to have the opportunity to join them and hopefully to learn more. Thank you so much.
Thank you ever so much, Annabelle. We are delighted now to have with us the Honourable Dr. Annabelle Bennett live with us. Congratulations, Annabelle, and thank you for that presentation.
Thanks, Anna-Marie. It's an honour to be here, quite truthfully.
Annabelle, you say you weren't cut out to be a researcher, a realisation that many researchers come to a point in their careers. It happens quite often, actually. Indeed, I put myself in that category. What's your advice to those that may be facing that scenario in terms of finding a new career direction?
I always think that when you're looking for a new career direction, you take what you've got and you realise that nothing is wasted. So it's just an opportunity to expand what you can do. If don't feel as if you're not utilising what you're doing at the moment in the next one, because you can't predict what will happen. I mean, I never thought at the beginning that I would use the science the way I did. It came in later, and then you just draw it back from your knowledge and you put it to work, and it really does help. So just be open and just grab an opportunity. If in doubt, say yes. If someone offers you something, say yes, because you can always change your mind if you find it doesn't work for you later.
Excellent advice. Very little in life is irreversible, isn't it? And a science degree is never wasted.
In many ways, it's like a basic. It's as much a basic degree as many others are. It gives you so much knowledge, direct and indirect, what you appreciate and what you don't appreciate. That's what I think anyway.
Yeah, it allows us all to interrogate data and information, something that is critically important nowadays, that we have a sea of information around us. We have a question from one of our fellows, Professor Francis Afarovich, who writes, "Congratulations, Annabelle. A few of my PhD students have gone on to careers in IP. Have you noticed any advantages or disadvantages to have this background and then go into law?"
Well, if you're going into intellectual property, it's not just an advantage, it's almost crucial. I mean, I think that
while not everybody in IP comes from a science background, there is no doubt that it is an enormous advantage. I mean, you just understand research, you understand what goes on in research, you understand the steps, and of course you understand the technologies, and you're not afraid, as I've said during my presentation, you're not afraid to ask. So, I mean, IP is a wonderful area. It's becoming more and more important, and it really is exciting. And whatever science you've got, you can build on that because you absorb other areas of science. I mean, I didn't know anything about telecommunications. You know, I was a biological side of things. So, but it doesn't matter because in the end, you have that mindset where you know that you can absorb it. And if it's told to you properly, you can pick it up and you can learn it.
Annabelle, you noted that indeed there's still a minority in law who have dual science law qualifications. What do you think can be done to better embed scientific methods more fully into the legal system?
I think it's to give, I think one of the things is to give students at universities the opportunity to go across disciplines. I think that's an enormous advantage so that, you know, students who are doing science get exposed to others, and perhaps students in other faculties can do one or two science subjects on their way through and then get a basic understanding of certain areas. So I think that not having silos to the degree that we do, to the extent that we can expand on those, I think we'll have a great advantage across all aspects, because one never knows where that science will come in. I mean, as you say, even data analytics and so many other aspects of it. I mean, now we're going into green technology. So anybody in business who's got a basic science understanding is already one step ahead in many aspects, I think.
It does provide a very basic literacy that assists in understanding a range of areas. You're absolutely right. And one final question, if I may. How do we bring greater enthusiasm for studying STEM subjects at school? We've all noted how critically important it is and that perhaps it's not seen or perceived as it once was. What can we do collectively and individually?
Science is exciting. Science, I don't know if science is being made exciting. Fortunately, I spent some time on the board of Questacon, and I looked at the young people there who were teaching at Questacon, and they call them inspirators. And that's what they are. If we can get the kids young enough, I mean, doing experiments and seeing these things happen, I think if you can get that enthusiasm up at a young age, even pre-high school, that stays with the kids. And I don't think that leaves you. And I think that understanding that there's excitement in science is what it's all about. And it makes people want to learn more. I can't imagine I can't think of another one, but it's there. It doesn't need to be thought through because it's actually readily available.
Thank you ever so much, Annabelle. I'm afraid that's all we have time for. We are absolutely thrilled that you've been elected a Fellow of the Australian Academy of Science. Congratulations again.
Thank you, and that's very kind of you.
Now, please welcome back Professor John Shine to introduce our next Fellow.
Thank you. Our next presentation is from Dr. Andrew Cuthbertson. Andrew has made outstanding contributions to Australian science as an entrepreneur, specifically as Chief Scientific Officer and Executive Director of CSL Limited, an Australian-based global pharmaceutical company specialising in therapeutic biological products for human health. It's now a great pleasure to hear from Andrew.
Greetings, and I'm sorry we can't be together in person. May I start by saying how thrilled I am to be elected and to join heroes and friends like Gus Nossel, Don Metcalf, John Shine, Dick Denton, Doug Hilton, Graham Mitchell, Dave Vo, and many others at the Academy. I'd like to say a few words about my passion, translational medical research. I have huge respect for colleagues who pursue pure scientific research in any scientific discipline. But perhaps because I did medicine first, I actually thought I wanted to be an eye doctor. I enrolled to do a PhD at the Walter and Eliza Hall Institute in transplantation immunology while starting my eye surgery training, and I never went back to clinical practice. I decided I wanted to try to make it as a medical research scientist. And I must say this was regarded at the time in the 1980s as a rather unusual decision. So instead of finishing my clinical training, I did a postdoc at the Howard Florey Institute and then went and did basic eye research at the National Institutes of Health in Bethesda in the US. And some of you may recall that in 1984, there were 3 back-to-back articles, not letters, but articles published in Nature describing the cloning, expression and characterization of human factor VIII, which is the protein that is defective in subjects with haemophilia A. And this extraordinary work was done by a team at a biotech company called Genentech in San Francisco. And this absolutely captured my imagination. And with the ultimate help of a great friend, Hugh Nile, a wonderful Australian scientist who was at Genentech at the time, I ended up working there through the mid-1990s. I had 2 projects. The first one was to try to develop a factor VIII gene therapy for treating haemophilia A. This failed completely. But I also had an eye research project using an anti-VEGF-A recombinant FAB fragment to treat the eye disease, age-related macular degeneration. And a big team ended up working on this project, and it ultimately became Lucentis, the first biotech treatment for AMD. So that was a lot of fun. And I was enjoying immensely working at Genentech, living in San Francisco. In 1996, I was visited by Ian Gust and Brian McNamee, who were then at the recently privatised and floated CSL in Melbourne. And their pitch was that I could do, what I was doing at Genentech in the US, but do it back in Australia, where I came from. And that's what I should do. And I must say this suggestion was a bit delusional on both sides, because on the one hand, CSL's R&D budget at the time was about $30 million a year, a bit less than Genentech's. But to be fair, on my side, I'd only ever managed at the most 2 lab assistants at that point. And so as a people manager, I was, shall we say, rather untried. But to jump forward, I've been at CSL for 24 years from that time. Our R&D budget's about $1.4 billion a year. We've got 1,600 scientists around the world in R&D doing translational research and biotech new product development. I stepped down from being head of R&D and chief scientist at CSL in 2018. But with COVID, I was asked to stay on and help with CSL's COVID response, particularly the vaccine response. So we've had a very intense, I'm sure we've all had a very intense last 20 months. I've had a very intense 20 months working with the research community, industry partners and government to try to accelerate onshore vaccine development and large-scale vaccine manufacturing. We had a wonderful but ultimately unsuccessful collaboration with Paul Young and the University of Queensland team and CSIRO, and an excellent and fortunately successful collaboration with the AstraZeneca Oxford team that resulted in onshore manufacturing by CSL of the Oxford AstraZeneca vaccine. And I would say, let's be positive. I think the combination of public health response and widespread vaccination will lead Australia out of the difficult isolation we've all experienced for the last 20 months. So I'm pleased to have been able to do quite a lot of translating over the past 30 years. The best part of it all has been to build a team of wonderful global scientific colleagues who really can develop new biotech medicines and vaccines from beginning to end, and launch and large-scale manufacturing and provision to people around the world. And I think it's summed up to a degree by two significant investments CSL is making right now that speak to medical research translation. They're both under construction and they are buildings, but the buildings really signify the people and the work that will take place in them. First, there's the new global headquarters of CSL. Those of you who've visited our 103-year-old site in North Parkville in Melbourne know it's museum-like. It's wonderful, but it's quite isolated. And we are building a large modern headquarters in the Parkville Medical Research Precinct near University of Melbourne, Royal Melbourne Hospital, WEHI, Howard Florey, etc. And it's not an office, well, it's half an office building, but the really important half are the bioengineering labs where we will make candidate medicines for clinical trials with our academic colleagues in, in Parkville, in Melbourne, in Australia, around the world. So that's exciting. And the other construction that's taking place, which also signifies translation, is a large-scale state-of-the-art plant next to Melbourne Airport, which will manufacture novel vaccines for Australia and for export. And that construction of that plant has started. So I think you can see the links between these investments and the fact that we will have engagement with our academic colleagues at the research level and development level. And ultimately, if they're successful, we'll have the ability to do large-scale industrial manufacturing of these new biotech products and vaccines for Australia and for the world. I should have more time for the University of Melbourne Council, the Grattan Institute, which is a research-driven public policy institute that I'm passionate about, and CSIRO, the Centre for Eye Research Australia, which takes me back to my first love of vision research. And of course, the Academy of Science and the Academy of Technological Science and Engineering and the Academy of Health and Medical Science, to all of which I'd like to contribute. Thank you.
Please now welcome with us live Dr. Andrew Cuthbertson. Congratulations, Andrew, and thank you for your presentation. It was so inspiring.
Thank you, Anna Maria. Great to see you. Great to see you.
You mentioned of being an untried manager when you first came to CSL. Since then, what have you learned about what makes a successful research team and how to build productive collaborations and a very successful multinational company? I mean, you spoke about going from $30 million to $1.4 billion in R&D investment and 1,600 scientists. That's far from a couple of people who you managed in a lab.
Yes, indeed. And there was risk, of course, that I would mess it up. And but to try to answer your question, Anna Maria, I think it's that particularly large-scale science or biotech product development is a very human activity, and and individuals can't do it, and it's really a matter of having a whole team of people with specialised skills who respect one another, with structure, but not so much structure that it becomes boring. Common goals, being really clear about common goals timeframes in which you're trying to do things. I think all of those come into it, but I would go back to, it sounds very simple, and I guess ultimately it is, but it's about having highly trained, good people and respecting them and leading them towards a common important goal.
Well, they've certainly benefited from your leadership, and as a nation, I think it's the globe, we thank you. What are some of the key challenges for translating research outcomes into a product that can be manufactured at scale?
Yes, well, it's a good question, and I think sometimes we, as a community, probably, I hope, value scientific discovery, and for us, say in the biotech industry, that means our scientific colleagues coming up with what we call a product candidate. And that might be 1% of the output of a great lab in a university or medical research institute. It's really identifying that high-risk candidate and then bringing all of the skilled people together to progress through preclinical animal studies, manufacturing at quality and scale appropriate to clinical trials, and then ultimately being able to manufacture millions and millions of doses of the product. And all of those skills are very specialised and require a lot of training. But I think recognising them and recognising those stages, putting those teams in place are probably the biggest challenge. But of course, ultimately, you would say you have to have a beautiful, novel candidate in the first place, and those candidates come from academic, curiosity-driven research.
Yeah, thank you, Andrew. Having onshore manufacturing of vaccines has been a game changer for Australia and for our region. What do you think that sort of capability and the need to have ongoing capability in that regard will mean for the future of the biotech industry?
Well, I'm passionate about the fact that onshore manufacturing is both economically and societally important for Australia. I would just say that my bias, and I think I'm right in this, is that the onshore manufacturing should be established for ongoing use, ongoing commercial use, and for example, to have a high-quality plant that can produce 200 million doses, clearly more than Australia requires, but to export those doses every year to the rest of the world, but have that plant available onshore in a time of crisis is the right conceptual approach. And that's certainly the approach that CSL are taking with the plant that's being built at the airport, in Melbourne at the moment. It is a plant, it's a commercial plant for export, but it's on Australian soil and therefore obviously available for Australia and our region in the time of a crisis.
Yeah, more than 100 years ago, investment in CSL delivered for Australia and has ever since in spades. I have no doubt that the future directions you're talking about will continue to do that and set us up for the future in ways that we possibly can't imagine at the moment. I'm sure I speak on behalf of many in thanking you for your leadership of this tremendous multinational company, for seeing it go from success to success. And we wish you well, now that you'll have a bit of extra time to dedicate, and very much look forward to continuing to involve you in the work of the Australian Academy of Science. I know you've been very critical for us in providing evidence and understanding so that we have been able to prepare advice for government throughout the pandemic. So thank you for that. Andrew, I'll leave it there. And again, congratulations on being elected a Fellow of the Australian Academy of Science.
My pleasure, Anna Maria, and look forward to it. Thank you.
Thank you. To introduce the next presentation, please welcome back Professor John Chown.
Thank you. Next, we'll hear from Australia's Chief Scientist, Dr. Cathy Foley. Cathy has made outstanding contributions to Australian science through promoting and supporting science across the wider community, including as the first dedicated Chief Scientist of CSIRO. She has also made distinguished contributions to high-temperature superconductors and to the development of field-deployable superconducting devices that locate valuable mineral deposits. It's now a pleasure to hand over to Cathy's presentation.
Hello, I'm Cathy Foley. It's really great to be here today. I want to talk to you about my work, which I undertook when I was at CSIRO, as a way of sharing some of the work I did that led to me being nominated and then elected to the Academy of Science. After completing my PhD at Macquarie University in nitride semiconductors, I moved to CSIRO as a research fellow. And I undertook work originally on glassy metals, but then there was a big breakthrough in physics that was to change my life. And I want to talk about that now because it led to me working on and creating a new Josephson junction, which led to many applications. This new breakthrough was on the front page of the New York Times, which is not a normal thing for physics in particular. And this was the discovery of high temperature superconductivity. So why is this so exciting? Well, when a material that is a superconductor is cooled to a low enough temperature, usually close to absolute zero, electricity can flow through that material with no resistance to its flow. This means that we can have things such as MRI machines, which we all commonly know today. But it also allows us to create a structure called a Josephson junction, which allows, when currents go through it, to have a nonlinear response to an applied magnetic field. This enables us to create a structure that can be used in a device, and it can detect magnetic fields that are so small we could pick them up from your brain or your heart. That's about 1,000 millionth of the Earth's magnetic field. This discovery was exciting because superconductivity is usually a material that is cooled with liquid helium, but this allowed superconductivity to happen at what was called high temperatures, which is liquid nitrogen, minus 200 degrees. But this made it easier to use and to be able to be used in a range of new applications. But there's always a catch. When you go up in temperature, often the material becomes more difficult. In this case, the material is a ceramic, yttrium, barium, copper oxide. And this is a tricky material. Not only is it a ceramic, but it also meant that it had lots of dislocations and difficulties in the material. It's highly anisotropic. The coherence length to make that structure I was talking about, the Josephson junction, needs to be about 1 nanometer to 2 nanometers. And of course, anyone who's worked with materials knows if one of the components is a gas, it makes it very hard to make quality material. So after developing a way of doing that, my next step was to see how can I create this Josephson junction, this 1 nanometer dislocation, so that we could create a structure so when currents moved across that had that response to a magnetic field. And the way to do this was to engineer the grain boundaries of the materials so that we could realign them to create a dislocation so that you ended up with that structure which was needed. The approach which I took was to create a step edge in a magnesium oxide substrate. You can see on the left-hand side micrograph there, and a strip of material going over the edge. And in the center, you can see a transmission electron micrograph that the blue arrow is showing the position of the grain boundary there, which is nice and clean and highly uniform, which meant that the structure had low noise, was easily implemented, and we patented this. It's been so successful that now that the patent time has run out, it's been copied by many different research groups around the world. We took this material and structure and put it into a device called a superconducting quantum interference device. The one we worked with initially was an RF squid, which could be inductively coupled into an electronic circuit. And what you see here is some of the early devices there and how it was put into a packaging in order to be a module that could go into a system. The application which was most successful was one for mineral exploration. We were challenged to see if we could develop the system that would allow us to detect large, high-value conductors deeply buried under the Earth's crust, particularly in Australia where there's a conducting overburden, which often camouflages the presence of these special ore bodies. The approach was to have a loop of material with a current passing through it on the surface and turning the current off and on. This induced an eddy current in the conductor deep underground. And during the off period, we were able to measure using a magnetic sensor, which we created, that decay. And from that, we were able to pull out the information that gave depth and size of the ore body. Over the 10 or so years we took to develop the system, you can see here the different versions as we went through our development program to creating what we call LandTem, which was commercialised under licence by Outer Rim Exploration, and they set up a startup company called Outer Rim Developments. You can see there from the experimental system on the left-hand side through various versions to the commercial system that's available and used today around the world. The reason why this was so useful compared to other techniques such as induction coils was that the squid system could measure directly the decayed magnetic field that came from a deeply buried ore body. And this is an example of a nickel sulfide deposit, which is a red ore body shown here. And the squid versus the coil was able to show at those deep levels, which is at the bottom of the measurement graph shown here, that there was quite a significant response. And this meant that we were able to have evidence of how well this system worked. So over the years, we went from going with our early field trials to being able to support the delineation and the discovery of a range of different mines. And this is probably the most successful one, which is a Cannington silver mine, $2 billion worth of silver, which we helped to delineate. LandTem now has a life of its own, and the only way I know what it's up to is when I go hunting, which I did in preparation for this talk, looking at the Australian Stock Exchange announcements. This is one just from October this year, where you can see I've circled where they use the CSIRO LandTem system to be able to delineate and detect a deeply buried ore body, which looks like it's a very interesting one indeed. Since that time, we've developed a whole range of other devices using that same grain boundary technology, such as high temperature direct coupled squids, as well as KA band, high speed, long range communication devices based on mixers and low noise amplifiers, which are completely packaged in cryo-cooled systems. And finally, another example is terahertz imaging, where we're able to see through a bit like Superman vision by operating at those very high frequencies. Now, some of the focus is looking at how can we make these devices even more sensitive by having many of them all linked together. And what you see here is at the top, a single DC squid, which has the normal sinusoidal response to a magnetic field. And underneath is an array of different size squids, which when they respond to a magnetic field, they have constructive interference that leads to a single peak in the center, which allows you to set the device up to be very sensitive to magnetic fields. So for me, that single grain boundary has led to a whole swathe of fantastic physics and applications, and meant that I've had the most wonderful research experience and career at CSIRO, starting from being a young girl at school, wondering about whether I could ever be a scientist, to actually living that dream, doing great work with a wonderful team and achieving many things. And so it was a great honour when last year I was announced as Australia's Chief Scientist, which I hope that I can deliver it again to the country in different ways. And of course, I should point out that I was not working on my own. I was part of an amazing team that still exists at CSIRO, and I want to thank them and thank the Academy of Science for the great opportunity of being elected to being part of the Academy. Thank you very much.
We are now delighted to have with us Australia's Chief Scientist, Dr. Cathy Foley. Congratulations, Cathy, and thank you so much for joining us today.
Hi, Anna Maria. It's great to be here.
we have some questions for you, and we remind our audience, if you have questions, pop them into the Q&A panel on the right of your screen. You note that having superconductors on the cover of the New York Times is not a normal thing for physics. How do you think we can make scientific innovations a more regular cover story?
I wish it was every day, and it still happens sometimes. But one of the biggest issues is for us researchers, we get so caught up with the detail and the actual specifics of the science that for most people, they don't really care about that. They actually want to know what is the clickbait, I guess, or the cover story that actually says, well, why bother? So what? And I think when we get better at explaining why many of us will, really commit our lives to doing pretty hard stuff, working long hours and often going through the highs and lows, which we all know research has. The reason why we do that is because we all want to not only just create new knowledge, but also see how it can be used to the betterment of society. And I think part of the time we forget that we need to think about the bigger picture and the vision of what it might lead to, as opposed to actually worrying about the science. So that's probably one way we can do better. I guess the other one is also maybe seeing that we have more people seeing career pathways into not just science research, but a plethora of different areas that can get a lot of value from having people with STEM education actually working in their different profession.
How have other research groups used the step-edge Josephson junction, and have any of these new uses surprised you?
That's a good question. Not really. I suppose there's a whole range of applications because magnetic sensing can be used from anything such as being an antenna through to being a qubit in a quantum computer, through to being a memory device. It can be something which works in a classical way or using quantum physics. So up until now, I guess the thing that surprises me is how few applications have actually got to the point where we use them every day. And part of the reason for that is you have to cool them. But the advent of these really compact cryocoolers is, I think, going to change that. And I don't think it's that far away from us actually buying our next desktop computer anyway, which actually will have a little mini cryocooler in it, because it will allow electronic devices, not just superconducting-based ones, but also other sort of more traditional semiconductors to operate it faster, better, more efficiently. So I think that's going to be the next step. But of course, we always have to, when we see something become commercialized, you've got to have a technology people want. You've got to have it with a price range that people will pay, and also a business model and a way of engaging a people interface or a user interface that makes it easy to use. And I think too often technologies get caught up along the way where we actually don't realize we've got to have the full package. It's not just the science, it's the other stuff, the science plus, that's really important.
Thank you. A question now from our audience. A warm congratulations, Kathy. I noted in your last slide there were 4 women out of 22 people in your group photo. How is CSIRO encouraging more women in the physical sciences?
Okay, well, actually, it was great noticing that, because that's a really old photo. That's really terrible, because I haven't actually worked with the team for a while. The team actually probably has about 50% women now, and it's something where over the time, we've actually, I've seen the increase in the number of women, but I should point out this particular research field of superconducting electronics has about 5% women around the world. In applied physics in general, you find that there's very few women. It's something which I think is terrible, and we're not embracing that full human potential. So what to do about it? In the case of CSIRO, I guess you'd have to refer to what they're doing at the moment. They're going through the SAGE program. Larry Marshall is the CEO, as a real champion of change, and has really pushed hard, leading from the top. He's got 50% or more women on his board. I think he's got more than 50% women on his executive team now. He's working his way through trying to make sure he's got gender equity with the next level, and he's working his way down. And it's quite interesting to see that middle management of research program leader, which has traditionally been very highly male-oriented, is actually building up to having more and more women, which is great to see. But part of the problem we have, though, in actual particular research fields such as physics, electronics, engineering in general, and particularly IT, the numbers of women are still far too low. And there's a whole lot of reasons for that. One of it is we don't necessarily encourage girls enough at school that this is something they can do. And there's a whole lot of work that's being thought of how we might address that, but we actually need to do it. The second is that sometimes the environment in the university system where girls might start doing an engineering degree or a physics or IT actually drop out because they don't feel welcome. So I think we haven't got the culture right. And then the other one, of course, is if there's few people that they can see who are role models, mentors, then it makes it even harder again. So, that leaky pipeline is real. It's one where, we've got a lot of plumbing to do to really fix it. And I think we have steps forward and steps back. And I think one of the things that bothers me is we make great headway and think, great, it's all under control. And then we think we don't need to worry about it anymore. As soon as we take our foot off the accelerator of trying to encourage more women into STEM, we find that it pulls back as soon as we take the focus off it. So it's something where we know it's a tricky problem. It's deeply and socially within the way we operate as a social community. And I think it's going to be something which still requires a lot of focus and effort.
Thank you for such a comprehensive answer. And you're absolutely right. It is the entire pipeline that we need to address, and it's a relentless effort. And just when we think we've got it right for gender equity, we need to look at all of the other forms of diversity as well. So Absolutely. There's lots for us to do. One final question, Kathy. What's your most audacious goal for your time as Australia's Chief Scientist?
Probably I want to see if I can achieve open access of research literature. I actually think that, as people may have heard, research is our superpower, and we've seen it over and over again and that's not just science research, it's going right through the humanities, through social science, through health and medical research. So across all the different academies, I think we cover it all. And it's all hidden, or not all, but too much of it is hidden behind a paywall. And it's the way we have the whole structure of the business of scientific publishing is a mixture of part of our peer review process, which we need to have the trust of the community and also amongst ourselves that the work that is published is something we can rely on. So publishers play a really critical part. But we also have huge contributions from others, from the research funding that is the content that is published, through to the referees who review the material, through to the editors and editorial boards. And I think that we've got a couple of things such as metrics as to what looks good is driving some behaviours which isn't making it an equitable system, through to the fact that imagine if every person in the world, but let's start with Australia, could read research literature. It would mean government policy, industry, general public, professionals, teachers, citizen scientists, as well as all researchers, because no one library is able to access all the research literature in the current business model. Mm-hmm. Imagine what would happen if we could read it all for all that wonderful work. And I think it's interesting, up until recently, people have been saying, why would anyone want to access research literature if they're not part of research, academia or research organisations? When I explain how it could be used, I've had a lot of aha moments, and I guess that's my big goal. It's quite a massive change. It's a very disruptive change, both for publishing and for Australia, with the way we organise ourselves with research. So I guess that's my big audacious goal, Anna-Maria.
Audacious indeed, and all strength to you. May you achieve that in Australia and influence the global landscape as well. Kathy, that's all we have time for today. Congratulations on being elected a Fellow of the Australian Academy of Science, and we look forward to having you in the Shine Dome to sign the historic charter book in the fullness of time as soon as we can all come together again. Thank you.
That'd be marvellous. Thank you, Anna-Maria.
Now we'll take a short break before wrapping up the day with session 5, starting at 3:45 p.m. Australian Eastern Daylight Time. See you back here soon. Welcome back to our final session of Science at the Shine Dome 2021. We have had 2 days listening to the most inspiring presentations from the newly elected fellows of the Australian Academy of Science. Thank you for all your questions. Please keep them coming. Now to introduce our next fellow, please welcome back Professor John Shine.
Thank you. Our next presentation is from Professor Lynn Beasley. Lynn is a distinguished neuroscientist and the 2015 West Australian of the Year, who has made a major contribution to the promotion and direction of science in Australia. She has been a champion of gender diversity and science in the classroom, in addition to shaping science policy. Lynn is also well known for promoting science and technology in the media. Lynn was elected in 2019, but was unable to join us in that year. So we're delighted to hear from her now.
Hello, everyone. Lynn Beasley, thrilled and honoured to be a Fellow of the Australian Academy of Science and coming to you from Perth, Western Australia on Noongar land. Let me tell you a little bit about my story in science. It starts at a place with a rather inauspicious name of Gravesend in the United Kingdom. I attended the local grammar school, and the most exciting thing we ever did was to visit Darwin's house. I even looked through his microscope, and at that point, I was going to be a biologist. It's a decision I have never regretted. I came from a family where no one would ever have dreamt of going to university, so it was a particular thrill when I cracked a spot at Oxford, and then up to Edinburgh for a PhD on a subject that didn't even have a name. We know it well now, it's neuroscience. And in particular, we studied neuroplasticity, a real no-go area at that time, and we did it in frogs. Now you can see at the tadpole stage, the eyes face to the side. But as it transforms into a frog, the eyes come to occupy a much more frontal and upward-looking direction. As it does that, the brain connections have to keep adjusting so that the input from the two eyes overlap to give you binocularity. We could see that pathway and see the changes happening in it. It was pretty exciting stuff. The first real evidence of a brain circuit changing in response to new experience, and we could see where the anatomical changes were happening. But from Edinburgh, I moved to Western Australia. Best move I could have made, because I wanted to continue my studies of the visual system and to see what was happening, particularly during recovery, if we could induce any. Now, at this point, I have to tell you a little bit about the developing brain. As it forms, two outpouchings develop, and they become the neural retina, the light-sensitive sheet that lines the back of the eye. At that point, the primitive nerve cells in the eye send nerve fibers all the way to the brain, find the visual center within it, and go to exactly the right address in there to set up vision. Amazingly, if you take an adult fish or frog and you cut the optic nerve, from that cut end, the nerve fibers go back to the brain, find the visual center in exactly the right spot again, and the animal can see within a month. Do that in a mammal, including in human, and the nerves frustratingly don't regenerate. Many groups, including ours, have tried to find triggers to get them to regrow, but frustratingly, they peter out after a very short distance, about 1 millimeter, which isn't far along even the optic pathway of a rat, which is about 10 millimeters long. We have yet to discover how to get long nerve fiber connections reestablished in the mammalian central nervous system. But while we were doing this, others were discovering the signal molecules in the eye and brain that allowed those initial nerve fibers to grow out and find their destination. So we wanted to know if they were rolled out during regeneration, back to the humble fish and frogs. We found that as the nerve regenerated, indeed, those signals in the eye and brain were switched on again. They stayed until the connections were made, and then they faded away. Intriguingly, when we looked in the mammals, although the nerve didn't regrow, the signals were switched on again. So if ever we can get nerves to regrow, the signals will be there to tell them where to go. Continuing this whole theme of the comparative aspects of optic nerve regeneration, we were very interested to look at a group that hadn't been analysed before. We knew a fair bit about fish and frogs and birds and mammals, but we hadn't looked at the reptiles. We decided to look at lizards. So we took adult lizards and we cut one optic nerve. We left the other side intact so the animal could see fine. And we waited and waited for the operated side to recover, and a year later, nothing had happened. But being thorough, we thought we'd better check what was going on inside. And to our great surprise, the nerve had grown in proliferation, but it had grown to the right and the wrong centers in the brain. The connections were very weak and unsynchronized, and the brain had built up a resistance to this information it really didn't want to know about. How could we restore function? Regeneration had happened, but function wasn't restored. Neuroplasticity to the rescue. Just 2 sessions a week of showing that operated side, the favorite food, and after 5 months, one small snap by a lizard was one very excited group of scientists. The nerves had now regenerated only to the right places. The signals were strong and synchronized, and the brain was listening to them. It's exactly the principles that we use to try and get recovery after brain damage, such as strokes. Neuroplasticity tells us that training is essential during the process of recovery. Now, towards the end of my career, I set up a second research group, and this was looking at colour vision, particularly in Australian animals, because we had read in textbooks that marsupials can't see red, only green and blue, like the placental mammals, except us primates. We thought that was really unlikely, seeing how bright the Australian environment is, and we were right. Australian marsupials that we studied can see red right through to blue, and they can see a colour we can't see. They see ultraviolet, so they see an even more colourful world than we do. So while I'm busy doing research and I'm on lots of committees nationally and internationally, I get a phone call to ask me if I would consider becoming the chief scientist of Western Australia. Fantastic. To become a facilitator between industry, government, academia and the community, an advocate for science, an advisor to government. Wow. Because science is integral to every part of our lives, and I was there to help promote it. Travelled internationally, promoting how we've done some great science. Barry Marshall and Robin Warren there receiving their Nobel Prizes for discovering how to cure gastric ulcers, right through to the automated mine sites where we lead the world. I was part of the negotiating team for the Square Kilometre Array, the largest science project on the planet of radio telescopes, now being built in the Murchison of Western Australia and the Currie Desert in South Africa. I brought FAMELab, one of the largest international science communication competitions, to Australia. We haven't won, we've come second, always been in the final, but we'll get there. And we're the only one of the 30 or so countries to have instigated a schools program too. I'm very proud of that. At a national level, I work with science lab technicians to get better science at a practical level in our schools, particularly with an online facility assist for our school teachers and for the technicians. Locally, I promoted citizen science. We need extra people helping with science, Dolphin Watch being a major example. Over 1,000 people have been trained and gathered data, even down to the level of individual dolphins in our 3 largest systems. Developing stem cells, really important for me and for everyone on the planet. And at a practical level, we set up a Microscopes in Schools program where over half of all the schools in Western Australia, the primary schools, 5- and 6-year students receive their own microscope. Working with Rotary, we've spread far beyond, even sending microscopes as far as Afghanistan. With a pretty positive feedback. Jaden, thank you for saying that we rock. I have a particular interest in young people on the autism spectrum. They're the Steve Jobs, they're the Einsteins of the future, yet they experience real problems through our educational and employment system. So we've set up an amazing program to mentor them in schools through TAFE and uni and job placements, over 200 now in job placements across Western Australia and in some of the biggest companies along our terrace. You can see them there. Gosh, looking back to me as a kid walking on the prom in Gravesend, never would have imagined I'd have met, let alone worked with Nobel laureates. Couldn't believe I would work with the second man to walk on the moon, Dr. Buzz Aldrin, and totally amazed that a fossil shark and a deep sea sponge are named after me. Thank you, science. Thank you, the Academy, for this honour, and thank you, everyone, for listening.
Welcome and congratulations, Lynn. It was a phenomenal presentation, and my warmest congratulations to you on your election to the Australian Academy of Science.
Thank you. It's a complete honour.
Lynn, you came from a family where no one would have imagined you going to university. What got you there? Was sheer determination? And what's your message for young people at school who may be considering pursuing a career in STEM, but may be daunted by the prospect of university and beyond?
Thank you. It's a great question. I think the first answer is to say, believe in yourself. You know, I think the imposter syndrome is very easy to say everyone else is going to be smarter than you. But I love the quote, it's not how smart you are, it's how you're smart, because everybody's smart at something. And luckily for me, it was science. Ignited, as I said in my talk, by going to Darwin's house. And I suspect that the specimens I looked at in his microscope were actually from Western Australia because they were carnivorous plants. But having a family that really backed me, that, you know, we weren't flush with money, but nevertheless, my family saw that as the most important thing for me. And I think it's working with the young people, working with their teachers and working with their families to give them that confidence to move to TAFE and to university. I'm a huge supporter of TAFE, as you know, and that can be a very good step for young people who don't quite have the confidence yet to move on to a university career. So seeing those steps, mentoring, hugely important. As you know, I visit a lot of schools and I work with organisations that have excursions into schools and great programs on the web as well, because COVID has done us, although a great disservice in many ways, has introduced some new programs that are really helping.
Thank you. Lynn, do you still keep a foot in the door in terms of research and particularly around long nerve fibre connections in the mammalian models, having conquered the nerves in lizards and frogs? Is that something you keep in touch with? I mean, every time I see you, you have so many interests and are helping so many people. I wonder where you find the time. But how about your previous research or perhaps current research?
No, I don't put a white coat on anymore. I support much more indirectly by helping those along their careers, by mentoring, by helping writing references, for opening doors and seeing connections. So, for example, last night I attended the Innovator of the Year Awards in Western Australia, and there were so many from the pharma system that came up, and several had grown through my lab, with a little bit of support from me along the way, but really their own talents. So that's the way I try and help. I think it's not positively dangerous with a white coat on anymore.
It's certainly not indirect. I know you have mentored and assisted many, many people directly, and you continue to. So thank you for that. What's been your biggest lesson when it comes to providing scientific advice to decision makers and politicians in particular to ensure it's heard and incorporated into policy?
I think they need to trust you. It's a very good question. And I think the expression, it's academic, you have to overcome that. They have to know that you'll be practical, that you'll look for solutions that will work. And you have to make sure that you offer them solutions to issues that they are addressing, as well as introducing new ones that they might not have been aware of. So it really is a dialogue. And I always used the analogy of a table, 4 legs on a table, industry, government, academe, community. A table stable with 4 legs or 3 legs, but less than that, it doesn't work. And looking to see how that trio or quartet can all work together, I think is extremely important. And then knowing it knowing how, as Kathy said previously, Kathy Foley, how to present it to the public in a way that they will see, hey, that's a good idea, that's going to help in some way. So I think that was the essence. And it's an art form that you learn, and you're always learning, but it's something that allows you an enormous privilege to have input to decision makers, and look also at the level of delivery of programs. So yeah, conversations, understanding both sides and taking it from there.
A final question for you. Has citizen science delivered any or revealing findings in your Dolphin Watch work? And what difference has it made to have 1,000 trained people sourcing data?
It's been enormous. And yes, it has, because, for example, we've discovered where and when animals are in different parts of the systems. We know, for example, where the mothers nurture their calves. Now, knowing that is really important, because we can set in place regulations to prevent jet skis and the like occupying them. So yes, it has a direct result. The other thing we have done is that we've put along the edges of our rivers, estuaries and our coasts special bins. So it's awful when a dolphin's entangled, and by having places to remind fishermen to put those lines away and not just leave them in the water. Some still do, but we're getting a real culture shift there. And we're in schools through Junior Dolphin Watch. So we reach out to schools across the state and run programs with them, because I firmly believe if we get young people involved, they talk to the generations above them, and that's how you get a change in culture.
Absolutely. Thank you, Lynn, and congratulations not only on becoming a Fellow of the Australian Academy of Science, but on your recent election as the incoming Secretary for Public Awareness and Education commencing next May. You already make a tremendous contribution to the Academy, not just bringing your expertise, but you're an absolute breath of fresh air in all that you do. And we very much look forward to continuing to work with you in the coming months and years. Thank you, Lynn.
I appreciate it.
To introduce the next presentation, please welcome back President Professor John Shine.
Thank you. We'll now hear from Dr. Simon Poole. Simon is an outstanding researcher and entrepreneur in photonics. He was a member of the team which developed the erbium-doped fibre amplifier. That's now a ubiquitous part of the optical fibre networks that underpin the global internet. He has also led the establishment of an export competitive photonics industry in Australia. So now over to Simon's presentation.
Good afternoon, ladies and gentlemen. It's my great privilege today to be able to talk to you a little bit about my areas of specific interest. That's a little bit different from most of the presentations that you're hearing today, which tend to be more technical. I'm going to be talking about something more commercial and in terms of the commercialization of photonics in Australia. First of all, let's clarify what we mean by photonics. Photonics is the science and application of light. So it includes optics, it includes all aspects of optics and their application to real-world problems. It's important to realize that photonics and the photonics industry is something like a $1 trillion per annum business. This isn't a small industry. This underpins so much of what we do as a society, from the internet through to solar power, through to the automation that we use in manufacturing is based on photonics. In Australia, we have a significant photonics industry here. We have an annual manufactured output of around about $4.3 billion worth of photonics products, which is similar in size to the dairy industry. Most people are very much aware of the dairy industry here. Very few people so familiar with photonics. We employ nearly 10,000 people in over 450 companies, and about 50% or so of the activity in photonics industry in Australia is related to optical communications. And this is an area that I'm going to talk about extensively during the remainder of my presentation, after I've talked a bit about a technology called the erbium-doped doped fibre amplifier, which was really my scientific contributions to the field. Now, as you know, optical fibres are basically the backbone of the internet. They carry the internet globally, but because of losses in the fibre, particularly Rayleigh scattering, the signal loses power as it travels along the fibre. So we need something that can amplify that signal in the optical domain rather than electrically. And that's what the erbium doped fibre amplifiers are. They're optical amplifiers. They've got a huge bandwidth, up to 10 terahertz, and they amplify the signal every 40 to 80 kilometers it travels down the fiber. And you can see over on the right there is an example of an EDFA module containing electronics and optics that form the EDFA to control it. And that's used in both indoor plant, like that 19-inch rack you see on the left, and also in outdoor EDFAs, the sort you'd see at the top of a telegraph pole for a hybrid fiber coax system. The EDFA is based around this erbium doped fiber, which is made by adding very precise quantities of erbium ions to the core of the optical fiber. And that formed the basis of my PhD at Southampton back in the mid-80s, where I was lucky enough to be one of the inventors of this EDFA technology. And there are now tens of millions of EDFAs in optical communications networks around the world, in Australia, globally, and under the sea, as we will talk about later. So that was prior to my arrival in Australia. After coming into Australia, I was involved in the research side, and in particular, building out technology companies coming out of research here in Australia. And the first one I want to talk about is a company called Index, which worked in the area of optical submarine cables. And this just shows a quick map of all of the submarine cables in the world. These are cables that connect with optical fibers the various countries and continents, and which carry nearly the entirety of the internet traffic between countries and continents. You can see there's a vast number of these out there. They all use EDFA technology to amplify the signals, and they all use an optical transmission technology called wavelength division multiplexing, WDM, which uses multiple colors or wavelengths of light, up to 100 different colors, each carrying 100 gigabits per second of data. And Index was founded in 1995 as a spin out from the Australian Photonics CRC. I left that and took some of the students with me to start Index as a manufacturer of fiber Bragg gratings, or FBGs for the shorthand. These are a type of optical filter used in wavelength division multiplexed optical communication systems. And the sort of things we're making shown at the bottom there as a packaged FBG that was used in many of our customer systems. The thing that made Index successful was our ability to package the gratings, and this was recognized when we were asked to bid to provide fiber gratings for the CMIWI3 cable system, which, when it was commissioned in 1998, was the world's longest undersea optical fiber system. It's 39,000 kilometers long, linking Asia, the Middle East and Europe, and also down to Australia, where it carried the video signals from the Sydney Olympics. Putting something in an undersea cable requires very high reliability. And for us, that translated into demonstrating one failure per 1.4 million years. That's the mean time between failure of 1.4 million years. We managed to demonstrate that to the customer, Fujitsu, and we won the manufacturing contract for that CMIWI3 cable. And on the back of that, built Index to be the largest manufacturer of fiber gratings for subsea fiber systems, with over $100 million of revenue per year and 300 staff by 2000. I left Index in 2000 and then started another company, which is now called Finisar, looking at a technology and application called wavelength selective switching. So a wavelength selective switch is something that enables the interchange of optical signals, that is the switching of them without it converting to the electrical domain or having to demultiplex them. It's the equivalent of a highway interchange, or with on and off ramps to take the signals on and off the fiber. And these wavelength selective switches make wavelength division multiplexed optical networks much more efficient and much more powerful. So in terms of where we went with Finisar, as I said, I was a co-founder of that with Steve Friskin in Sydney in 2001. And we invented and developed the core technology which is used in all WSS these days, which is a thing called liquid crystalline silicon switching. And that's shown on the right there, the LCOS chip circled there. It's a technology that came out of the display TV industry in the late 1990s, and we repurposed this for telecommunications. Finisar Australia has now shipped over 150,000 wavelength selective switches. There's an example of one there. And when I left Finisar in 2018, they were the leading global manufacturer of wavelength selective switches, 40% global market share, 100% of all the manufacturing which we've done in Sydney has been for export, with over $1.5 billion of cumulative exports for wavelength selective switches. And they're still running. There's still 250 people working there at the factory in Sydney, developing and making these wavelength selective switches. After Finisar, which I left in 2018, went to work with a company called Silight, which we had founded back in 2013 and which was starting to move forward in the development of the way that we look at measuring and inspecting people's eyes. Eyes are incredibly important, as I'm sure you know, but there are so many issues with them. Something like a third of the population of the planet have some form of vision impairment, about half of which is untreated. There is a problem as you age, and particularly in some poorer countries, where cataracts cause blindness, which is a very easily treatable problem. It takes 20 minutes to cure, but is a major impairment to people's quality of life. Similarly, myopia, short-sightedness, doesn't sound like a big problem. You wear glasses, but it actually causes many more problems later in life, and it's really turned into an epidemic of myopia, both in Southeast Asia in particular, but also increasingly in Europe and the US. And Silight addresses the markets for measuring and quantifying these diseases. Our technology, called HPOCT, or hyperparallel OCT, has been under development now for the last 8 years. It's a new way of imaging and measuring the properties of the eye, and creates those sort of beautiful 3D images that you can see at the bottom of there, which is basically the front half of someone's eye, showing the cornea and the iris, the pupil, the lens, and so on. We're about to launch that product, coming very soon. So really watch this space. It's very different from the telecoms work I've been doing previously, but it's equally fascinating. And again, manufacturing and developing this in Australia, this time down in Melbourne. Yeah, thank you very much for your attention.
We're now delighted to have with us Dr. Simon Poole. Congratulations, Simon, and thank you for joining us today.
It's great to be here. Very, very much appreciated.
You've helped invent the EDFA back in 1986. And as you say, tens of millions of these are now in use around the world. Has that technology changed much since its invention?
The core of the technology has not really changed at all. What has changed is the manufacturing scale that is now done at. The techniques that we developed for manufacturing the fibres back in the mid-80s are still very much the ones that are used now. They're refined, and 30 years of development it improves things, but it's still basically the same technology for the fibre manufacturing, the same laser pumping technology that existed before, but an awful lot of refining of that technology and taking it through that commercial learning curve.
Thank you. You said you needed to demonstrate an incredibly low failure rate to win the CME-WE3 contract. How competitive was the market in 1988, and how does one guarantee a 1 in 1.4 million day failure rate?
It comes at the time we were really the only people who could get close to that. It was an amazing coincidence of ability. Two of the students that I took with me to start the company had done their PhDs in fibre Bragg gratings, and one of them was in the physicochemical properties of the grating, how stable it was and what changes over time. So we were able to show from basic science of the glass chemistry that the changes that we made to the glass to create these gratings would be stable. And then on top of that, we had to put in place a mechanical testing regime, which came from one of the other people that came into the company at the start, to prove that the fibres themselves wouldn't break during that process. That was a known technology which we applied into the grating and added on the sort of underpinning physical chemistry of it. In the end, it came down to a theoretical proving. We couldn't demonstrate actually that we could get 1.4 million years. We're somewhere now at around about a million device years in service, and we still haven't had a failure. So we're really close now to proving that 1.4 million years.
That's extraordinary. You're clearly a scientist, inventor, entrepreneur. At heart, it's in your DNA. I want to talk a bit about Finisar and your work there. So the wavelength selector switch technology came from the television industry in the 90s. Yes. What inspired Finisar to repurpose this technology? And is that the trick, to be able to find these sort of things and think about them differently? Talk me through some of that invention and innovation.
Sure. Well, when we started Finisar, we sat down and we approached this from a market perspective. What is going to be required by the telecoms industry in 5 to 10 years from now? Because it takes that long, somewhere between 5 and 7 years, to take a new technology from concept through to a real product. So it was sitting down, working out what we needed to do. And we said, okay, we identified a number of things. Which ones can we do in Australia with the capital that we could raise around it? And that identified wavelength switching as the outcome. We then went through a whole bunch of different technologies, trying out different ideas. So we started the company really with a market idea, not a technology, which is sort of quite different from where so many people think of a tech company. But it's really how a company has to run. It has to have a market problem to solve. And the way that we came to work on the LCOS was we've been visiting a company in the US, a couple of companies in the US, where we were thinking of acquiring technology, other technologies from them, and using that technology and bringing that technology back into Australia. And on the plane on the way back, Steve had basically the brainwave that said, we can do this better if we look at a totally different approach, having looked at pretty much everything that everybody else had already done. There was this brainwave, we could do this better. And at the time, there wasn't an LCOS industry. The display projection TV industry had died once flat screen TVs came out, but there was this technology sitting there that if you rethought how you could do it, you could use that. And yes, repurposing other second-hand technologies, nothing wrong with that, finding new applications for it. It's great. It greatly reduces the risk and it greatly reduces the development cycle needed to get there.
And a final question for you, Simon. What advice would you give a researcher who's looking to commercialise their research?
I would say spend time at conferences, not listening to papers, but going to the trade shows and talking to people in the industry and finding out what the problems are that need to be solved. That's how you make a company that's successful. You solve a problem. You don't create a company that's successful by loving a technology. That's the wrong approach. Falling in love with the technology is how to lose money and lose a company very quickly.
Very sound advice. Thank you ever so much, Simon. And again, congratulations on your election to the Australian Academy of Science.
Thank you very much.
Now, please welcome back Professor John Shine to introduce our next fellow.
Thank you. Our next presentation is from Professor Margaret Shield. Margaret, a distinguished chemist, is a leader in the scientific and university sector. A trailblazer for women in science, she's Vice-Chancellor and President of the Queensland University of Technology and Chair of the Queensland Museum Network. She's a strong advocate for science and scientists, especially early career scientists, women researchers, and outstanding scientific mentors. She's played a pivotal role in national science policy and has consistently sought to ensure that Australian scientists have access to world-class scientific infrastructure and funding schemes. We'll now hear from Margaret.
Hello.
Thank you for this opportunity to talk briefly about the science that I did early in my career, and then some of the initiatives that I've done subsequently or led subsequently to support scientists and researchers. So I commenced my scientific journey at the University of New South Wales, studying chemistry, in particular using a technique known as mass spectrometry to analyse large biological molecules. Mass spectrometry does that by analysing molecules in the gas phase and measuring their molecular mass, or more accurately, their mass to electric charge ratio, as a means of identifying or characterising those molecules. At the time I started my PhD, the challenge in analysing big fragile molecules such as proteins was we thought that we needed big machines to do that. But as it turned out, the real barrier was transferring these fragile molecules into the gas phase. Once that was solved, together with lots of other developments, mass spectrometry is now used as the underpinning technology in proteomics and a range of other analytical tools, such you might have heard of metabolomics, lipidomics, and so on. The work I did in my PhD was around how we could break these molecules up into different fragments as a way of identifying them. Subsequently to that, I moved on to several postdocs, and at the same time, John Fenn was starting to publish work where he was using a technique known as electrospray to ionize large molecules. And the trick with electrospray, or the beauty of it, was that instead of trying to force these molecules into the gas phase, what electrospray does is actually remove the solvent very quickly. And in doing so for these very large molecules, it produces multiply charged ions. And so because we're measuring the ratio of the mass to the charge, we could then use electrospray routinely with very conventional instrumentation, such as that shown here in my laboratory at the University of Wollongong. So then the scientific challenge became how we could do this and what we could, the kinds of systems we could analyse using electrospray. It was a very gentle technique. So early on, we found that we could analyse very large proteins, such as monoclonal antibodies. Later pushed that technology in the use of characterisation of synthetic polymers and a range of other different complex systems, and then moved on to studying fragile complexes that were held together very weakly, such as DNA duplexes or protein-protein and protein-DNA complexes. The latter part of that work I did in collaboration with Jenny Beck as I was moving into leadership roles at the University of Wollongong, initially as Dean of Science and later as Deputy Vice Chancellor of Research. From there, I moved to the Australian Research Council as CEO. And with the support of Minister Kim Carr at the time, we led a range of initiatives that have had an impact on research, both in terms of supporting research and science, but also many of these initiatives are still in place today. One of the early initiatives was internationalising the ARC schemes. At the time, there were a range of barriers to internationalisation, and we had the opportunity to remove them in one go. Various infrastructure initiatives, in particular, the support for the funding package for the synchrotron in 2012, which was absolutely critical to maintain that important facility. Spent quite a bit of time talking about how we could better engage with industry, a conversation that's still very much alive today. And as a consequence, we established the ARC Industrial Transformation Training Hubs and Training Centres. Support for a range of underrepresented researchers was also undertaken with a range of initiatives, in particular for Aboriginal and Torres Strait Islander colleagues. We developed the Discovery Indigenous Scheme and a network to support Indigenous research and knowledge. And finally, one of the last initiatives before I left the ARC was the support that the ARCs provided to the then Chief Scientist, Professor Ian Chubb, through ACOLA for a range of programs developing policies around the general theme of securing Australia's future. But one of the areas where I'm particularly proud of and particularly devoted an enormous amount of effort together with my colleagues at the ARC, was addressing the issue of gender differences in success rates in ARC schemes. And so early on, I was told that this was a problem that would sort itself out, that as the number of women moved into senior positions, their success rates would start to normalise, and it really wasn't something that I needed to worry about. However, being a scientist, I started digging into the data together with my colleague Andrew Wells, and we discovered that the difference in success rates was actually most profound, not at the later stages of academic careers, but rather at the early stages and in that period 0 to 5 years post-PhD. And so as a consequence, we reviewed the way that the ARC constructed its discovery scheme, and in particular the way that Australian postdoctoral fellowships were tied to senior chief investigators. And we separated out the early career researcher scheme and created the DECRA scheme in 2011. Not only did that have a difference and an effect on the success rate for different genders, but I think it's also been an important scheme in leading and seeding more independent early career researchers, which is important for Australia's future. We also looked at the way in which we evaluated track records, and as they were called at the time, and introduced the concept of research opportunity and performance evidence, taking into account different opportunities for research, career interruptions, and other factors that may impact on performance. So not just using simple metrics and one vision of a particular type of career that you get a track you get on and don't get off. And that concept of rope has actually now found its way into most Australian higher education institutions. We also, at the other end, at our most prestigious, the Laureate Scheme, looked at introducing and did introduce 2 named fellowships for women, in particular the for Science, Technology, Engineering and Maths, the Georgina Sweet Fellowship, named after one of Australia's earliest academic scientists from the University of Melbourne. The Georgina Sweet Scheme was designed to signal and encourage women to apply, as we know they tend to often hold back and wait till they're particularly ready or more certain about applying for grants. But we also added a component of funding to enable those that were given the award to undertake a range of activities of their choosing to promote other women or women in science more generally. And the variety of those schemes can be seen here when you see some of the very distinguished awardees and the way in which they use that fellowship to support early career researchers, high school students, other women through awards and grants. And I think it's a fabulous example of a small investment empowering very talented individuals, leading to great innovation and impact. I call out there Michelle Coote, who was my proposer. I'm very grateful for her nomination of me for the Academy, and also that she used her fellowship together with some other women in the Royal Australian Chemical Institute to establish several awards for women in chemistry. I've never asked Michelle, but I understand that possibly the idea behind some of the thinking around that was an article I wrote for the Australian Journal of Chemistry in 2011, pointing out that within the RACI scheme of awards, women had been far less successful and far less likely to be nominated. So Michelle and colleagues established the Margaret Shield Leadership Award in the RACI to recognise women scientists in part for that. The notion of recognition and reward and championing diversity, whether it's being in medals for my own discipline in mass spectrometry or whether it's in the images that surrounded me every day at the University of Melbourne, has been one that I've been passionate about throughout my career. And I congratulate the Academy on the steps that it's made to encourage women, both as fellows, but also in the various medals and prizes, the Academy Awards. So that's just a very brief intro into my career. And just a final note is that now that I've returned to now that I'm at QUT, I've actually managed to return to my roots and do a small amount of mass spectrometry when I have time. Thank you.
Please now welcome with us live Professor Margaret Shiel. Congratulations, Margaret, and thank you for a very inspiring presentation.
Thank you, Anna Marie, and thank you to the Academy. And it's a great honour to have been elected. Thank you.
Margaret, what was the most important skill from your research career that you brought into play during your time as ARC CEO?
I think that well, one of my colleagues in mass spectrometry said many years ago was infinite patience, because the early mass spectrometers that we worked on very rarely worked. So, you know, you spend a lot of time looking at a small oscilloscope, wishing for a signal. But the more, I think, significant skill was that in order to build that laboratory that I was able to build at the University of Wollongong and the instrumentation and so on, I really needed to learn how to collaborate with colleagues, because the funding schemes available at the time, and I always think this has been a strength of Australian science, required you to identify other people to collaborate with. And so that required me to talk to researchers, say, in the biological sciences school and in chemistry and outside the university, and sort of understand their science so I could see whether I could integrate it into a proposal for a new mass spectrometer. That I could then use both as a combination of collaborating with them and then also pushing forward the science. And that capacity to kind of probe other people's science and problems and frame them into, you know, grant proposals or funding schemes, that ultimately I think was the skill set that I took into the DVCR role and the ARC role. And I think it was, you know, whereas in America, for example, you know, I'd go into university departments, there'd be a mass spectrometer in everybody's own lab. And so they didn't have to do that. Yeah. And I think that's always been a strength for those of us who've had to have that kind of activity.
Thank you. Your research background led you to, quote, dig into the data around gender equity in academic careers during your time at the Australian Research Council. What was the biggest perception versus evidence divide that you encountered during that time? And perhaps more importantly, what do you see is the biggest divide now? What's the next frontier?
So in that particular case, so we had a very talented data analyst at the ARC, Young, and Andrew Wells, who was my colleague who worked on that. And the biggest difference in perception was that it was only a problem of the pipeline and women coming through to the senior levels. And I was told that over and over again. In fact, told not just leave it alone, you know, academia will catch up. And Andrew and Young, you know, went through all the data and all the evidence and said, this is not a problem of pipeline. This is an early career issue. This is women not having the same sponsorship, mentorship, other opportunities early in their career. And so that was the big divide there. And then the biggest divide today, I find, one of the things that I'm passionate about at the moment is trying to identify how you can choose and reward good mentors. So, you know, when we did the first era across the sector, and when I used to visit all the various universities in my time at the ARC, outside the GOA, where there was depth across most, say, science departments, yeah. When you went to a Wollongong where chemistry was really strong, there was a great leader or several great leaders, and in our case, it was the late Leon K. Macquarie. If you went to physics at Macquarie, Jim Piper was there, and physics at Macquarie was really strong, or nursing here at QUT, Patsy Yates. So what I'm doing a little project on the side with some colleagues and is to try and work out how do I identify those people who are good mentors and reward them and use that as a sort of promotion tool within universities and so on. And that's, I think, using H indices or broad data, you know, to reward people or, you know, dollars is not kind of, that's the gap, you know, getting focused on that. And I want really as a university leader or someone's trying to promote the next generation, find out who the leaders within my ambit are.
What a fantastic initiative. I look forward to learning more about it. It might become a pilot for something that could be rolled out nationally.
It is, in fact, once we get through the next stage, I am actually going to talk to Kathy and others. I've mentioned it to Kathy finally to see if there's something we can do there.
Terrific. Francis Saparovich writes, congratulations, Margaret. You mentioned the Australian synchrotron as a major item of infrastructure. If funding were not an issue, what do you see as a major item needed for Australian science?
I think Francis would be a better place to say that than me now. But look, the reason I mentioned the synchrotron was it was so touch and go whether we could save it at the time, and I don't know where you were in government in relation to that, Anne-Marie, but I just thought it was going to be this great tragedy for Australian science that we'd gone to and as did Kim, and it was with his support and many others, and Keith Nugent and others who sorted out and ANSTO. So I mentioned that in particular. I think I worry about my main, without picking a particular technology, the fact that now we are so reliant in the university sector for the infrastructure provided by universities through what was the funding stream of international students. That the Melbourne would not have been able to do the Doherty, which we've seen on the news every night, the Bio 21 and the facilities we built for CSL, the facilities I have here for hydrogen, all of that is built from that revenue. So I think the bigger challenge for me, Francis, is and Anna-Marie, is the total investment into research infrastructure and where that's going to come from.
Thank you. We've gone over time, but this final question is too important not to ask. What advice would you give, particularly to early career researchers from underrepresented groups, to turbocharge their career opportunities?
I think try and do something original. I think look for the opportunity. That was my postdoctoral advisor at Utah said that. Take everything you've done and find a niche. A bit like Simon said, address the market. Don't try and look for an original idea. And that's what I loved about the DECRA scheme. When we released those great young minds, we got much more interesting proposals in many ways because people took more risks at that early stage.
Thank you, Margaret. You have already an extraordinary legacy, and I very much look forward to seeing your further work at QUT and beyond. Congratulations once again on becoming Fellow of the Australian Academy of Science.
Thank you very much.
Now, please welcome back Professor John Shine to introduce our final new Fellow presentation.
Thank you. Our next fellow presentation is from Dr. Gregory Clark. Greg is a renowned scientist, technologist and businessman with an outstanding career both in Australia and internationally across a spectrum of industries from IT and communications to media, space and finance. His scientific contributions have been in the areas of solid-state physics, chip technology, neutrino physics, space technology, media digitization and cyber analysis. Greg has also continually supported student education and involvement. Now to Greg's presentation.
My name's Gregory Clark, and I want to start this presentation by saying what an honour it is to be elected as a Fellow of the Australian Academy of Science. In this talk, I want to cover a little of my scientific history and how that followed into a more commercial life. I have a BSc Honours degree from the University of Tasmania in physics. I then moved to a PhD at ANU in nuclear physics under Dr. Peter Tracy. The thesis I undertook was on excited states in beryllium-8 and oxygen-16. These states were critical in the evolution of light nuclei. It was a very fortunate time for me to be at ANU. We had absolutely outstanding standing leaders. We had Sir Ernest Titterton, who was head of the department, and we had Sir Mark Oliphant, who was the director of the school. These gentlemen attracted many outstanding physicists to ANU: Willie Fowler, Edward Teller, Tony Lane, Tom Tomborello, and so on. Very stimulating. And I think this made me certainly realise that Australian kids can contribute to science at any level. It was very important for me. Titterton and Oliphant and Baxter from University of New South Wales were very integrated in the Australian political world, which guided Australian policy in science and engineering and allowed us as scientists to build world-class facilities, such as the 14UD accelerator at the Australian National University, and others, such as ANSTO. Following ANU, I went to AERE Harwell as a Harwell Fellow. My nuclear work there continued under Australian physicist Dr. Joan Freeman in the study of beta decay. However, transformationally for me, I also moved into solid-state physics studies, particularly in ion implantation doping in semiconductors. After several years, my family wanted to return to Australia, and so I joined the CSIRO Division of Mineral Physics under Dr. Ken McCracken, a very productive time and a very productive group of people, with most of my time in uranium geophysics, a lot of it with Western Mining Corporation. However, I was attracted back to solid-state physics and the world around that area, and moved to Oak Ridge National Laboratory Solid-State Division. The biggest breakthrough I was involved in there was laser annealing for doping semiconductors, still used in industry today. We did this jointly with Bell Laboratories. Subsequently, I was recruited into the IBM Research Division in New York and spent 15 years there on silicon physics, thin films, and then a particular experiment to determine the mass of the neutrino, if indeed it had a mass. And we showed in that experiment that if it did have a mass, it had to be less than 20 electron volts, a good result. I was also involved in the fabrication of the first high-temperature superconductivity squid there. At that stage, IBM asked me to come to Australia and fulfil requirements of the Australian Government Offset Program, established by Senator Button, a very powerful stimuli for research in Australia. To get government contracts, companies had to invest in R&D in Australia. IBM invested in PC manufacturing plant located at Wangaratta, which turned out to be the best IBM PC plant in the world. And together with Alcatel and Siemens and Melbourne University, we established the ACCI near Melbourne University, a very successful research group that did a lot of work in software development, medical imaging analysis, and large data transmission. Another thing I was very proud of at that stage was that IBM put a supercomputer at AIMS near Townsville, the Australian Institute of Marine Science, and to model the flow of water through the reef, mangroves, and then out into the coral. AIMS won a major UN environmental award for this work. This time in Australia was very important to me as I realised how close pure R&D and commercial ventures were and how it was easy to integrate the two. I returned to IBM research laboratories to do research, but also spent time at IBM headquarters as Director of Telecommunication Initiatives. At this stage, I moved out of research and into corporate life. I moved to News Corporation, Fox Studios in LA, and became president of technology with worldwide responsibility for technologies and technology companies in those enterprises. We had something like 40 technology companies. It was exciting times in media as we moved from an analog system to digital systems. It took we took Fox Network digital, the first network in the US to go digital. We had pay TV evolution, digital sport, animation in movies, and so on. The most important company, from my perspective, was NDS, News Data Systems, out of the Weizmann Institute in Israel, under a man called Adi Shamir, who developed this technology for providing encryption and authentication for pay TV. A lot of wonderful people I worked with at that time, among them John O'Sullivan, who is a member of this academy and contributed greatly to the improvement and upgrading of pay TV quality and efficiency. At that stage, I became interested in satellites and took a job as the Chief Operating Officer of Space Systems Loral, based in New York, but with offices in Silicon Valley and around the world. It was the world's largest manufacturer of commercial satellites and the second or third largest operator. We had, again, 40 companies there, and while I was there, we launched over 90 satellites. One of these companies was Satmex, a satellite company in Mexico, which stimulated me to make a similar initiative in Australia to provide high-speed data, whether it be video or educational data or whatever, to rural and remote areas. With Space Systems Loral, I formed a company called KCOM, to provide data to rural and remote Australia. We had FCC filings for that, from the North Pole to the South Pole, the largest frequency filings in Region 4 for satellite operations. But then it didn't ever evolve into reality because NBN came along with their own satellite systems. So that didn't ever receive the outcomes that I had hoped for. Back in Australia now, I'm very happy with my life experience. I now look forward to contributing to the Academy's activities, and I have got some specific goals there, but I'm sure they'll change. Specifically, I'd like to see the Academy address more specifically and be proactive in major current issues that are facing Australia, such as communications, cybersecurity, and technical independence. I'd like to, secondly, bring back some of the Australian scientific diaspora as appropriate. We have something like 720,000 Australians working overseas, and 80+% of those people have degrees. The third thing I would like to start thinking about more under the auspices of the Academy are some of the big issues we have, such as water, southern oceans, a whole slew of these. So thank you for your time, and thank you for having me join your Academy. Thank you.
Now we're delighted to have with us Dr. Greg Clark. Congratulations, Greg, and thank you so much for joining us today.
Thank you.
So you noted in your talk about how Academy Fellows Sir Mark Oliphant, Ernest Titterton and Philip Baxter were right in the political world, which guided Australian policy in science and engineering. While noting theirs was a different time and context, in your view, are there lessons for our modern day fellows to take on board from the way these fellows influenced Australian science policy?
I certainly do think there are lessons to learn, and I think now is a crucial time for us to implement that. Those gentlemen had access to the highest levels of politics and could influence politics. And now I feel that we don't have that influence, or I'm not aware of it, to the degree we should. And just now, as we go to changing our policy to nuclear submarines, it's crucial that we as a group of scientists participate in that debate, particularly in areas such as training and educating nuclear engineers and nuclear physicists to implement and take over those roles. I think if those gentlemen were here now, they'd be all over this issue, and we would be playing a role both behind the scenes, but also publicly about what we should be doing. And they were so well connected internationally that we would be bringing people here who could help us in this training program.
There are certainly many big challenges before us, and it's difficult to imagine any one of them not requiring science and technology to inform its way forward and the solutions to them. So the more we can do in that regard, the better. Yeah. Could I take you to your own field of expertise? Solid-state physics was obviously a big passion of yours during your career. What drew you to this area of physics?
It was almost accidental in that when I finished my PhD at ANU, I went to AER Harwell, as I mentioned in my talk, and there was a use of accelerators there for ion implantation, and also around the world was just gaining momentum. So I worked with a man called Jeff Durnley and FRS, and we were very successful in a number of different areas using implantation. Some of that work we did with the University of Bologna and other Italian groups, and we integrated into what was occurring with some of the big chip fabrication companies around the world. So it was just it was almost it was a juxtaposition of nice events.
Yeah, right. You said your time in Australia made you realise how close pure R&D and commercial ventures were, and how easily it is to integrate the two. Australia has a strong research base, but our track record in translating research outputs into economic benefits could be improved. What are your views on this?
I certainly think it's getting better, and as we look around now, I'm quite amazed at the number of startup companies that I see that have 25 to 40-year-old, and that gives me great satisfaction occurring there. I think that there is a opportunity to integrate more with the large companies that do sell product here, whether they be a media company or an internet company or whatever. And then I think there's a greater opportunity to integrate our research with them than we do now. And that's why I like the initiative that Senator Button undertook to really force international companies, particularly those of size, to make use and do work here in Australia if they were to get business contracts. And I think that could still occur. I don't know why we dropped that.
Yeah, it's a good point. Certainly worth revisiting. One of the challenges that you've cast for yourself and for the nation is that you're keen to bring back some of the Australian scientific diaspora who are currently working overseas. Right. What incentives need to be in place for that to happen, and what are the benefits?
The benefits are that I think it's good now that we have a lot of international scientists coming and working here and basing themselves here, and I think we benefit from that to a huge degree. But it always amazes me as I move around the world, the number of Australians with high skill base that I run into. And I'm aware that there's something like 800,000 Australians working overseas. I've got no basis for these numbers, but I think they're pretty accurate. And something like 80% of those have degrees, and something like 50% of those have engineering or science degrees. And many of them would like to come back here. It's getting a suitable job. So I think it's mainly just getting companies up and running and having momentum. I think we've got a lot of universities and that are starting to employ, or were until recent economics stages, starting to employ more people. But I think they've got to go into the commercial environment rather than just the pure academic environment. So then I think it's just a matter of us using those skills and making products here in Australia, whether they be software or hardware, that can be exported internationally.
That's all we've got time for. I'd like to thank you so much, Greg. And again, congratulations on becoming a Fellow of the Australian Academy of Science. We're absolutely delighted at that and at the opportunity to work more closely with you going forward. Thanks ever so much.
Thank you.
That now brings us to the end of 2 deeply inspiring days of presentations from the Academy's newly elected Fellows. They had offered us a rare opportunity to look into the depth and breadth of science in Australia across so many disciplines. Thank you, our audience, for joining us and for all your questions throughout the day. We hope you've enjoyed hearing from our Fellows. This event also brings us to the end of Science at the Shine Dome 2021. So thank you to everyone who has joined us throughout the year to celebrate excellence in Australian science. I'd like to particularly thank our sponsors for supporting excellence and supporting the Academy. My gratitude also goes to the talented staff across the Australian Academy of Science who have delivered Science at the Shine Dome 2021 in an online format, working through the various challenges that has delivered. With particular thanks to Paul Richards, Dr. Tom Carruthers, Lisa Croker, Olivia Condon, Tim Newport and Luke Buckle. And of course, to the Academy's Council, Executive Committee and Fellowship. You are the Academy, a true national treasure that inspires the nation, and we wouldn't be here without you. All of the Science at the Shine Dome events have been recorded and will be available on our website. Be sure to revisit them and share them with others, and look at the remarkable science that we have shared and celebrated over the course of 2021. My name is Anna Maria Arabia, signing off from the Shine Dome in Ngunnawal country. Good afternoon.